We're almost at the top of the hour. You probably should start on time, and I'll let you lead it off. We have 50 participants, but more I'm sure will join as we're going. We did have around 300 sign up for this, and I suspect that a lot of them will be arriving just on time. So whenever you're ready.
Yep. Thank you, Jamie. Good morning, everyone. My name is Wang, and I'm from AbbVie, and I'm the current chair of ICSA Midwest chapter. It's my great pleasure here today on behalf of my chapter and our collaborator from NIC ASA to welcome you all to the 7th ICSA Midwest chapter and NIC ASA joint fall meeting. Just to give you some background or history of this meeting. This local meeting started back in 2015 in the meeting room in AbbVie Park with just less than 40 participants. But over the past several years, thanks to the hard work of all the volunteers from both chapters and the organizing committees from different companies, including AbbVie, Astellas, Abbott, Takeda at that time, Baxter, Eli Lilly, and lots of universities, including Northwestern University, UIUC, and Wisconsin-Madison. So over the past years, the meeting gradually evolved into a multi-hundred participants local meeting with multiple sponsors. And we added a lot of more freebies like free cocktail session, or the war session, free caricature, and student poster awards. So lots of fun happened after that. Because COVID, we have to change it to virtual format in 2020 and 2021. Although we lost lots of the face-to-face fun activities and interactions and network opportunities, we can outreach more now to invite more prominent speakers across the country due to this convenience of virtual format. So this year, as Jamie mentioned, we have about 300 registrants for this meeting, which is record high. I'm really proud of what we have achieved so far. And I want to express my sincere gratitude to all of our prestigious speakers from industry, from academia, from FDA, especially our keynote speaker, Dr. Yaning Wang, for accepting our invitation to share the newest research and use cases in the clinical trials, regulatory submission approval, real-world evidence, and the machine learning big data. I also want to thank all the volunteers to make this happen this year. It won't happen without your hard work. So before I turn the stage to Jamie, the president of NIC ASA chapter, I want to announce that after 5 years of service, I will pass my chair position to my colleague Xiaohong Huang from AbbVie in 2022. But I'm not leaving the chapter yet. I will serve as the past chair for 3 years so that I can continue to contribute to the chapter and joint meetings. Okay, so that's all I have to say from my chapter's perspective. Jamie, back to you.
So thank you, Lee, and it's been a pleasure to work with the ICSA to organize these sessions. It's a little unfortunate that we couldn't meet in person the last couple of years, and I'm looking forward to the day when that is able to happen again. So in the ICSA, we have a president, past president, and a president-elect, and the term for the president is intended to be 2 years. And so my 2 years are now up. We've had one nomination come in for the president-elect, who is Jung Park from Astellas, which is the company from which I also come. And we have a current president-elect, Surab, and I'm unfortunately poor at pronouncing Surab's last name, but Mukhopadhyaya, I think I've messed it up, but he will become president next year and will be the primary organizer for next year's conference. Usually we do these over 2 days, but as we've been making them virtual, it's only been a single day. And we usually have some short courses the first day. So all of you who've registered will become part of our mailing list. And so you'll get messages about what we're planning on doing and what's going on. So you'll hear about that. Just a couple of things in terms of how we're going to do things. We do have some breaks scheduled. The meeting will continue. The Zoom meeting will continue while we're on break. We are recording the session, and that recording will appear at some point on the NIC ASA chapter page in the ASA. So you can come and listen to sessions that you've missed or want to hear again, or share them with your colleagues if you wish. That should be available to everyone. There is a chat function, and at the moment, all of you will have been muted and your videos will be turned off. You are able to turn them back on, and so if you start to ask the question, you want to do it with your voice, that's fine. You also are welcome to put questions into the chat, and we will monitor that and make sure that we are able to get that to our speakers as well. I do ask the speakers, if you will, to put yourself on video when you're doing your presentation, and we will set it up so that you can share. So hopefully things go well. We don't have the budget to have professional folks looking after our Zoom meeting, but worked really well last year, and we're looking forward to a great meeting today. As Lee says, we have some really excellent speakers. Thanks for all who volunteered to actually present to us, and I'll let Lee introduce his plenary session, and I'm going to stop sharing the program.
Yeah, thank you, Jamie. I see Yaning is already online, so maybe Yaning, you can use this time to check your sharing function. And then I will formally introduce our keynote speaker. Great, Yaning, we can see it. Yeah.
Can you hear me too?
Yeah, I can hear you too. Yeah. And we are like 3 minutes earlier than the schedule. I believe people are joining now. We have 99 participants, and as I said, we have 300. Hopefully, gradually they will dial in. So Jamie, do you want to start now or wait 3 more minutes? Maybe people, they're pretty.
I think it's actually probably slightly better to wait the 3 minutes start right on time.
Exactly. Yeah. Maybe we can. So your term is two years instead of 3? I thought you were 3, right?
This was your advice, Lee. It used to be just 1, and you said you should do it for 2 so that you get to do the organizing at least 1.
I thought we end up with 3. Maybe I didn't remember clearly.
In some sense, it's really 6, because president-elect, president, past president really is a 6-year term. So. Oh yeah. Now I remember. Yes. Yeah. So, Dr. Yaning, are you in East Coast? Or
yes, I'm in Maryland.
Maryland. Okay. Great. So it's not Chinese time. Okay. And the company is called Creterna?
Yeah. Creterna Science and Technology. Yeah, Creterna.
Okay, Creterna. Just want to make sure I pronounce it clearly. Okay, Creterna. And since I did some commercial or advertisement of this meeting on LinkedIn, I realized that from registrants list, we have lots of people joining. That's not local. Typically this meeting is a local meeting. The participants are mainly from local companies, local universities. But now I saw East Coast registrants, West Coast registrants, and someone who's not even in the pharmaceutical business. So that's really exciting. Thanks for all. Yaning, I think your presentation may attract lots of participants for this talk. Okay. I think we're almost 8:10. Okay.
I would say go ahead.
Yeah,
Thanks.
let's do it, I think. So morning, everyone, again. Now it's my great pleasure to introduce our prestigious keynote speaker, Dr. Yaning Wang, to share his firsthand experience in the accelerated approval, the controversial accelerated approval of Biogen's Alzheimer drug, Aducanumab. And his title of the talk is Misconception Effects of Aducanumab approval. And Dr. Yaning Wang is a former director of a division of Pharmacometrics in CEDAR FDA. And congratulations to Yaning. He's now the current CEO of Creatinor Science and Technology. That's quite a change. Congratulations. He was in the division of Pharmacometrics in the Office of Clinical Pharmacology at FDA until September 2021 and oversaw reviews, research projects, and policy development within the division of Pharmacometrics for all disease areas. Dr. Wang received his PhD in pharmaceuticals and a master's degree in statistics from University of Florida in 2003. He also obtained a master's degree in biochemistry from National Doping Control Center and a bachelor degree in pharmacy from Peking University in China. Dr. Wang has published over 100 papers and given over 280 presentations at various national and international meetings. He served as a board member of the International Society of Pharmacometrics and is a fellow of ISOP. He's a member of the advisory committee for Chinese Pharmacometrics Society and a member of the editorial advisory board for the Journal of Pharmacokinetics and Pharmacodynamics. So this is a great topic, and I'm so looking forward to hear more story behind it. So thank you, Dr. Yaning Wang, and the floor is yours.
Thank you. Thank you for the nice introduction. And of course, thank you for inviting me to talk about this very interesting case. In fact, this is my last review before I left FDA last month. I believe this is the first meeting in this country where I can publicly discuss the details about the review and the regulatory decision-making process. I believe this is long overdue because there's so much misinformation about this case out there. Therefore, I hope today I can set the record straight by explaining facts and clarifying all the misconceptions about the Aducanumab approval. But before I start, I want to show my highest respect to this FDA review team for Aducanumab, because people should remember them as a group of heroes. They are absolutely heroes because they did not give in to the pressures from all sources. Instead, they applied the best regular science and analyzed the precious data in the most expensive way. And most importantly, they listened to the patient's voice and finally approved this safe and effective drug timely to benefit millions of patients. I want you to remember all these names. They are heroes. Once again, I want to repeat that. Now let's start. So this is the program timeline. As you can see, there are phase 1, 2, 3 studies, but today we will focus on the 3 studies that are related to the efficacy. 103 in the middle is a phase 2 dose ranging study. And then 301, 302 are the 2 pivotal phase 3 trials that started almost at the same time with the same design. This is a snapshot of the entire regulatory interaction history between FDA and the sponsor. And of course, you already read a lot of news in the media about how FDA interacted with the industry, but this is the FDA side story about how we interacted with the sponsor. So in December 2014, at the end of phase 2 meeting, the sponsor discussed with FDA about the design of the 2 pivotal phase 3 trials by using a special protocol assessment procedure at FDA. This is the highest level of agreement between the sponsor and FDA, so that we agree on some key features such as study population, endpoint, and dosing, so that we don't easily change all this, after the trial is finished. And I want to emphasize that at the design stage, the effect size assumed to power the study was agreed between FDA and the sponsor. And of course, not all special protocol assessment agreement can be reached between sponsor and FDA, but for this one, in September 2015, FDA and the sponsor reached agreement on the special protocol assessment for both studies 301 and 302. Then the trials started, both the 2 phase 3 trials. Unfortunately, in March 2019, the sponsor announced the termination of the phase 3 program based on the results of a pre-specified interim futility analysis. But after this announcement, the sponsor realized there was signal in the data sets to support the drug's effectiveness. So the sponsor requested a meeting in May of 2019 to have a meeting with FDA. This meeting happened in June 2019. At this meeting, FDA and the sponsor discussed the preliminary analysis of the existing data and agreed that this drug or this program should not be abandoned because the available clinical data suggested the drug may be clinically active and did not provide convincing evidence that the drug is ineffective. And the most important part is to explain conflicting results or partially conflicting results between the 2 studies. And the sponsor went back and did more extensive analysis and came back in October 2019. At this meeting, the FDA Neurology Division agreed the results of the 2 studies are interpretable and suitable for additional consideration, and therefore recommend the sponsor to submit this as a formal BLA. Then in July 2020, the submission was finished, and we started our review under the priority review. Then in November 2020, AC meeting happened through an online meeting. Unfortunately, as you can see, the votes were very negative. Out of 11 AC members, 10 members voted no for approval of the drug, for approving the drug, and 1 member was abstain. After this meeting, in January 2021, the sponsor submitted additional data to this BLA, and as a result, the PDUFA or the time clock was extended by 3 months. So the PDUFA date was changed from the original March date to the June 7th, the new PDUFA date. Then during this process, 2 additional internal meetings, we call that medical policy and program review council meetings. These are essentially the internal advisory meetings with the FDA to discuss this case. And after that, another center director briefing meeting happened in April before FDA finally granted accelerated approval to this drug. So this is the quick summary of the regulatory history. And I will start with the early termination decision by the sponsor. And again, all the information I'm presenting today is in the public domain, and I provide a link at the bottom of each slide. In fact, this information was already presented at the AC meeting because even the sponsor agreed. When they made the early termination decision, there were 2 very important assumptions. The first, and this, of course, this early termination was based on the conditional power, assuming the future unobserved effect would be equal to the observed effect at that time. And the second one is pooling the study results from the 2 studies, also assumed the results from the 2 studies should be similar. Under these two assumptions, then the studies were to be considered futile if both studies have conditional power less than 20% for both the high dose and the low dose. And the statistical analysis plan also specified other data in addition to this pre-specified futility criteria should also be considered. But let's look at the actual data. Yes, at the design stage, people assumed these are two identically designed studies and their results should be similar. But let's look at what actually happened. So these are the data at the interim analysis. As you can see, there were two doses within each study, low dose and high dose. If you pay attention to the low dose and the high dose from 302, all those three arms showed positive signal, suggesting the drug is slowing down the disease. The minus percentage in the bracket suggests the percentage of slowing down the disease quantified by the CDR-SB, primary efficacy endpoint. The only exception is the high dose in 301 study. In fact, this high dose not only did not improve the disease, in fact, it is significantly much worse than placebo because it's 15% worse than placebo. And clearly, based on this observed data, no one will agree the results are similar between these two studies. That's why this one of the key assumptions was not met based on the actual data. And if we analyze this, since they are different, so the studies should be analyzed independently. If we analyze these two studies independently because of the different results, then the high dose in the 302 could have reached a power almost 60%, way beyond the 20% power criteria. Therefore, the study should not have been terminated if the data had been analyzed this way. And the second assumption is that effect size should have remained the same in the unobserved future results at that time. But when you look at these results, the first row was the results at the time of interim analysis, and the second row is the results at the final analysis. As you can see, especially in the 301 high dose, the results improved significantly, suggesting the second half or the results after the interim analysis clearly showed the high dose was better than the placebo. Otherwise, you could not have reached this almost equal result at the end. So the second assumption was also not met. And even in the 302, the high dose also improved over time. That's why both assumptions were not met. But people can argue, how could we know this assumption was not met at the time? Because we had not collected the future data yet. That's understandable. But we could have checked the results for the consistency or similarity assumption at the time of the futility analysis because all the data were available. Anyway, so let's get into the detail of the studies. So the first very important study is this dose ranging study 103. So yes, this study's primary endpoint was safety and tolerability, but two very important efficacy endpoint, including the primary efficacy endpoint, CDR-SB, were monitored for over a year. That's why the efficacy results are fully interpretable under this randomized double-blinded study. 5 different dosing regimens were studied against placebo. And here I want to specifically mention this last one. That's the titration within the ApoE4 carrier from 1 milligram up to 10 milligram per kilo, because this arm played a critical role in a later protocol revision for the 2 pivotal studies. Because during this study, they found that as long as we do a slow titration to increase dose within the ApoE4 positive patients, the safety could be very, very well controlled. And this is the study design for the 2 pivotal trials, 301 and 302. Like I mentioned, initially, and there are 2 doses, low dose and high dose against placebo. So based on the ApoE status, initially, the low dose allowed 3 milligram for the ApoE positive patients and 6 milligram for the ApoE negative patients. So there was this prior knowledge, ApoE4 positive patients had a higher risk for AE under the same dose. That's why they end up with a lower dose, even though they are in the same low dose group. Similarly, for the high dose group, initially, the ApoE4 positive patients could only titrate up to 6 milligram. Remember, their low dose was only 3 milligram. And the ApoE4 negative patients, their high dose could reach 10 milligram relative to 6 milligram in the low dose. But once the sponsor realized the titration regimen could control the AE, so in the middle of the trial, the high dose for the ApoE4 positive patients were changed or increased from the originally planned 6 milligram to 10 milligram. So now after this revision, or we call that protocol version 4, the high dose is the same between the ApoE4 positive or negative patients. So this is a very important change I will keep referring to in my later slides as pre-PV4 and post-PV4, so you can remember what happened at this time point. So this is the top line results for the 2 studies. And the first row is the primary efficacy endpoint, CDR-SB, and the next 3 rows are the 3 secondary clinical endpoints. And I think most people, including the AC members, pay a lot of attention to the 2 numbers in the squares on the slides. That is the high dose for both 302 and 301. As we can see, just based on the p-value, 302 was successful for the high dose because the p-value was 0.012, but the 301 study failed for the high dose. As I explained earlier, the effect size is essentially 0. And again, if you only pay attention to these 2 numbers, you can argue, yes, one trial was successful, one trial failed. But remember, there were low dose and 3 other endpoints. Yes, there was a pre-specified test sequence, and we're all statisticians today. And I'll explain the test sequence to control the type 1 error. But if we look at the data in the totality, and you can see from the low dose part, two trials are very consistent. And even for the other secondary endpoint, especially the ADAS-Cog and the ADCS, the average daily activity, those 2 endpoints used to be the co-primary endpoints for other Alzheimer's drugs. The reason we did not use these 2 is because these patients are very mild patients, early stage AD patients. And those 2 endpoints used to be used to prove drugs for moderate or severe AD patients in the past. So we thought these 2 endpoints may not be sensitive enough for this very early stage patients. That's why the CDR-SB was actually a new endpoint. FDA even published a guidance on this, thinking this may be a more sensitive endpoint, especially for this early AD patients. But it turned out that the traditional primary endpoints were still sensitive enough to capture the benefit of both the low dose and high dose in both studies. That's why on the title, we say these 2 studies are only partially discordant. In fact, you'll be surprised how much more consistency we can find when we analyze data in a more extensive way. Next, I will focus on the clinical pharmacology principle that we used to support our full approval recommendation. Yes, FDA eventually applied accelerated approval to prove this drug, but within our review, in fact, also within the clinical review, we recommended full approval without requesting any additional trial. And I will explain our rationales here. So there are 3 key points to support clinical pharmacology approval. The first one is the positive exposure response relationship for multiple clinical endpoints from all clinical trials. Here we used the individual patient concentration measured over time when the dose was titrated up. And here the response was also slightly different from the primary statistical analysis. We used the disease progression as our response. And the second is the positive exposure A beta, or the biomarker relationship quantified by this SUVR, and the consistent SUVR clinical endpoint relationship across multiple compounds with similar mechanism of action. In fact, at the AC meeting, one of the key points the AC members argued was there were so many failures before this drug. And some of those failed drugs also improved A beta level, but they did not show clinical benefit. How come this drug succeeded? So we were trying to answer this question by collecting all those compounds to show why this drug is different from the previous failures. And the last point is also based on a request from one of the AC members, because at the AC meeting, because of the pandemic situation, we had the first online meeting. And some of the presentations, including the clinical presentation and the statistical presentations, were pre-recorded and put on the FDA network before AC meeting, so that the AC members and the public can review before the meeting. So those presentations were not broadcast at the day of the AC meeting. And that left the impression to the AC members, FDA only emphasized the positive information for this drug, or we assumed the 302 results was the truth and tried to look for reasons to explain why 301 failed. So one of the AC members requested, why don't you make the opposite assumption, assuming 301, the failed result represented the truth, meaning the drug was not effective, and then evaluate the chance of seeing the positive result in 302. So that's the background for this third bullet point. So in fact, as you can see, we took the AC members' feedback very seriously. Not like what they said, FDA ignored the AC members' opinion and all that. No, we took their recommendations very seriously. We collected more data and we did much more extensive analysis to answer all those questions. In fact, after doing the requested additional analysis, we became more confident, even than when we were at the AC meeting, about the drug's effectiveness. That's why we made the final decision. It's not we ignored their recommendation. We took their recommendation seriously and confirmed our belief. So let's look at the first point. Like I said, the exposure is the longitudinal drug concentration within each individual, taking the dose titration into consideration. And the response included the 3 key clinical endpoints at all visits, not just the last visit. And in addition, we applied a natural disease model for the placebo arms, which I will explain in detail in the next slide, because this is a very critical point to explain why the pharmacometrics analysis or the exposure response analysis reached a different conclusion than the primary statistical analysis. And finally, our response is the slope of disease progression on a larger scale. The reason we did the larger scale transformation for the clinical endpoint is all the clinical endpoints are scores. They have lower and upper limits. So we did the transformation to make them more normal. But here is the study stratified results based on the exposure response analysis. Here response is disease progression reduction. As you can see, no matter whether it's 301 or 302, the x-axis is the dose, placebo, low dose, high dose, and the y-axis is the median disease progression slope. So different from the primary statistical analysis, both studies showed positive dose response or exposure response, suggesting higher the exposure, better the response. And that's true even for 301. But as you can see, the magnitude of the effect size is clearly smaller than in 301 compared to 302. Let's look at why that's the case. Remember, there was this very critical revision in the middle of the trial. So if we split the trial into 2 parts, pre-PV4, post-PV4 for both studies, now you can clearly see why the 301 results looked different from the 302. It's mainly because the pre-PV4 period. Here, as you can see, even when we applied the exposure response, The data from the pre-PV4-301 also showed a lack of dose response or exposure response. And here the high dose is, again, not only worse than the low dose, and numerically it's even worse than placebo. Just like the primary statistical analysis. But if you look at the post-PV4 in 301, there is this very positive dose response that's entirely consistent with the 302. Then you may ask, how come when we combine the pre-PV4 and post-PV4 in 301 under the exposure response, then we can reach the plot on the left. So intuitively, when you combine these 2 points, it should reach somewhere in the middle. That's how you end up with the results like this on the left. The high dose should still be better than low dose and the placebo. But in the statistical analysis, you have seen the high dose, even the overall average, is worse than placebo. What's the reason between these two analyses? And this is one of the main reasons. Remember, I said we applied a natural disease model, again, based on the observed data from the placebo arm. You can see over time, that's the x-axis, the largely transformed CDR-SB follows almost a perfect straight line. And the exposure response modeling approach took advantage of this underlying relationship. Why is this important? Remember, I said after post-PV4, the termination happened in March of 2019. So almost half of the patients within this post-PV4 did not reach the last visit, that is the 78 week. So there's a lot of missing data for the post-PV4. In contrast, the pre-PV4 period, most of the patients reached the last visit. So those patients have observed data at the last visit. For the statistical analysis, it applied this MMRM approach without assuming any underlying relationship over time. But in the pharmacometric analysis, we assume this linear relationship over time, which is strongly supported by the data. By the way, the model did not start with a linear model without testing other structures. The model pre-specified multiple structures to test the time dependency, and ultimately the data picked the linear model because of what you see on the screen. So once you have this linear assumption, there's not too much problem if we don't have the last visit, because as long as you have the first 3 visits, the linear model will easily impute the last visit. That's why the relative weight between the 2 periods under the exposure response analysis almost is equal. But in the statistical primary analysis, the weight from the pre-PV4 dominated the final average. So that's the main reason between the 2 analyses. Again, we apply the same approach to other endpoints. Like I said, for ADAS-Cog and the other clinical endpoints, even the statistical primary analysis showed consistency between the studies. That's why you see even the exposure response showed a similar pattern. But even this endpoint was influenced by the pre-PV4. As you can see, the dose response was not consistent, even for this endpoint. But the overall average, and most important, the post-PV4 from the study 301 was entirely consistent with 302. Similarly, for the ADCS, average daily life living endpoints, once again, it's highly consistent, and this is also consistent with the statistical analysis. So far, I presented you the exposure response pharmacometric analysis. Next, I will present the results from the primary statistical analysis, but in a different way, okay, without assuming all this linear model or using the concentration, the individual concentration. But I want to present it in a different way rather than just the table and focusing only on those 2 numbers. So I present it in a longitudinal way and putting all the studies side by side, stratified by the randomized dose. And the left 5 panels are from the 5 doses studied in the 103. The middle 4 panels are the 2 arms separated by the pre and post PV4 for 301, and the last 4 are for 302. So in total, we have 13 panels. Of course, these are not independent panels. Within the same study, even though they are randomized different doses, but they are still correlated because they are compared with the same placebo group. So they are correlated, but they contain a lot of independent information. So here, any dots above 0 means the drug is better than placebo. As we can see, among 13 panels, only one panel, that is the 301 high dose pre-PV4, did not support the drug's effectiveness. All the other 12 panels are supporting the drug's effectiveness. And people may ask, why is this panel so different? And of course, at the AC meeting, and also in the review, the sponsor spent a lot of time trying to figure out why this happened. And clearly, from a common sense perspective, this looks very much like an outlier. But why? Sponsor initially came up with 2 reasons. The first is the dose. Remember, before pre-PV4, the high dose used the low dose, for the ApoE4 positive. And the second reason is, even though these are randomized trials, but because of bad luck, the high dose group ended up with a higher percentage of what the sponsor called rapid progressors. Those are patients who progressed very fast after randomization, no matter which drug they're taking or which arm they're taking. So unfortunately, the high dose ended up with a higher percent of fast progressors. But when we did all the sensitivity analysis, these 2 reasons, low dose and fast progressor high percentage, could not fully explain this very unusual results. Remember, this is significantly worse than placebo. So in our review, in the clinical pharmacology review, we believe type 2 error is another factor here. Yes, at FDA, we pay a lot of attention to type 1 error. But in this case, we should also pay attention to type 2 error because there are so many tests, different doses, different trials, different periods, and different time visits to test this efficacious drug again and again. And when we test the efficacious drug, this is a statistical group. I don't have to explain what is type 2 error. When we test an efficacious drug many times, some of them should fail. Even if you power the phase 3 by 90%, that basically means we allow 10% type 2 error. So if you have 10 chances to test the efficacious drug with 90% power, one of them should fail. That is expected. So people should not be surprised that one of these 13 panels did not follow the overall pattern. That should be expected. Yes, the magnitude of not supporting drug is extreme, but this is what the type 2 error exactly means. And in fact, if all of them are successful, I'd be surprised. Or the only word to explain everything consistent is that's extremely good luck. In fact, this is normal. When we look at the same data, in the same way across the other endpoint, of course, this endpoint was not measured in 103, so we only have 8 panels. The similar pattern showed, okay? Only the 301 high dose showed this outlier behavior. Same thing for the other endpoints. So with all this, I want to say, no matter whether we use exposure response analysis or we just stick with the primary analysis of the statistical analysis, we will reach the same conclusion. Across multiple endpoints, across multiple trials, the clinical endpoint itself is already very convinced to support the drug's effectiveness. Okay, let's move on to the second one. The drug's mechanism is to reduce A-beta level, which is quantified by SUVR. So we also explored the exposure-SUVR relationship. In fact, this one, there was not too much controversy. Even the AC members agreed. There's a clear evidence from all 3 studies. The drug reduced A-beta level without any outlier. But the key question is whether reducing A-beta will lead to clinical benefits. That's the controversial part. So that's the second part. And I'm showing you the exposure response for SUVR on the left. Again, there's no controversial, everyone agreed. The key controversial or disagreement part is the plot on the right. So the sponsor present this SUVR versus CDR-SB plot at the AC meeting by combining the data from all the 3 studies and all the doses. On the x-axis, this is the change from placebo for SUVR and the y-axis is improvement of the clinical endpoint. As you can see, again, just look at it intuitively. Most of the data support a positive relationship. It's almost linear between these two endpoints. The only exception is the 301 high dose. Again, if you apply common sense or even the most rigorous statistics, I would argue this data or this plot clearly demonstrates 301 high dose is an outlier. I think any reasonable person will not believe the 301 high dose represent the truth and everything else represent a false positive. But again, this was not able to convince the AC members. That's why after the AC meeting, we collected all the data from all the previous failed antibodies and also other ongoing trials to look at the same relationship between these two endpoints. And the requirement is all the trials should have very high quality data, randomized double-blind data with at least 1 year period. These are the drugs we found. The top 3 in the green box are the 3 drugs that are promising. Of course, the first one is Aducanumab. You have seen the data. The other 2 drugs, BAN2401 and the drug from Eli Lilly, those 2 drugs, at the time of review, we already have public information about the phase 2 results. Then in the middle is the Roche drug, Gantenerumab. This drug failed 2 large phase 3 trials, but the program was not terminated. In fact, the sponsor analyzed data and realized the doses were too low, and as a result, the A-beta level reduction was not enough. So the sponsor increased dose by almost 10-fold to continue the study. Bottom, the 3 drugs, they failed the phase 3 trials and the programs were terminated. When we put their data together on the same plot, again, the biomarker versus clinical endpoint relationship. The black dots are for the Aducanumab 3 studies. Orange triangle are for the 2401. As you can see, these are 2 entirely different compounds, but they follow almost exactly the same relationship. And the other drug, Lilly drug, we did not find publicly available information for the SUVR data, but we found another way of expressing the A-beta, that's called the centroid. So again, when we put the Lilly phase 2 data results on the same plot, again, it followed the same path. But the most important part is when we put all the failed drugs on this same plot, now the reason for the failure is very obvious. As you can see in this group of failed drugs, the green square or the cross and all the signs, yes, some of them did reduce the SUVR or A-beta, but the magnitude of reduction of the biomarker was so little. It's pretty much below 0.1 units. That's the reason why all these drugs failed, because their reduction of the biomarker or the target was not clinical meaningful, was too little. Yes, some of them reached statistical significance because of the large sample size, but it was not large enough to trigger clinical meaningful improvements on the y-axis. In other words, for the next drug, if you can find a safe dose that can push this biomarker into this region, the chances of triggering clinical meaningful improvements on the CDR-SB or other clinical endpoint will be much higher. So I think this plot clearly explains why Aducanumab is different from the other drugs and also is similar, because they all follow the common biomarker clinical endpoint relationship. It's just where they are on this curve in terms of their dose. You know, if these drugs are safe enough, we can push the dose high enough to reach this region. And I believe based on this evidence, their clinical endpoint should succeed as well. So that's our second supporting point. The last one, again, these are, you guys are statisticians. I don't have to go through this in details because I prepare this for physicians, for others who don't understand it, the null hypothesis, you know, this false positive finding. Since we have placebo data, we can easily do a lot of virtual clinical trials by sample with replacement from the placebo group. then randomize those individuals to low dose, high dose, placebo. This is truly ineffective drug because these are all coming from the placebo arm. Then to maintain the correlation of all 4 clinical endpoints over all time points, we randomize individual as a whole. Then we analyze data with exact same statistical analysis, MMRM, and we quantify what is the chance of observing the positive result as in 301, 302, if the drug is just like placebo. And these are the results we should see, okay, under this null hypothesis. As you can see, no matter which endpoint we're looking at, the 4 endpoints, or any one of the dose, or any one of the 3 time points, they should all be around 0. And this also highlights, just based on chance, some of the placebo corrected effect size can go to the extreme positive side or the extreme negative side. Either side can happen. But by average, half of the points should be above 0 and half the points should be below 0 if the drug is truly just like a placebo. The same thing for 103. But what actually happened is this. Again, I presented similar way, but it may look messy to your eyes. So let's just focus on the numbers at the bottom. I just counted, okay? Out of this 301, we have 4 different endpoints, 2 different doses, 3 different time points. I'm not emphasizing these are 24 independent assessments. They are all correlated. But out of these 24 different combinations, 18 points are above 0, supporting the drug's effectiveness. And for study 302, 23 out of 24 points are above 0. And for 103, 17 out of 20. This is not even close to half and a half. That's why if the drug is just like placebo, intuitively, what do you think of the chance of observing this? So when we did the detailed analysis, the number came back to be the false positive chance for 302 is less than 1 out of 10 million. We did not even calculate the accurate number because our competition was not fast enough to conduct more than 10 million virtual clinical trials and analyze them the same way using MMRM. It was very time consuming. So we stopped at 10 million. We felt that was small enough. Even for 301, the false positive chance is 1 out of 10,000. Why do you think 301 has false positive results? Because remember, we look at all the data, not just the one number from the high dose to CRSP. In fact, 75% of the data are positive, even for 301. And similarly, when we analyze the false positive rate for the 103 with very small sample size, but because it has 5 different doses, 2 different endpoints, and 2 visits, the chance of seeing what you see in 301 is also 1 out of 10,000. So when you combine all these 3 small false positive chance, I can almost argue it's practically impossible to see the 3 results overall positive finding if the drug is not effective. And finally, I want to, this is not for the statistician, but this is for the physicians, because I think this presentation is recorded. I want the experts from the outside to see this one, because we heard this a lot when we present these results. Some physicians or experts believe since the CRSP ranged from 0 to 18, it's a very, very wide range, we should have at least 1 or 2 units to be clinical meaningful. But I would argue these experts do not fully understand the trial design and the included patient population. Here, I want to explain what happened at the design stage. Remember, we reached special protocol assessment agreement at the design stage. So even at that time, FDA and the sponsor assumed within the 78 weeks period, placebo arm, within this very mild patients, the deterioration or disease progression of this endpoint could only be 2 units. And the drug's effect is only assumed to be 0.5. That represents 25% improvement or reduction of disease progression. This was considered clinical meaningful, okay, after almost 2 decades of research and with all these failures. We believe this was clinically meaningful. This is certainly not a magic drug that can totally stop the disease progression or even reverse. But at this stage, we believe even 25% improvement was good enough. Now let's look at what actually happened. In fact, the CDR-SB only deteriorated 1.5 to 1.7 units in the trial. It's not even 2 units. And at the same time, the high dose improved by 0.4 units, not the 0.5. But the percentage is exactly 25%, just like what we planned for. So I would argue this effect size is not small. It's exactly what we hoped for. Last, this is probably the very relevant part for today's talk. Again, I want to upfront make it very clear. We did not look for errors intentionally. We found these problems during our reanalysis after the AC meeting, when we did the additional analysis. And also, this is not a pharmacometrics versus statistical problem, because in fact, if you ask the statistician from the sponsor, they will agree with all of our analysis, because in fact, after the AC meeting, the sponsor submitted a more than 100-page report pointing out many misunderstandings or factual errors in the FDA's AC backgrounder statistical part. So this is not pharmacometrics versus statistics. This is, we agreed with most of the sponsor's statistical arguments, and we found even more problems. So these are the 3 key points from our statistical colleague at FDA to argue why the drug was not effective. So the first point is they have to find evidence to explain why 302 was a false positive. And the main reason was they believe this is a false positive because the placebo worsened, not because the drug was working. So I will explain that in details. Then the second is there's no relationship, or there's a lack of relationship between the SUVR, the biomarker, and the clinical endpoints. That argue against, even if you reduce the biomarker, clinical endpoint should not improve because there's no relationship. So this is the second point. The third one is the statistical sequence test, because the low dose for the primary endpoint did not reach the 0.05. It actually is 0.09. Based on our statistical colleagues' understanding, all the secondary endpoint, even though they reached statistical significance, they should be discarded because it did not pass this hierarchical test. But once again, this is a misunderstanding of the sponsor's statistical plan. So let's look at these issues one by one. First, the drug was not working. The positive result from 302 was due to placebo worsening, not because the drug was working. This is one of the slides presented at the AC meeting. Let's focus on the 302 on the left. The blue bars and the x-axis is time after randomization. Y-axis is change from baseline. So this panel is placebo. This is low dose, high dose. The blue bars are the data pre-PV4, and the red bars are post-PV4. Oh, by the way, the figure in the middle is only for the ApoE positive subgroup. But the numbers at the bottom that labeled as overall includes both ApoE positive and ApoE negative patients. So these 2 figure and data, the numbers are based on different data sets. Let's look at the number first, because there was a typo or error in these numbers. If you look at these numbers, the point on this slide is for the placebo group, from pre-PV4 to post-PV4, it got worsened by 0.24 units. But if you look at the high dose, it only got worse by 0.08 units. That's why the difference between the high dose and the placebo got larger in the post-PV4 period. So that's driving the delta between high dose and placebo. But in fact, those 2 numbers were switched. Actual correct number should be 1.37 should be for the high dose, and 1.25 should be for the low dose. Once you correct these 2 numbers, the worsening within the high dose is 0.2, which is very similar to the 0.24 for the placebo. And as a result, the placebo corrected delta for the high dose is very consistent, whether you look at pre-PV4 or post-PV4. So this error led to this misinterpretation of placebo worsening is making the delta larger at the post-PV4. In fact, this hypothesis was even exaggerated by the plot. If you look at the plot, again, within the ApoE positive patients, the high dose post-PV4 did not change at all, zero change. But if you look at the placebo, it got worse than 0.6 units. Remember, the delta was only 0.4 units. But here, the placebo worsening itself is 0.6 units. It's huge. But when we try to reproduce this plot, when we did the sensitivity analysis, the false positive, we could not reproduce this plot. Instead, what we got is this plot. Here, I want to emphasize, we included not only the ApoE positive patient, which is the lower panel, we also included the ApoE negative, that's the upper panel, for all 3 doses. Focus on the plot on the lower right corner. This is what actually happened for the high dose pre and post PV4. As you can see, even for the post PV4, it also got worsened by a significant magnitude, which is totally different from the plot on the left, which is no change. When we shared our results with our statistical colleagues, it turned out that this data set on the left, or the figure data source, was based on a preliminary data set that was not audited. This is not the final data set in the BLA that should be analyzed for approval. So it turned out that middle figure for the ApoE positive people, this data source for this figure is even different from the data source for the overall data set. Remember, this data set are correct. The overall data set at the bottom of the slide, they are based on the correct data source. So this error contributed to this placebo worsening conclusion, which I believe heavily influenced the AC members' vote. The second point, again, this is one of the slides presented at the AC meeting. Conclusion is in the title. Lack of correlation between the biomarker and the clinical endpoints. How the analysis was done? Focus on the plot on the left. The statistical team only took the high dose and took the individual points within the high dose and the y-axis. Remember, the sponsor showed was the group level average, which was balanced, randomized group. But within one dose group, for example, the blue one is for 301, the red one is for 302. As you can see, 302 not only did not show the positive relationship, that positive relation totally disappeared. In fact, is slightly negative. Why? Because these individuals within the same randomized arm, they are very different. The lower end people, they are very different from the higher end. Similarly for the 301. So this is a simple univariate analysis without adjusting any confounding factors. And similarly, this is the post PV4, okay, in the 302. Once again, the point on the slide is that there's no relationship. If there's any relationship, is negative. Then I will ask the question, how can an obvious positive relationship just disappear? Remember, if we look at the randomized, balanced groups from placebo, low dose, high dose in 302, there is this consistent positive relationship. The more SUVR reduction we have, the more clinical endpoint improvement. But if you only take out the high dose and look at the individual scatter plots, this is what we'll see. It's a negative correlation. Why? Because this is a highly confounded relationship between these 2 endpoints. What are confounded? So many factors are confounded. We just grouped patients within the high dose into 4 groups based on their SUVR into 4 quartiles. Okay, this is the lowest one, the lower 25%, and this is the highest 25%. As you can see, many of these confounding factors, ApoE positive percentage, baseline AD drug use, and age, a lot of factors are confounded. That's why seeing this relationship It's not surprising at all when the true relationship is positive like this. In fact, in the pharmacometric field, we see this kind of error all the time because our division deals with this challenge almost daily. Because our exposure response analysis is sometimes based on one single dose data. And we know very well, most of the time, individual exposure on the x-axis or the biomarker is not only related to the y-axis response, but also related to many other confounding factors that also affect the y-axis, y response. That's why we believe this is not the right analysis. It's a very misleading analysis, again, that influence the AC members' votes. The correct
Excuse
way
me
to
for
look at
interrupting, Yanning, but we have about 5 minutes left.
Okay, I'm
We have had a couple of questions entered into the chat, but if you want to wrap up and then maybe answer a question or two, that would be great.
Absolutely, I'm almost there. So we believe the correct analysis is based on the original randomized balanced group within each study. Okay, for 301, as you can see, over the wide range of placebo all the way to 10 milligram, the correlation is very strong. And the 302, once again, is also very strong. 301 is the exception. But if you look at placebo low dose without outlier, they're entirely consistent. The last point, the hierarchical sequence. Based on our statistical team's interpretation, the sequence should be like this. You test high dose on the primary, low dose primary, everyone has to pass 0.5 before you test any endpoint in the low dose. Remember, the low dose for the CDR-SB was 0.09. Because of that, nothing matters for the secondary endpoint. That's the point. But this interpretation of the statistical plan is not correct. In fact, sponsors submitted this after the AC meeting to clarify this misunderstanding. The language on the left is in the original protocol. And the sponsor submitted a picture to show what the language on the left really meant. They actually planned to do this low dose for the secondary endpoint, low dose for the primary endpoint, and high dose for the second part simultaneously once the first one is passed. In fact, this exact same analysis was already used in previous NDAs because this design was used multiple times to test 2 doses or 2 regimens on multiple endpoints. I took out the review from the previous NDA, and you can see the statistician fully and correctly understood how the test should be done, at least in the previous NDA. I'll quickly go over several small molecules after the drug was approved, because once again, people argued, oh, you only collect the antibodies. How about those few small molecules? This is a small molecule. On the left is the dose dependency reduction of the biomarker. P-value highly significant. Yes, the drug reduced the biomarker, but look at the magnitude, 0.4, 0.6. Just like all the failed drugs, the magnitude of biomarker reduction is too small. As a result, on the right side, clinical endpoint all failed. Another small molecule, same thing on the left, biomarkers statistically reduced. On the right side, clinical endpoint failed. Another one, same story. You put all those points on the same plot, whether it's large molecule or small molecule, they failed because their A-beta reduction was not large enough, even though they reached statistical significance. So in summary, I believe, or the entire clinical pharmacology team believe, and plus the clinical team, the clinical endpoint evidence itself was more than enough to support the drug's approval, to support the drug's regular approval, not accelerated approval. And there are so many misconceptions out there due to lack of understanding about the detailed data and some unprofessional media reports. Once again, even though our reviews were all published, I was expecting this journalist or AC members, experts to read our reviews in detail. But based on the news and the message coming out after our review got published, I don't think they read our review very thoroughly. So I encourage them to read every review from the beginning to the end very thoroughly to understand the details. And finally, I want to re-emphasize, FDA made the correct decision for millions of patients by listening to their voice. And once again, I will thank this group of heroes for making the right decision for the patients. And of course, I want to thank the scientists from Biogen for standing up, for having the courage to point out all the misunderstanding errors in the FDA's AC backgrounder. And most important, I want to thank many patients, the thousands of patients and their family members in the clinical trials for their efforts and contribution to all this precious data. We should not just look at those two numbers and make a decision so easily. We should take advantage of every piece of evidence and do the best analysis to have the best decision for millions of patients and their family members. Thank you very much.
There was an interesting question from Simon Gu, and I'm going to ask it, but Simon, if you want to clarify, you're welcome to unmute and ask. But he said, what does this approval mean for future submissions? Will this type of rescue support be expected from the FDA for other borderline drugs? And I'm not sure that's exactly the right question, but can you comment around that a little bit?
Once again, we don't think this is borderline results. If you have a result as positive as 302 with 4 different clinical endpoints, again, they are correlated, but they are not highly correlated. Every one of the clinical endpoints measures different aspects of disease. They are all highly statistically significant with clinical meaningful improvement. And I did not cover this in detail. The sponsor did 80 different subgroup analysis for that study, and 79 of them showed the drug's consistent effectiveness. This is not borderline. If in the future, and even 301, again, 301 is not a failed study in my eyes and in the clinical pharmacology team's eyes. We look at all the data in a totality way. We don't think this is a borderline case. If in the future we have a case like this, our team and our office will still recommend full approval. If you can find, look, people have different interpretation on borderline. On this one, yes, if you ask the FDA statistical team, they will tell you this is not even borderline. This is terrible. The 302 is false positive. Everything in 301 was the truth. The drug was not working. Different people with different backgrounds will look at this in a very different way. So I don't think this is a borderline case.
Okay, thank you. Can I ask one more, do you think, Lee? Or
Sure. Go ahead.
So the other one, there were several questions, and there's probably not time for all, but Hong Lee asked, as multiple confounding effects such as ApoE4, disease progress, etc., may factor in the endpoint evaluation, may I know if the FDA recommends to adjust the well-recognized confounding effects in both primary analysis and exploratory analysis, and it may further impact and inform the labeling strategy? Any comment on that?
Well, absolutely. In fact, the primary MMRI analysis already included multiple confounding factors. And in the exploratory response analysis, we also adjusted for those confounding factors. That's why we keep emphasizing we need to include all the data from randomized groups and still adjusting for the confounding factors. In fact, we believe if you take a single dose, like take only the high dose, it is impossible to correctly adjust for the confounding factors. And that's what you will see in the review. Because when you don't take all the doses in a randomized way and only look at one dose, it is impossible based on our own experience. Sometimes the confounding factor, the confounding relationship was so strong and so complex. Using the typical multivariate linear confounding correlation or covariate analysis, it's impossible to correctly even correct those confounding factors.
So, Dr. Wang, thank you very much for your presentation. Go ahead, Lee.
Thank you very much, Dr. Yanning Wang. I think this is very educational and crystal clear about the thinking process from the FDA side. I learned a lot today. I agree with you. I see the courage from FDA in these controversial cases. Normally, we consider FDA as more conservative, right? But in this case, you guys did such thorough scientific investigation and under such a big pressure and approved it. Kudos to the team. Hopefully time will tell, right? Now lots of companies are developing these drugs right now using the biomarker. Hopefully, yeah, all the patients will benefit at the end. Thank you.
Thank you. Thank you. Thank you for your, I guess, confirmation of the message. In fact,
Yeah.
I would encourage everyone in this meeting, you're all very established statisticians. You should read our reviews and form your own opinion, okay, from a neutral perspective. Don't be influenced by my presentation. Read the review yourself and see whether you agree with the sponsored argument against the FDA statistical analysis, and see whether you agree with our pharmacometric analysis. Thank you very much again.
Thank you. Thank you so much for your time. So I guess, Jamie, it's already 9:15. We don't need another break, right? Maybe you can just start your presentation for your session.
No, I'm thinking that people can stand up and go use the restroom and we won't notice. So please take breaks if you need to. I think we probably should try and stay on time. And so, Rick, do you want to introduce your session a little bit, or?
Yeah, hi, Jamie. No, you just, well, the Astellas presenters for the session are scattered all over the world, different time zones. And we weren't sure how well this would work for us. So we did a pre-record ours, and I think you can just go ahead and play it, Jamie.
Okay, I have to get to the beginning here. Just a second. Are you hearing it?
Nope.
Nope. Okay, just a second. I have to share differently. No, that's not what I want. Doc share. Share screen. Share sound. Okay.
Transplant. I want to begin. Yeah, thank you, organizers and members of ASA and ICSA. In this session, we're going to be talking about Astellas' successful real-world evidence submission in lung transplant. I want to begin right away, but first, a couple of quick housekeeping notes. There are several speakers for this session, and although some of us have pre-recorded our presentations, all of us are online. We would like to ask you to hold your questions until the end, but if you have a burning question, you can try the Zoom chat, and maybe one of us might be able to answer during the talk or shortly after while the next speaker is presenting. And with that, I will get started. So to be honest, the interest in this topic is almost certainly because it was a successful real-world evidence submission, as these news headlines show. If it wasn't successful, I'm not sure we'd be speaking here today. An important detail that might be implicit in the headlines, but in my opinion, not clear enough, is that the submission was based exclusively on real-world evidence. That is, there were no sponsor-supported clinical trials and no links to previous submissions. Here's a list of speakers and topics that will be covered. I will let each speaker introduce themselves, but the first three speakers are from Astellas, and between the three of us, we'll go over the background of the submission and aspects that we think are most of interest to statisticians working with real-world data and real-world evidence in a way that's critical to an NDA. There's also a speaker from CDRG and a speaker from the FDA to share their experiences and give us their perspectives on the project. Presumably, we all know who the FDA is, but the CDRG is probably unfamiliar to many of you. Briefly, they're the organization that operates the real-world data we used in our submission, and they provided expertise with that data and statistical resourcing for this project. The three groups, Astellas, CDRG, and the FDA, we haven't coordinated our talks, so it'll be interesting, I hope, for us and you to see where our perspectives overlap and where they deviate. My name is Rick Croy. I was the global statistical lead from the Astellas drug development team on the project, and I'll be covering the need to know basics of the therapeutic area and a lead up to the submission. I'll also cover notable items of agreement with the agency, as well as submission contents that may be of interest to statisticians. Here's a disclaimer for the three Astellas speakers. Basically, it states that although we are employed by Astellas, any statements of fact and opinions are our own and do not necessarily reflect the opinion or position of Astellas or any of its subsidiaries or related entities. Here's an overview of the submission timeline. Presumably, everyone is familiar with the FDA framework for real-world evidence that was published in early December 2018. And almost simultaneously with that, Astellas approached the FDA to discuss the possibility of using real-world evidence to address an unmet medical need in lung transplant recipients. As for the rest of the timeline, there's nothing particularly unusual about the items here, except perhaps some multiple type C meetings and intermediate follow-ups. But that aside, the only thing that I think is notable is the total duration. And indeed, from a beginning to end was about 20 months, which is very fast for a new drug indication, given that we started with 0 data and no study protocols. You can compare that, for example, to a large phase 3 protocol, which itself can take many years from conception to completion, especially in the area of transplantation. Here's some need-to-know background about the therapeutic area of transplantation, which can be summed up with just a few points. Transplant surgery has been going on for a long time, but it wasn't really until the first calcium inhibitor, or CNI, called cyclosporin, or CSA, was introduced in the late 70s that patients could survive beyond a year. And because of the huge success of that drug in the 1980s, the US started putting laws into place to regulate transplantation, including mandatory collection of data for all transplant patients. And that data is collected in the Scientific Registry of Transplant Recipients, or the SRTR database. Astellas' drug, tacrolimus, which is also CNI, is one of only 2 CNIs for transplant. It was approved for liver in patients in 1994 and took a dozen years to get kidney transplant patients and the small group of heart transplant patients approved and on the label. It's also important to understand a few points about immunosuppressive medications, or IMEDs as I call them. There are many IMEDs that patients need to survive after a transplant. The current standard of care includes CNI medication. That's considered the cornerstone of therapy for transplant patients. And CNIs have a narrow therapeutic window that require dosing to be tailored for each individual patient. So dosing recommendations in the label suggest an initial dose followed by target trough blood concentration levels that are controlled by therapeutic drug monitoring. And the reason it's important for you to know this is just to point out that large amounts of data are needed to support such labeling recommendations, and this proved to be quite a challenge for this patient population. So why not run a clinical trial for lung transplant patients? Well, to answer that question, it's important to understand that the function and problems of each organ is unique enough to require their own approval program and indication. So the simple answer to the question is that there are just so few lung transplant patients available. And you can see that from the annual number of patients in the figure here, which is quite small. So after all this time, why bother looking for approval? Well, given what I said on the last slide, it probably won't be a surprise for some of you to hear me say that we simply didn't think a clinical trial was feasible before the FDA's real-world evidence program. Aside from that, there, of course, is a strong medical need because there are no IMEDs approved for this patient population. The number of patients is also increasing, albeit still a small number. And very sadly, there are challenges from payers. Some patients could not get reimbursed for CNI use because it's prescribed off-label. Also important is that tacrolimus is already widely used. So it's important to patients and physicians to get some dosing recommendations on the label for the patient population. I believe that most of you are familiar with the published framework. So you'll recall that it defines broad parameters around the use of real-world evidence, but it doesn't endorse any particular approach or detail how data standards and inspection readiness for real-world data will be handled. If you're not familiar with the framework, you can easily find it if you Google it, and we'll bring you to this link below. Nevertheless, I will say that there are many statements that provide me with some key takeaways, and that is the FDA doesn't have all the answers, but they are genuinely open to the use of real-world evidence and want to work with sponsors to make the program work. I've highlighted a few sentences from statements by FDA leadership that are called out in the document. And so those are the key takeaways for me. All right, so here is a list of some of the more notable items of feedback and agreement between Astellas and the agency. Regarding the SAP, as David and Josie will tell you later, our pre-planned analysis compared TAC and cyclosporin, the two CNI medications. Nevertheless, the FDA made clear that their decision to approve or not approve would be based on estimates of tacrolimus performance, not on treatment comparisons. The FDA was also very clear about their concern over missing data. We didn't discuss the reasons at length with the agency, but presumably the concern results from generally less rigorous data collection practices in most real-world data compared to registered clinical trials. Most interesting to me was the agreement to have label recommendations for dose based on published literature, and notably, it was primarily from heart transplant patients. The basic background to this is that there just wasn't that real-world therapeutic drug monitoring data that I talked about available for lung transplant patients. And there was scientific justification for the use of exposure results in heart transplant patients. Some agreements regarding the use of our software. One, this is what was used for the analysis. The FDA was accepting of that. They did ask that we submit the code and documentation, though. And two, FDA did require some technical assistance. They had some trouble with the LightText editor being used by the R programs. And a second thing was they needed R code that would allow them to read in SAS and Excel files into R. The dataset submission. So we weren't sure if datasets would be required since we didn't own or generate the data. And although we planned not to provide it, the FDA stated that they did want these during our discussions. And from a statistician's point of view, I think you'll find these items most interesting. The native format for the real-world data was something called standard analytical files. Actually, I think it's standard analytic files, sorry, or SAFs. The structure is not at all CDISC compliant, but the FDA still agreed to receive these non-CDISC structure, but they did want SAS export files. The SAF dataset and variable names were also longer than what the required version 5 SAS dataset allows. For example, variable name length was more than 8 characters in many cases. And after going through some possible solutions to get around this, we did some testing also of the version 8 SAS export, and the FDA did agree to receive version 8 files instead of the standard version 5 files. We proposed to have CDRG submit the datasets directly to the FDA since we didn't have the rights to the data. The FDA investigated this internally to see if they can make it work, but were unable to accept it in the end due to internal policies and infrastructure. So we did end up asking for and getting permission to get the data so that we could do this ourselves. And finally, collection forms, the data collection forms were far more extensive than we needed. Remember, this is 20 years worth of data. So and it collects information about all transplant patients, so it's quite extensive. And the FDA did agree to receive only the relevant annotations. Here's a list of statistics-related deliverables made in the submission. Of course, there are the regular planning documents like protocol and SAP, the CSR, and we also provided the raw data sets as well as an Atom-like data set. These were all in version 8. Partially annotated data collection forms we submitted, but because the real-world data is 20 plus years old, we only provided the current forms, with one exception where we believe there was a value added, and that was IMED's collection, where the old form better showed how the data were collected. A couple of reviewers' guides for the raw and Atom-like dataset. The latter included much information on the input-output of our programs as well. Defined files, which took a lot of work due to the non-CDISC format. And finally, our code used in the analysis, as well as extra code to convert the data sets into the different formats used by our analysis code. And that is a wrap for me. So without wasting time, I will turn this over to David Nimke.
Thank you, Rick, and thank you to the Northern Illinois chapter of the ASA for having us today at your meeting. I'm David Nimke. I lead our real-world evidence U.S. group at Astellas. My group specializes in the design and conduct of secondary use database studies at Astellas. Today I'm going to talk about the team's process for developing a process that was ultimately accepted by FDA. There were a number of questions asked and decisions made along the way. For many, this isn't going to be surprising. Producing real-world evidence doesn't differ from other research in that there is certain evidence that you would like to collect, a few different ways you can go about doing this, and all the options come with trade-offs. At our very first meeting, the team generated a list of things that we thought would be needed in an ideal situation or submission. A best case scenario or wish list, actually. At this early stage, we didn't try to figure out where information was per se, or even prioritize the information. We thought about exposures, how we might describe the patients, and which outcomes were most important in transplant. This list became the basis for our identification and selection of potential data sources for evidence. With our list of data elements in hand, we began to search for possible sources of data. Each source came with its own strengths and limitations. Transplant sensors offered potentially the deepest data on patient care and status. Trade-offs included generalizability and questionable feasibility. Claims data is routinely used in post-authorization safety studies, but there are well-recognized data limitations, including the lack of clinical data. Registries, when they exist, can also be a source of secondary use data. The Organ Procurement and Transplant Program and the transplant community has benefited from the evidence generated by the SRTR for years. The SRTR is a solid organ transplant registry that collects data on all transplant candidates, recipients, and donors in the US. For illustrative purposes, I started building out a heat map of sorts for the data elements that were desired and where we might be able to reliably capture them. Each row represents a variable, condition, risk factor, exposure of interest for the team. Along the top, we had some data options. The left column represents the SRTR without linkage. The right column represented the availability of these data elements in claims databases. The middle column represented the SRTR with linkage to claims. Each option had certain benefits and trade-offs that were considered in relation to the data evidence that we identified previously. No data sources had trough information. Typical of registries, many therapeutic area-specific data elements were collected. We considered how data linkages might further augment the data with trade-offs of increased cost, time, complexity, and a reduced sample due to the need for overlapping patients. Importantly, we had a very strong source for death and graft failure data in the SRTR. By this time as a group, we had a pretty good idea of what our primary evidence needs were, where we might get the data, and what was not feasible. Given the SRTR registry represented a census of lung transplants in the US, had near complete capture of our study endpoints, reliably gathered information on treatment, and included transplant-specific variables of interest, we found ourselves in a very desirable situation. There was fit-for-purpose secondary use data that was available to us. At this point, we move forward with designing the study. Having had the discussion of what evidence we would like to generate and having a good understanding of the contents and structure of the data, we were ready to formulate our research objectives. The primary objective looked at the use of immunosuppressive regimens in lung transplanted patients in the US and the incidence of transplant-related outcomes over time in the US. The secondary objective evaluated the real-world effectiveness and safety of tacrolimus plus MMF in lung transplantation. In order to meet these objectives, we designed a cohort study using secondary use data from the SRTR. The SRTR transplant follow-up forms collect data information at transplantation discharge and then on an annual follow-up form thereafter. The decision was made to look at endpoints 1 to 3 years post-transplant, with 1 year post-transplant being considered as the primary analysis. The study collection criteria were chosen to represent a pretty generalizable population. Exclusion criteria 1 through 3 were applied because patients with these characteristics are special cases and generally have poor prognosis. Exclusion criteria 4 and 5 were data source specific and necessary to establish an index state for the planned analysis. Exclusion criteria 6 was necessary because our study was designed to look at specific interventions, and the lack of recorded immunosuppressive regimens in this particular population would indicate a data quality issue for the record. The team worked through a number of questions as it related to tacrolimus. What should be the comparator? Did it make sense to have a historical comparator when transplant techniques have evolved over the past 40 years? For this indication, no immunosuppressive agents were approved, yet every patient received them. Is it still appropriate to compare tacrolimus to these other drugs, even if they represent standard of care? Also, immunosuppressive regimen maintenance is regimen-based. Do we need to isolate the attributable contribution of tacrolimus? Could we even if we wanted to? After much discussion, we arrived at a solution that focused on standard immunosuppressive regimens, and measuring how treatments and survival had changed over time. In addition, we supplemented this historical information. We supplemented our data with historical information from the literature to characterize the importance of calcineurin inhibitors. Outcomes also generated a lot of discussion, and here we took some inspiration from regulatory experience stemming from clinical development. We focused on overall survival and graft failure as primary endpoints. These were reliably captured in the SRTR with exact dates. We chose a handful of composite endpoints based on clinical development. Other outcomes captured in the SRTR were summarized as the data collection permitted, since some of these were collected as having occurred on follow-up forms without the SRTR requiring an exact date of the event. Another big point of discussion had to deal with addressing immortal time in the database. Data collection began at a patient's discharge. Consequently, if we wanted to index on transplant date, a patient's eligibility in the study would be conditioned on them surviving until discharge. This time period between the transplant and discharge represents immortal time, or a period of time where the patient is not at risk in the study period. This can lead to immortal time bias when the rates of an event include immortal time. The team considered two options for addressing this. The first option was an incident user cohort design where patients are indexed at discharge date and followed. This is a straightforward solution to remove immortal time threats when comparing active treatments. The second option was left truncation, an approach that allows patients to enter the at-risk period later in follow-up. In this scenario, the patients would be indexed on their transplant date, and they would enter the at-risk period at the time of discharge. We ended up having both in the protocol, with indexing at transplantation with left truncation chosen as the primary analysis, and indexing at discharge chosen as a sensitivity analysis. The primary analyses included descriptive survival analyses by treatment regimen and treatment era. In addition, multivariable modeling was done to compare OS and graft survival between the immunosuppressive regimens. We adjusted for factors associated with treatment choice and the outcome. The study proposal was submitted to FDA for comment. We were pleased with the responses that we got back from FDA regarding the proposed approach. They did have some comments. They requested more clarity around the analysis of secondary endpoints as it relates to missing data. They also expressed an interest in some pre-specified subgroup analyses. Interestingly, they gave us a first indication of how they were going to decide whether or not the application would be successful. The FDA welcomed the comparative analysis as supplemental. FDA communicated that efficacy would ultimately be assessed based on non-comparative evidence of the rate of death or loss of graft failure among patients receiving tacrolimus and MMF. With this feedback in hand, we move forward with protocol approval and study execution. Josie will tell us more about the results coming out of this study and our activities preparing for the submission. Thank you.
Thanks, David. It's my pleasure to take you through the rest of our story and how the plan David described played out in the actual analysis and interpretation. I'm not going to present all the study results, just a small selection of what we expect or hope to be more interesting for a statistical audience. Firstly, let me introduce myself. I'm Josie Wolfram. I lead a small team focusing on leveraging real-world data to support our development projects. And before focusing on real-world evidence, I worked in biostatistics. The first step in conducting the study was to apply our inclusion and exclusion criteria so as to define the cohort. About 12% of transplant recipients were lost through the criteria, about a half of those due to death in hospital. leaving us with a total of over 26,000 adults and 725 children. We then characterized that cohort with respect to demographics, disease characteristics, etc., and with respect to treatment regimen received. I've selected to present on the slide a visual of treatment use over time. Here you see the count of transplants over the 20-year inclusion period. The total number of transplants performed over that period tripled, or the yearly number tripled. The colours represent the immunosuppressive regimen at discharge and illustrates that over this period, tacrolimus plus MMF has come to be by far the dominant regimen. Some patients didn't receive one of the two drug combinations, either alone or with steroids, and these went into a other group. I will now leap to talking about analysis of our primary outcome. Upon receipt of the tabular estimates of cumulative incidence, we were surprised by very wide confidence intervals. That we had an issue was even more clear when we got the Kaplan-Meier plots. The TAC MMF arm had over 15,000 patients, well above the other arms, and yet we see a big jump in the event estimate very early on in time and very wide confidence intervals. What was going on? Here, the same plot, but this time with the numbers at risk shown. Firstly, the number at risk at the time of the first event, then at 6 months, 12 months, etc. In the orange arm, or the TAC MMF arm, we had very few patients that had entered the risk set on the day of the first event, and with very few patients discharged and events already occurring, the KM calculations resulted in a high and imprecise estimate over the first week post-transplant. And to quote from the reference book, this early instability in the point estimator of the survival function may propagate throughout the entire curve. And it did. When we looked closely, we found 4 events in the TAC MMF arm that occurred within a week of the transplant date. Where our reference suggested deleting such events, we felt more comfortable reassigning the timing of them. So we shifted these 4 events to be on day 10 after transplant, which was when the first event on any other arm was observed. This resulted in a much more normal looking KM curve. Given these 4 early events essentially resulted in an uninterpretable estimate, something else had to be selected for primary interpretation of the trial and for the label. Astellas' selection was the day 10 shift analysis I just showed you, because timing from transplant was consistent with the original endpoint choice. Our sensitivity analysis with time from discharge, that's the middle one here, looked pretty similar to the modified left truncation analysis, that's this one. And well, we put the 3 of them into our report. During their review, FDA made requests to assess robustness using various approaches. Well, they're written here, I won't read them out. Broadly speaking, these only gave small changes to the estimates or similar changes across exposure groups, and FDA ultimately chose time from discharge for the label. As well as the additional analyses, FDA were interested to understand the reasons for the early hospital discharges, but this was something we couldn't ascertain from the data, and going back to sites in this setting wasn't an option. As well as the KM analysis by exposure group, we conducted Cox modelling. These were not sensitive to the left truncation issue. We had a predefined list of variables for potential adjustment, and nearly all of these were selected by the Lasso model predicting treatment and also in the subsequent univariate outcome models. If you squint at the table footnote, you can see the list of covariates we adjusted for. The results were consistent with the modified Kaplan-Meier analysis and appeared to indicate better outcomes on the TAC-containing regimens. The adjusted model did temper the hazard ratio somewhat, and much of that that adjustment was attributable to the time confounding. Indeed, differences in baseline characteristics seen across regimens were investigated by time error and seemed to be largely the result of changing patient characteristics over time. The Cox modelling was as much to understand the risk factors as to compare treatments, and there may well be residual confounding of unknown direction and size in these comparisons. And although FDA had been clear that their key interest was the results within the tacrolimus-containing regimens, this comparative analysis at least didn't raise any concerns, so it was supportive. With this slide, I'll make some comments around data quality and handling of missing data. We could be confident about the ascertainment of the primary endpoint, but secondary endpoints collected in the submission of periodic forms we had less confidence in. So we summarise the availability of follow-up forms and also availability of results at the individual endpoint level. And the level of missingness proved to be 5% or less in the TAC-MMF group, with the exception of only one endpoint. And finally, in a simple approach typical for binary endpoints, we performed sensitivity analyses in which loss to follow-up was considered as an event by including it in composite variables. The Sankey plot here shows the reported immunosuppression regimen at discharge and then at 1 year. However, we would have looked beyond 1 year, given we presented outcomes through 3 years, but the lack of the incomplete reporting, so the data incompleteness spoke against it. So that was an example where data availability influenced what we chose to do. To limit the impact of potential strange values or outliers, we handled covariates as categorical variables defined by clinically meaningful categories or simply rough quartiles of the observed distribution. And as we saw, the length of hospital stay and early discharge and early events led us to look very closely at the distribution of the length of hospital stay. And although a small proportion, we did see a number of patients that were apparently discharged on the day of transplant, and that's something that's not plausible. So we speculated that it represented discharge perhaps to another hospital ward, but I guess this is data that one just has to accept in a big real data set. So I hope that's given you some insight into our analysis and review journey, which led to the inclusion of the study results into the label. And this is the paragraph introduced into the clinical study section, with the 1 year post-charge estimate of graft survival being the statistic presented for each of adults and pediatrics for the two TAC-containing regimens. Turning now to some operational considerations. This was Astella's first experience carrying out a study with the SRTR registry. We had the choice between insourcing or outsourcing the data handling and analysis. We opted for outsourcing to CDRG, who you'll hear from, and CDRG operate the registry. We were using 20 years of data. Some elements of data collection had changed over those years, and we figured having their expertise and experience with analysing these data would really be of added value, which it was. This was also Astella's first experience with a real-world evidence study for efficacy-related regulatory decision-making. The first two bullets on this slide speak to inspection readiness, and they are also additional advantages to having CDRG collaborating on this project, as they could know the data collection and reconciliation steps best, and by being deeply involved with the analysis, we could better communicate related sort of regulatory considerations and how that reflected in the in the data flow. We wrote a plan that detailed our QC expectations and review steps, and in particular ensured that the exposure group assignments and covariate selection decisions were finalized prior to any analysis by exposure group. I think it's fair to say that for CDRG, it may have been a learning curve on requirements for a pivotal study, as we requested duplicate programming for key analyses, and we collaborated on a considerable amount of reshaping of their documentation to support the data and program submission. Finally, working with an academic CRO also resulted in our first submission with all the programming in R rather than SAS, giving an additional new element on top of it being a real-world evidence-based submission. So actually that was my last slide, and so that concludes the presentations from Astellas, and I'm looking forward to hearing the perspectives from CDRG and FDA next. Thanks for your attention.
So Jamie, the next two speakers are doing it live, in case you didn't figure that out from the end there.
No, I had worked that out. So they're welcome to start sharing. I think it's available now. Tim, you go ahead.
All right, I believe I'm sharing some slides.
We're seeing them, yep.
Great. My name is Tim Weaver. I'm a biostatistician with the Chronic Disease Research Group. Slides should be moving forward. I'm going to end up talking quite a bit about some of the same topics we've already discussed, but more obviously from the data vendor side, and in particular about some of the way the data collection really informs some of the decisions. So just as way of background, as mentioned, I work for the Chronic Disease Research Group, which is a division of the Hennepin Healthcare Research Institute here in Minneapolis. As a part of that, I work on the SRTR project, which is operated under contract from the Department of the Health and Human Services and the Health Resources and Service Administration within there. So it really has the weight of the government behind all this data collection. And the final piece of this kind of puzzle here is the OPTN, the Organ Procurement and Transplantation Network, that really deals with a lot of the data collection as well as rules around transplantation here in the US. And the contractor for the OPTN is the United Network for Organ Sharing, UNOS, another kind of research organization. So with that, a big part of the SRTR is the SRTR database, which has data on transplantation in the United States. The primary source of that data is data directly collected by OPTN on transplantations. And then to supplement that, we pull in data from Centers for Medicare and Medicaid Services, as well as National Information Services Death Master File. So as Josie mentioned earlier, we have very good confidence in the specific endpoints for the study, mainly death or graft failure, where we are able to supplement those data sources from multiple places. Now, the HHS requires SRTR to make the data available to the public while honoring applicable statutes and requirements for patient privacy, and for SRTR to respond to requests from the public for data needed for bona fide research or analytic purposes, to assess the performance of HRSA contractors and transplant programs, and for other purposes. So a big part of the work we do are producing kind of surveillance reports, kind of censuses, as we talked about, assessing performance of transplant programs, organ procurement organizations. We produce an annual data report that describes the overall state of transplantation in the US every year. And as already been mentioned, our standard analysis files, which have been used for this study. With that, there's also lots of analysis or data that can be made available by request to a bunch of different groups that are kind of listed there. So to talk a bit more about how the main sources of the data, the OPTN data sources come directly from transplant centers, organ procurement organizations, histocompatibility laboratories. It's all collected in an online system called UNET, which allows kind of everything to happen in real time, so transplant programs can register candidates for transplant. When an organ is donated, the match run, the sorting of candidates can all happen in real time. The UNET system includes data on every organ transplant and donation that has happened since 1987. So it's very broad in that we have, like I said, information on every transplant. But as we've already been talking about, this is obviously an observational survey, kind of a census data collection process. And so there's this trade-off between breadth and depth. And we're really limited by what is directly on the forms that the transplant programs fill out, which gets directly into the OPTN and then into our database. And so all the information we have is really limited by the specific wording as well as rules around the collection cadence of these forms. So to start talking about how that influenced some of the outcomes, as well as the drug regimens that we talked about in this study. As Jody mentioned, we have a good sense of when death or graft failure occurred. So by OPTN rules, the transplant recipient follow-up form must be submitted within 14 days of notification of the recipient's death. It asks for a specific date of death or retransplant. And so that was what allowed us to do specific time to event analysis, as we've been talking about today, like the Kaplan-Meier estimates or proportional hazard models, things like that. On the other hand, as we talked about, some of the other kind of secondary safety endpoints didn't have the ability to do a time to event analysis, and that was because of the specific wording in the data collection forms. Some other outcomes don't ask for specific dates, but they're worded in such a way that if an event has occurred since the last status update, so here the example I'm giving is, has a patient been hospitalized since the last status update? And those kind of questions without a date obviously don't allow for time to event analysis. And so as we kind of talked about earlier, we had to adjust what sort of outcome we could describe these secondary outcomes as, and we settled on a kind of crude rate where if an event happened between year 1 and 2, between 0 and 1, between 0 and 2, to account for instances of a data collection form on year 1 post-transplant not being collected, and then a transplant form or a follow-up form being collected on year 2. Since the last follow-up, there's this ambiguity there. Similarly, for a drug regimen, we talked already about the left truncation not being able to be allowed in the study in the at-risk group until hospital discharge. And that is a direct result of OPTN policy, specifically the timely collection of data, where the transplant recipient registration form must be collected 42 days post-transplant or at hospital discharge. And this is the form where drug regimen is collected. And so that is where that requirement for the left truncation issue with this data set came into play. And it's kind of a standard setup for a Kaplan-Meier, but as we saw from the results, it can have an undue impact on the estimates. Turned out not to be as much of an issue for the proportional hazard because we could adjust for the stay after transplant. So I'm going to kind of go through this pretty quickly and say I thought this was a great approach where we looked at addressing the four for recipients who were discharged from the hospital early. And we can see this outsized impact here. And I decided to show these as survival curves. We were looking at cumulative incidence. It's just 1 minus what we were looking at earlier. And one thing that I thought was interesting about this process when we started to really discuss what was going on with these 4 recipients, the question of, well, what is medical rationale for these? And there was kind of lots of stories we could talk about that would sound very plausible about what could cause it, but that's not available in our data. So we really had to take this sensitivity analysis approach of seeing, well, what is really the impact of these 4 recipients? So as we saw, we shifted the event date to be the earliest of other groups. And you can see this really big shift in the number at risk at the first events here. Similarly, indexing just at discharge date, we can really increase the number of events, or excuse me, the number at risk. But overall, the results have very little impact on those 4 points. And I thought that was a really reasonable way to approach those 4 points in this scenario with kind of restricted data availability. And then just kind of wrap up some of the technical aspects that we implemented here. Everything was in Markdown, which I thought for this kind of long-running study where we're interacting with numerous groups of researchers, that was a nice way to go about it, in that there's always a one-to-one tie between kind of running your results and the code. And we tried to have the smallest number of kind of intermediary data sets we could to hopefully make the communication between the groups a little more smooth. And we utilized some kind of somewhat standard software development practices. We had a goal of modularity as best we could. So reusing common functions for all subgroup analysis and reporting, used a dplyr paradigm, which I find really readable. And so hopefully when we hear from FDA that hopefully that was helpful. And then, as mentioned before, we had code review for all aspects of the study and documented that via GitLab merge requests, which is kind of just as I mentioned, a somewhat standard software development practice. And with that, those were kind of my takeaways from this study, and I'll pass the mic over to the FDA. So thanks a lot.
Tim, we need you to unshare.
All right, there we go.
We see your screen in.
Okay, so can you guys see my screen?
In presentation mode, yes.
Yes, great. Okay, so good morning, everyone. My name is Taehyun Jung, and I'm a statistical reviewer from Office of Biostatistics, CEDAR, FDA. In this presentation, I would like to share statistical perspective during the review of Prograph application. For disclaimer, this presentation reflects the views of the presenter and should not be construed to represent FDA views or policies. As the excellent presenters from Astellas and CDRG already provide most of the key information about the study details and the data, my talk will focus on the discussions during the submission stage and the review stage. In addition, I'll address the importance of acquiring fit-for-use patient-level data and share my thoughts based on the Prograph application. Well, as you may know, the format of real-world data are different from clinical trial data. There are various sources of real-world data, and these data do not have the same shape. That is, there is no uniform format. Therefore, unlike RCT, the collected real-world data may have longer variable names or longer labels. And in addition, these data may not be managed using FDA data standards such as CDISC study data tabulation model, SDTM, or analysis data model, ADAM. Of course, there's still limited guidance compared to the guidance in clinical trials. The SRTR is a real-world data source that contains all these challenges. It is a patient-level observational data. And SRTR use their unique variable names and labels, so their length were greater than 8 and 40 characters respectively, which doesn't fit into the current review practice. And the data was neither in SDTM nor ADAM format, thereby no ADSL, no ADA kind of data format existed. And the single final data set include all efficacy, safety, and baseline characteristic variables with one record per patient. Well, as a stat reviewer, this gave me lots of challenge from the beginning. My training in CDISC and familiar codes that I use for RCT submission became not helpful, so I had to start from scratch. More than that, new issues arose during the pre-NDA meeting. The first issue was about using new SAS transport format of version 8. Second issue was submission of data through file transfer protocol. The last issue was using different programming practice for all data management and analyses. For submission, the applicant wanted to submit data with SAS transport format version 8, which doesn't follow the file format for the FDA submission of all electronic data sets. According to the FDA's study data technical conformance guidance published on July 2020, expert version 5 is the standard file format for the submission of all electronic data set. The table describes the maximum permissible number of characters, and compared to version 5, you can see version 8 definitely allows more character than it, that is up to 32,256 and 32,000 characters, which my real-world data have in its characters. The second issue could be even problematic because the applicant suggests to send the data by file transfer protocol that's outside of the electronic submission gateway. So the data will not reside in FDA electronic document room. The third issue was submitting all codes using R Markdown, thereby no codes will be written in SAS for data management and analysis purpose. So based on the study conformance guidance and no information of the data, we didn't agree with the applicant for using version 8 in the beginning because it appeared only beneficial for lifting the limits in the characters, but includes multiple drawbacks such as increased data size by including more bytes, had concerns for no native mechanism for audit trails or referencing data sources. Although version 8 is advertised as non-proprietary, but wasn't sure whether it is vendor neutral, which is important for FDA policy. To handle this issue, we first requested the applicant to submit a sample data set of 200 subjects in both version 5 and version 8, and all variables that will be included in the submissions. We also requested a relevant data dictionary. And after my data exploration and discussion with several data experts, we reached agreements that accepting version 8 is a different matter, although FDA cannot require version 8. As the data was readable, manageable, and didn't truncate information in version 8, we decided to accept it for this application. However, we refused to accept data submissions via FTP and asked the applicant to submit through electronic submission gateway. And this is because FDA ESG enables the secure submission of regulatory information for review and is our preferred method for transmission. For all transmission that are 10 gigabytes or smaller, the applicant must use FDA ESG. The third issue was not a big problem because R Markdown provides easy way to read codes with their outputs and oversee the whole process with the generated format like in HTML. As R is an open data source program that requires continuous updates, we ask the applicant to specify all the R package and their alternatives if applicable. Now, I will share the discussion during the review stage. The applicant pre-specified the primary analysis and the sensitivity analysis, and all analyses only included patients who survived until the date of discharge. The primary analysis accounts for the character of the left truncated and right sensor long transplant data. The index date was transplant date, and the time at risk effectively began at the discharge date. And no events could be observed from transplant to discharge, that is the hospitalization period. In contrast, the sensitivity analysis indexed on the discharge date and it excluded the hospitalization period. Thus, the sensitivity analysis didn't consider left truncation. In the study report, the applicant modified analysis with index date and hospitalization period equivalent to primary analysis. However, this was not pre-specified in the statistical analysis plan. The 4 early events that occurred before day 7 were manually shifted to day 10, and this modification only occurred in the adult population of the tacrolimus IR plus mycophenolate mofetil, the MMF group. So here's a visual summary of the 3 different analysis methods. You can see the primary analysis index on the transplant date, account for the hospitalization period, and analyze the data primary event as is. The sensitivity analysis index on the discharge date and analyze the primary event as is. The post-hoc or modified analysis follows the method used in the primary analysis, but manually shift the 4 early events to day 10. The applicant chose day 10 because that was the earliest event occurred in the other regimen, which is the tacrolimus IR plus azathioprine. Primary analysis demonstrated non-robust estimates in the adult population because of the four early events. The red line indicates tacrolimus IR plus MMF, and the green line indicates tacrolimus IR plus AZA. From the primary analysis, we can see in the red circle that the 4 early events impact the cumulative incidence from the beginning, so you can see a bump. And it continued through the year and until the end of the 3-year study. And this happened because a small number of patients was discharged and included in the risk set, but the events occurred early, like in day 4. So in the beginning, you can see only 44 patients were in the risk set compared to other regimen. Left truncation approach is sensitive to these events that occur while the risk set is small. As a result, the TAC-IR plus MMF adult group primary analysis demonstrated 14% of cumulative incidence of graft failure or death. That's 86% survival. In contrast, the sensitivity analysis included all 15,000-ish patients in the risk set when the first event occurred. And this is because it didn't consider left truncation and all patients entered the risk set at the same time. The sensitivity analysis demonstrated approximately 9% of cumulative incidence, that is 91% survival. The post-hoc analysis still keeps the left truncation until discharge, but has larger risk set of 2,300 because the first event was scheduled to happen at day 10. So until day 10, this analysis could allow more discharge patients. Similar to the sensitivity analysis, it demonstrated approximately 91% survival. We learned the small initial risk set of the primary analysis led to the non-robust estimate with 86% survival. Post-hoc analysis that manually shifted 4 early events appeared to demonstrate stable estimate with 91% survival. However, this analysis was not pre-specified and just showed up in the final study report. Also, this modification was only conducted in TAC-IR plus MMF arm of the adult population. So we didn't agree to the post-hoc analysis and recommended the applicant to use discharge date as the index date, which is the sensitivity analysis. We were not sure whether these 4 events truly occurred based on the patient's condition or just a record error. It was also arbitrary to push these events to day 10 to increase the risk set on purpose. Moreover, the method was changed after looking at the primary analysis results. However, the treatment group was defined at discharge, so moving the index date from transplant date to discharge date appears suitable, which demonstrated a 91% survival. Now, I'll briefly talk about the importance of acquiring fit-for-use patient-level data. When assessing real-world data, it is important to consider the relevance and the reliability of the underlying real-world data. The term relevance includes the availability of the key data elements that captures relevant exposure, outcomes, and covariates. Relevance also includes sufficient number of representative patients for the study. The term reliability includes data accuracy, completeness, provenance, and traceability. As I have no time, I will quickly introduce completeness and accuracy and refer the others to the RW draft guidance for further details. And this guidance was published last month. So data accuracy implies the closeness of agreement between the measured value and the true value of what is intended to be measured. Completeness is related to the presence of necessary data, so it's mostly the missing data. So as the SRTR SAF, the standard analytic file, contains data on all lung transplant candidates, recipients, and donors in the United States since 1987. The applicant identified 15,478 adult and 450 pediatric patients between 1999 and 2017. And this amount of data was sufficient to demonstrate the outcome of tacrolimus IR plus MMF. The overall quality of the patient level data was acceptable and met the fit for use, as it captured all representative patients and necessary variables in very detail. It provided the granularity of the data. Also, death date had additional layer of adjudication through the Social Security Death Master File. Therefore, there was no concerns on relevance, and low percentage of missingness, which relates to completeness, and accurately coded levels and values of the variables, which relates to accuracy, gave less concerns on the reliability. However, there was some data quality issue because some length of hospitalization reports appear to be not plausible. In this figure, you can see a number of patients had no hospitalization. They had the transplant surgery and got discharged right away. And lots of patients were discharged within a week. The applicant provided possible reasons for this unusual distribution of the length of hospitalization. They said it could be it could be data entry errors, or it could be a discharge from inpatient service to another unit. Although the exact reasons were unknown, we moved on because it didn't influence the validity of the analysis. In Prograph application, now I would like to wrap up the talk by sharing my thoughts in the real world data management. The Prograph application, the raw data file was provided in several pieces of the SAS file. The applicant converted to CSV files and then loaded to R to create a data set of interest. Majority of the programming was performed in R Markdown, which provided both the codes as well as the comments and intermittent results. As a stat reviewer with zero knowledge in SRTR database, I think this was a great practice because it provided a very transparent data management procedure and efficiently executed the codes. And like the RCT NDA submission, the applicant provided an analyst data reviewer guide and looks and feels like a defined document. And these documents were very helpful when understanding the complexity of the data structure and the data management process. So this is the data management process. And in each interim stage, you can see the flow of the output data, which becomes an input in the next stage, and so on. The R Markdown output in HTML format looked good and was helpful during the review, even though we have coded our own code for analysis. So during the review of the Prograph SNDA, RW SNDA application, we learned many things. Most of all, getting fit for use patient level data is essential. Several attributes like relevance of the data and reliability of the data need to be reviewed. And because this is not like a familiar randomized clinical trial data, providing a sample data will make the reviewers get the pictures of the data in the early stage. Especially through this Prograph application, we learned again the importance of transparency of the real-world data. Because real-world data sources are various and non-uniform, it'll be difficult for us to understand the data structure comprehensively in the beginning and needs to be started from scratch. Therefore, providing all necessary documents will aid the review process. And personally, I like the R Markdown and the dplyr. Those were nice tools, but be aware that this is an open source program. Therefore, packages for some methods could have slightly different results. And it is important to submit all relevant analytic package, including any company-owned package that used for analysis. And complex features like LaTeX and R are not necessary and may not be compatible with our computer environment. Therefore, simple and basic codes are just fine as long as they are readable and executable. Well, I think this is the end of my presentation, and thank you everyone for listening. Thank you.
We do have some time for questions. There were a couple that were online, and I know Rick answered at least one of them. I think I chimed in as well. Anyone would like to ask a question, please, you can unmute yourself or you can type it into the chat. We got a very informative talk. Thanks from the chat. So a question for Ryan, is this the first RMD review for you?
Yes, this is Ryan from FDA. Yes, this is my first time to review the real world data application for efficacy. And many of my work was even reviewing the real world data for safety. So it was a very a wonderful experience to have this application.
You said you've used it, or you've reviewed applications where they were using it for safety. What were the sources of that data?
Well, I'm afraid I cannot talk more about outside of this talk, but it's using registry data like well, from like different sources provided by this sponsor. So yeah, but it was mostly using registry data, and that was for safety, but this time it was for efficacy.
Thank you.
So kudos to this. I really learned a lot also. I think this is a wonderful example of using real world data for submission. Hopefully, AbbVie can learn from this too, and all other sponsors, right? This is one of the most innovative approaches so far, not using the RCT, but using the real world evidence. So I have a question. For the I know FDA is really opening up to these innovative approaches. Like it happens a lot in oncology, right? For synthetic control arm. This one is a little bit different. It's purely using the real world evidence rather than constructing a control. So can you comment a little bit on the FDA's view on the synthetic control arm for other indications, like not in oncology?
Oh, well, I have to say sorry that I cannot talk about anything outside of this review. So, yeah. Mm-hmm. But I want to say one thing about the program, how they met the fit for use. Maybe that could be helpful for some question. So the review team found the assessments of the study variables, such as the endpoints of interest, that's the death and graft value at 1 year, to be relevant and reliable. Those are the two important factors required for the fit for use. And given additional study features of complex capture of patients undergoing transplant and the complete capture of relevant clinical information, we found the data from the SRTR to be fit for use. So that will be my high-level answer.
Okay, and it's interesting you accepted the R markdown as a submission package. So does that mean FDA now is more and more open to non-SAS related programming languages? What about Python?
So well, as you know, lots of our submissions are using SAS, and we are all familiar to SAS and trained in SAS. But for real world data application, especially for this time, we accepted R markdown. And because the reviewers can work on that, we think there's no problem. And we cannot ask the sponsor to work on specific statistical progress, right? So I think we're more open in that. If I don't have knowledge in Python because I mostly work on R and SAS, but maybe, who knows, like in the future, yeah.
Mm-hmm. So as you said, the R versioning, because every time you upgrade to R version, the previous program may not work, right?
Right. So that was the challenge because some package needs some updates. So I asked our computer team to like update all my R and also the R Studio. But we had some difficulty when running the LaTeX special package. And it didn't work with our computer environment. And every time I have to ask the computer team to get the authorization for this installation, which impeded my work. So I forgot about that, the sponsor code at the first time and write my own code to do my review. And then after the sponsor resubmit the codes without the LaTeX package, now I start comparing the results. So I think that's the way it worked.
Very nice.
Well, wait till you get a submission in Excel, and then you might decide you have to put limits on which software.
Yeah.
I have a question for the panel generally, which is to say, tacrolimus is a little bit of a unique product in that it was approved in the 90s and we're still selling it today. And so we're still interested in it. So there's been enough time to collect a lot of real world data on it over that period of time. What other places do you think we would be able to do something? Do we need to have registries that go a little bit further? How do we see this sort of thing being generalized in other settings?
Wow, that's quite the question. I think that so David Nimke, Astellas. I think that there are going to be situations where companies are entering or starting product-based registries and other things like that. That could be a reliable source potentially for getting information on drug use in an off-label situation or setting. You're absolutely right that this situation was very different in that we had, I guess, a great deal of off-label use and for immunosuppression, graft failure, I guess, prophylaxis, prevention, in order to, across all of these solid organ transplants. So it is quite difficult. I think that one of the things that's important, I guess, is to think about what information is maybe needed to either augment the evidence that needs to be gathered, or and not necessarily limit yourself to, historical control arms or what we happen to do, which was, actually develop the exposure arm of evidence using real world data. I think the real world data has a lot of opportunities, and those opportunities are based on just understanding what those holes are and trying to fill as many gaps as you can that would, help FDA and others that we're talking to make good decisions. But you're absolutely right, Jamie, that the extent of off-label use in this setting is very different than maybe other settings. And I welcome anybody else's comment.
I would add that as it was mentioned earlier, that this was a really high quality real world database, that's been around for a long time and sponsored by the government, you know, and helps make the U.S. make decisions on transplantation and whatnot, probably was a valuable thing. Certainly, the quality of the primary endpoint. So maybe other, you know, whatever other registries are out there that have really high quality data, I would imagine, should be looked at for these kinds of uses.
But I would hope there are opportunities out there that don't require all recipients of a treatment over 20 years to to meet the requirements. I think we had, you know, really quite an exceptionally large volume of data available to us with a good data source. But I don't think, I assume not every real world evidence label change submission will have, you know, nearly 16,000 patients in the treatment of interest collected over 20 years.
This might be the last one if that is the requirement, right?
Thank you.
I guess one other thing that I would add is that it's awfully hard to identify off-label use in other real-world data sources because the intent of the treatment's not something that's actually collected. So if you're looking at something like claims or EMR, you know, you might have patients that appear to not have the indication that the drug is used for, but you know, there are lots of reasons for that data might be missing. You know, the physician might be treating kind of empirically based on like what they're seeing and thinking might be the case, or there have been historically stigmatized diagnoses that have reduced the diagnosis rates of some diseases. So the off-label use of drugs is not an easy thing to study.
Yep. Anyone else with a question? If not, I might go back to Dr. Wang, because we didn't quite get through all of his questions. And then we can maybe have a slightly longer break, Lee. So one of the questions that was asked in the chat for you, Dr. Wang, was earlier this year, the British Medical Journal published a meta-analytic paper showing no relationship between SUVR and MMSE using a number of trials, including the 301 and 302. I'm wondering if the FDA has done similar analysis. Put you on the spot, I'm not sure you're still there.
Daniel, right? I don't know if he's still on.
I see his name, that's why I was thinking it, but he may have stepped away.
Right, that's
Well,
my guess. Yeah.
Lee, do you want to break until?
Yeah, maybe we can take an early break, right? To
Got 12 minutes since
yeah,
we stole the previous break.
Exactly. Yeah, so we can still start at 10:50.
Hi, sorry. Can I have a quick question to Dr. Jin regarding our markdown submission? I wonder if FDA has any guidance or policy regarding validated versus regular R coding, R section.
Hi, thank you for asking a question. We have no guideline yet, so it totally depends on the reviewer's ability. And if just one thing that I want to address is because it's an open source, and when you use package, we want to be careful on using just one single package, because some package with the same method could give different results, slightly different results. So that's what I try to do during this review. And using any like dplyr, that's good. I didn't have much experience in dplyr through my R experience, but this time I learned a lot, so that was a good experience. But we don't have a specific guideline yet, but maybe that will come up and in the future.
Okay, thank you.
I have a question. So for your review, did you have to go back to compare to SAS just to verify the R results?
No, I all worked on Yes, yes. Not for the data management. So I follow all the data management using the R, but for the final data, I did both analysis in SAS and also in R to verify the results.
Thank you.
This is really good. Again, I think FDA's image used to be like very conservative, right? Only SAS, very rigorous, very traditional. But from these first two talks, we have seen that they're really opening up. The Yaning's example, public, Adcom committees, they all say no, but they courageously against that and said yes for that drug. And this one also, FDA is actively engaging with sponsors to evaluate the real world evidence and for the submission, even using R. This is really good signal for us, I think, to push more and more innovative approaches from the sponsor side. And I'm really glad FDA is really opening up and be more and more innovative. That's correct.
I just wanted to add a small point about the use of R. We were quite conscious about the fact that it's open source. So we were thoughtful about which packages, how widely used they were, or if any of them had been validated. So yeah, we certainly recognize that some very obscure package could be troublesome. We didn't find ourselves in that position, but yeah, I think moving forward, if folks want to use R, they ought to be thinking of that as well, because yeah, that could be trouble.
Yeah, version control is very troublesome. So I guess in your submission, you have to specify which version, what kind of package you use, right?
Yeah.
Okay, so if no more questions, we can go to the break and we'll be back on 10:50.
Lee, just one quick question. So our session is coming up next. We have speakers from outside of the United States right now.
Okay.
Can they test a little bit the Zoom link just in case? No, I'm sorry. Use the Zoom to test the screen sharing a little bit during the break, just to make sure that everything works.
Sure, please. Yeah, of course.
Okay.
Yeah.
Okay, great.
But we will still start at 10:50.
Yeah.
Okay.
Sounds great.
Yeah.
Thank you.
Thank you, everyone. We'll be back in 8 minutes.
So hello. Can you hear
Hey,
me?
Ivan. Yeah, I can hear you.
Okay, I will share the slides. So how it's can you see that?
Yep. Coming up nicely.
Okay, great.
If you want to move the slides to see, yeah, it's fine.
Okay, perfect.
Great.
Yeah, I will stop sharing. Oh, actually, I can pause. Okay. Okay, great. Thanks for the invitation, and I will see you in 10 minutes.
About 7 minutes, yeah.
Yeah.
you guys were able to make it work, right?
Yes.
Yeah, yes. Mm.
We'll start in just a minute or so. I'll let you guys introduce yourselves. So
Sounds great.
taxi time now, so whenever you're ready, you can go ahead.
Okay, so good morning, everyone. Welcome to this session on machine learning methods for risk prediction. My name is Li Hui Zhao. I'm an associate professor of biostatistics at Northwestern University, Feinberg School of Medicine. I'm very happy to have 3 speakers in this session. All experts in machine learning and deep learning methods. So I will introduce as they speak. So the first speaker we have is Professor Yifan Peng from Department of Population Health Sciences at Weill Cornell Medicine at Cornell University. He obtained his PhD of computer science at the University of Delaware. After that, he did a research fellow at NCBI before he joined Cornell. So Dr. Peng has pretty broad research interests, which include biotext mining, bio-natural language processing, clinical natural language processing, and especially of interest of relevant to this talk, deep learning for healthcare, and also biomedical image processing. Thank you, Yifan. Dr. Wang, please go ahead.
Thank you, Li Hui, for the introduction. Hello, everyone. This is, can you hear me?
Yes.
Yeah. Thank you. So this is Yifan Peng from Population Health Sciences at Weill Cornell Medicine. I'm currently an assistant professor. My research interests includes natural language processing and medical image analysis. And recently, we are also interested in applying NLP, natural language processing, and medical image analysis in survival analysis to predict. So I'm really happy to get invited to this workshop. And today I'm going to talk about our studies on leveraging longitudinal and multimodal EHR in the clinical predictions. So before I assume that most of the audience in this meeting are quite familiar with the survival analysis, but I would also like to give a very brief introduction on survival analysis before we get into details into our studies. So the survival analysis is a task to predict the time to events such as a patient's mortality or a patient being hospitalized. The name survival analysis comes from the clinical research where we want to predict the time to death. It differs from traditional regression by the fact that parts of the training data can only be partially observed. In other words, we call those data sets records sensors in the context of survival analysis. So in this example, in this figure below, I want to illustrate what censored and uncensored records means in the survival analysis. So considering a clinical study which investigates some diseases and has been carried out over a period of time from T of O to T of 0 to T of N. So in this clinical trial or in this cohorts, we know we have several types of participants. So for example, patients A and B experience an event after the enrollment, and patient C drop off. Let's see. So patient C dropouts after a certain time, after the enrollment, and patient D was lost to follow up with no recorded events, and patient E did not experience any events before the study ended. So in this very tall example, we only know the exact time of events for patients A and patient B. In other words, their records are uncensored. For the remaining participants like C, D, E, it is unknown whether they did or did not experience an event after the termination of the study. The only validated information that can be observed for participants CDE is that they were event-free up to their last follow-up. Therefore, their records are censored. So given this cohorts, we want to build a model to predict the probability of the time to events for each participant in the cohorts, and that is the task of survival analysis. So formally, we want to build a model to estimate the survival probability of a participant I who has not experienced an event beyond time of T. There are many existing models that can do this task. One of the most appealing model is called Cox model. It has this form. So the Cox model can be viewed as consisting of two parts. The first part is called baseline survival function. We use the S0 of T to represent this function. And the second part is the explanation of a function psi of xi. So this is a basic formula of the Cox model. And from this form, we can see that the first part of this formula is independent of the risk factors of each participant. Oh, by the way, so in this formula, we use xi to represent the risk factors of participants I, and we use t to represent the time of event for that participant. So from this formula, we know that the baseline survival function is independent of the risk factors of a particular participant, and the second part is independent of time. But it can be customized by using the risk factors for each individuals. And in the Cox model, the function of psi is nothing but a linear combination of the risk factors of xi for each participant. So from this formula, we can easily tell that there are at least two disadvantages or limitations of the Cox model. First is that because the psi function is a linear combination of risk factors, so usually we only use a structured HR to extract those risk factors from the records. In other words, those risk factors are usually predefined by the clinicians. So maybe there are other risk factors that are overlooked by the clinicians, and how to utilize those unseen or not well-known risk factors in the Cox model to improve the performance of the model is a very interesting topic that's diverse investigation. And the second disadvantage or limitation of the Cox model is that it's a time fix. So because this is psi function is independent of the time t, so we cannot build the temporal relation between each visits into the model, so that we cannot monitor the changes of features over the time. So therefore, the motivation of this talk or the studies is to expand the psi function in the Cox model to relieve the problem or the limitation of the predefined risk factors, we try to apply the deep learning based methods with multimodal data to integrate different types of data into the Cox model. And second, we want to relieve the limitation of time-fixed disadvantages in the Cox model by utilizing a recurrence neural network, which is also a deep learning-based method to model the longitudinal EHR or to model the temporal relation between each visits or each time points for participants. So based on those motivations, I would like to talk about two projects in this talk. In the first project, we investigate an empirical study of using radiology reports and images to improve the ICU mortality prediction. As the title suggests, in this study, we try to integrate the multimodal EHR into the Cox model to predict the patient's mortality, to predict the mortality, probability of mortality for patients admitted to ICU. So let's talk about the first projects. And this project was published in the Health NRP 2021 this year. So to predict the patient's mortality in ICU, this is a very important task. And in the ICU, there are several scoring systems that are widely used, and they play a very important role in the ICU management. The current scoring system that are based on the are all based on the structured EHR, and one of the most widely used scoring system is called simplified acute physiology score, or we call it SAPS-2. So this scoring system, SAPS-2, utilizes the demographic information of patients like age, race, gender, and also lab tests or other virtual signals to predict the mortality of patients admitted to ICU. Although this score is widely used in the ICU management, there, like I just mentioned, they have limitations of only using the structured HR to predict the mortality of patients. So how to further improve the performance of such scoring system in the ICU management would be a very interesting topic, and our study tried to integrate the multimodal data that, into the model to improve the performance, to improve the predictive performance. So here I show you architecture of our model. So it's actually quite simple. So for the baseline model, we have the clinical variants, and we use the Cox model as a backbone of the survival model. And based on that, we add two more data modalities into the model. One is the reports, the clinical reports in the EHR. So we added because we feel that there are a lot of hidden information mentioned in the reports that cannot be extracted to form the structured HR. So if we can use some deep learning based model to extract the hidden features or to extract the features from the reports, then those features may provide additional information to the clinical variables to the survival model. And besides the reports, we also have the check accessory images, because this is one of the most widely used image modality in the ICU. And again, we also use some deep learning methods. Here we use the convolutional neural network to extract the features from the images with the hope that the images provides additional information on top of the reports and clinical variants to the survival analysis. So we use two models to extract the features from the reports and X-ray images, and we combine it into a larger features, and we put them into some multi-layer perceptions to reduce the size of features. And then we combine, we concatenate the features from the text and the images to the traditional clinical risk factors, and then we feed the factors into the Cox model and see how and evaluate whether the additional information will be helpful. So this is a general, a very abstractive description of the architecture we use. To validate or to assess the performance of this architecture, we use the data sets of MIMIC-4 data sets, which is data sets collected at Harvard University for the patients admitted to ICU. And in order to fairly compare or model with the traditional SAPS2, we excluded patients who had no CHECK-SATS studies before the measurement has been completed. And as a result, we end up had almost 10,000 patients, and 22% of the patients were deceased in the ICU. So this is data sets we use for the evaluation. And here are some experimental sightings. We try to compare our models with different groups of features. The first is sepsis-2 scores we directly got from the MIMIC-4 data sets. The second is the SAPS2 risk factors we extracted from the MIMIC-4 data sets, and then we use the Cox model to guess the survival probabilities by using those risk factors. And then we also extracted the text labels that are commonly mentioned in the clinical reports. So here we focus on 14 common lung diseases mentioned in the clinical radiology reports. And the fourth group of features are text features extracted from the reports. So here we don't specify what type of feature we are interested, but only using some deep learning model to blindly extract the hidden features from the reports. And in this study, the text features has a dimension of 768 dimensions. In other words, it's quite large and quite as compared to the risk factors, the text features are quite high dimensional. We hypothesize that those text features will bring some features that cannot be easily observed by the clinician. And similarly, we extract another high dimensional features from the CHECK-SATS, and then we combine those different types of groups of features together to get joint features before we feed them to the Cox model. And to evaluate to compare different models, we use the C-index, which is most frequently used to evaluate the survival models. It's quite similar to the AUC and defined as a ratio of the correctly ordered pairs to comparable pairs. So I will not go into details about this evaluation matrix. So here are some interesting results we observed from the MIMIC-4 data sets. So here we compare the contributions of different features in the Cox model. And the first observations we find from the results is that the multimodal feature did add additional information to the risk factors. So that's the first observation we find from this study. And second, we find that both the text features and the image features provide additional information as compared to only using the risk factors. But on the other hand, the text features and the image features are quite similar. This is also understandable because here we only use the radiology reports from the EHR, and the radiology reports describe the check X-ray. So in other words, the reports didn't provide additional information for the images, or the images didn't provide additional information if we already has the text features. So that's the first result. And we also compare the Cox model with deep learning based model in the survival analysis. We find that if we only use the traditional predefined risk factors, so we find that the Cox model and deep learning based model achieve the similar results. Even though we add the text tables, labels extracted from the radiology reports, we still get this very similar results between the Cox model and deep learning based model. But if we add high dimensional features extracted from the text and the images, then we find that the deep learning based model outperform the Cox model. And on the other same times, because the joints features are quite high dimension, it's almost impossible to train a traditional Cox model on such high dimensional features. So that's the first task I want to cover. I want to talk about. So in this task, we try to add high dimensional features extracted from the reports and the images to the survival analysis to predict the mortality, patient's mortality in the ICU. In the second task, I want to talk about how to model the longitudinal EHR into the survival analysis. And in this study, we use the another data set we use another task which predicts the patient's mortality of patients with heart failure. So a general question, interesting question is that why we changed to another task, and the answer is actually quite simple because we only have the longitudinal EHR for this task at hand. So that's why we pick another task. But I hope that's the underlying knowledge or observations we observed from our studies can be transferred to other survival analysis tasks. So again, so currently the state-of-the-art methods to predict the mortality of patients in heart failure is to use 14 findings or 14 diseases mentioned in the radiology reports. Those 14 findings were manually defined by the cardiologists, and this because they thought that those 14 diseases or findings are correlated to the mortality of patients with heart failure. And we want to first, again, we want to expand this PSI function in the survival in the Cox model to incorporate the high dimensions features from the reports, like the tasks we did before. But additionally, we also want to model the longitudinal radiology reports to track the changes of those 14 findings mentioned in the reports. So based on that, so here is the architecture of our model. Instead of just using one report, in this study, we use the multiple reports in the observational window, and we predict the 30 days mortality prediction, 30 days mortality, or 1 year mortality, or all-time mortality in the prediction window. So by using this experiment settings, we can build a model that utilize the longitudinal reports from T of 0 until T of U in the observation window. And for each report, we use some natural language model to extract the hidden features from the reports. And we use a LSTM model to build the temporal relation between the reports. And after that, we extract the hidden features from the last states. Oh, by the way, this LSTM model can pass the information from the beginning of this study of the enrollments until up to the T of U, so that this model can kind of memorize the important information longitudinally. And after we build this model, we use the features from the last states as aggregates hidden features from the whole longitudinal reports. And then we feed these hidden features into a Cox model or deep learning based survival analysis model to do the predictions. So that's the architecture of our model. And to evaluate the model, we use the data sets we obtained from the Canal Medicine. Specifically, we use the CT reports for heart failure patients from the New York Presbyterian Hospital discharged with a billing code ICD-9 or ICD-10 between January 2008 and July 2018. The time to events or the mortality were calculated as the number of days between the most recent report dates and the death dates if disease or the last follow-up dates if censored. So for the most recent report dates, we mean the dates of T of capital T between from the T of U. So we kind of shifted the baseline in this cohorts. And here I show you the characteristics of the data sets. We have more than 10,000 patients. And the 10 days mortality is around 10%, and the 1 year mortality is around 50%. And each patient has around 3 reports. And the average number of each CT reports consists of a little bit more than 300 words in the reports. And we compare 3 models: the Cox model, the multi-layer perception, or the deep survive model, which is multi-layer perception based, and also the LSTM model, which try to model the temporal relation along the timeline of the CT reports. And for the features, we evaluate the 14 labels predefined by the cardiologist, and also the common lung diseases mentioned in the CT reports, and also the hidden features extracted by using a deep learning model called BIRDS. And again, we use a C-index and AUC to compare the performance of different models. So here are the results on this data sets. There are several observations. So number 1, we find that by model the longitudinal reports along the timeline outperforms the models that only use the last reports as the inputs to the Cox model or the MLP model. And secondly, we find that, again, the deep learning based model outperforms, this one outperforms the Cox model, greatly outperforms the Cox model. So this again proves that the hidden features provide additional information to the predefined risk factors by the clinicians. So so it's really interesting to see that the reports, the narratives, the images can be helpful in the survival analysis task. And the third observation is that although the LSTM, the longitudinal data the model using longitudinal data achieves the best performance is not significantly outperforms the model that's only used the last reports. So in other words, in the future work, we feel that we probably need a better way to model the longitudinal reports, or we need to find out why the last reports contributes most in the prediction. So one assumption is that, one hypothesis is that although the longitudinal, the history reports provides additional information, most predictions rely on the findings from the last reports, findings in the last visits. So the history visits didn't actually contribute much to the survival analysis. But we definitely need more data sets and more time to investigate such limitations of the LSTM in the survival analysis. So that's actually concluded the task, the talk today. The motivation is quite straightforward. We have the Cox model and we know their limitations. And we try to expand the Cox model by integrating the multi-modal data in the EHR, and we also try to model the longitudinal data in the EHR by using different types of deep learning models, by integrating different types of deep learning models in the Cox model. And in the future work, there are a couple of directions. First, currently we only use the deep learning model as the feature extractors to extract the hidden features from the text and images. We didn't fine-tune the whole, or we didn't train the whole architecture. So here, for the fine-tuning the whole architecture, I mean, we didn't update the weights in both the Cox model and the deep learning model. We only use the deep learning model as feature extractors. So it's probably interesting to update all the weights in the model to further improve the performance of the survival model. And secondly, although the hidden features provide additional information, because it's hidden or because the deep learning based model is generally a black box, we don't know how those hidden features can be mapped to the input. So how to increase, in other words, how to increase the interpretability of the deep learning based survival analysis would be a very interesting topic that we will investigate further. So last, I want to thank some funding sources to support this work, and that's actually all the topics I want to discuss today. And I think I still have a few minutes to ask questions you may have.
Ivan, actually, we are running a little bit behind, I think.
Oh,
so,
okay.
So we will save the question to the end.
Sure.
So I will be a little bit brief on my introduction. Our next speaker is Yu. So she's a PhD student in biomedical informatics at Northwestern University. She has her main research interest is machine learning application in precision medicine. She has applied those methods in several disease areas, including type 2 diabetes and cardiovascular disease. Yu, please go ahead.
Hi, everyone. Thanks for coming to the talk. And today I'm gonna be presenting my work of using the, of using the deep learning survival models for cardiovascular disease prediction. So traditionally, the model that used for cardiovascular disease prediction, a commonly used one is called poor cohort equation model. So this model is based on Cox regression model and is race gender specific. The poor cohort equation model is developed based on 5 big cohort studies, including ERIC, CHS, Framingham original, Framingham offspring, in cardiac studies. So poor cohort equation have a pretty decent performance in white male, white female, black female prediction in cardiovascular disease prediction. So for example, the white male achieved 0.74 and white female have 0.8 and black male has 0.71. Black female has 0.81 C-index. Recently, the deep learning model has becoming more and more popular in prediction studies. So there's several deep learning survival models in the field that has been proposed. So however, it's unclear how this model performs compared to the poor cohort equation in cardiovascular disease prediction. So for this study, we're gonna compare the performance of the several state-of-the-art of deep neural network survival models to the poor cohort equation in cardiovascular disease prediction. Our data set, we use the same data set for comparison purpose. We use the same data set as the poor cohort equation, which is the 5 big cohort studies as we mentioned previously. So in this data set, overall we have 23,000 patients, and for each for the black male group, we have about 8,000 patients. Black female have about 1,000 patients, and black male has 10,000 patients. Black female will have 2,002 patients around. Overall, the average age, the mean age is 57 years old, and the mean systolic blood pressure is 127. Mean high density cholesterol is 51. And the mean total cholesterol is 217. There's about 4.6% of the patients have a history of diabetes. About 26% have a smoking history, and about 31% has a history of hypertension. We also have so this is our training data set, and we also have our external data set, external validation data set, a MESA data set. MESA data set has about 4,259 patients, and the mean age of this population is 61 years old. And these are the following, the other characteristics of those population. So about the deep neural network survival model, in general, the neural network survival model can be grouped into 2 categories. The first category is the adaptation of the Cox proportional hazard regression model. So examples of this are like DeepServe, CoxNet. There are adaptations of Cox regression model. So the idea of this model is, in general, deep learning model is used as the feature extraction models, and the feature that extracted from the deep neural network models can be used as a component for the Cox regression model for prediction. The other group is called fully parametric model. So example of a fully parametric model or NN survival model. So in this model, the time to event is fully discretized as is discretized into different time intervals. So in this study, we're gonna compare these 4 different models we just mentioned to the poor cohort equation. So DeepServe and NNet survival, CoxNet, compared to the polar cohort equation for different race and sex prediction, including white female, white male, black female, black male. For our features, we use the same 7 features as it is in the polar cohort equation for comparison purpose. So that include age, total cholesterol, high density cholesterol, systolic blood pressure, history for hypertension, a smoking history, and history of diabetes. Just the introduction of a general introduction of how this different models look like. So for the deep surf model, we have as a usual, like the feed-forward network, we have an input layer and the hidden layers and the output layer. So the deep surf model would output the HWXI here, which is after we apply activation function on the final hidden layer, this would be our output. So the HWXI would then be used in the Cox regression model to replace the original XI transpose beta here in the Cox regression model. For the NNNet model, it's very similar to deep surf model, but in the final output layer, we would output theta I. So this theta I is the result from applying activation function on the hidden layer, on the final hidden layer, and the theta I here would be used to replace the right here, the Cox regression model's XI. So the XI would be replaced using theta I from the neural network model, and we still keep the beta here in the original Cox regression model equation. For the NNNet survival model, it would discretize the time to event. So instead of output the hazard in the final layer, it actually outputs hazard at different time intervals. So for example, the H1I here would be the hazard at interval 1 for individual I, and it would also have the hazard for individual I in time interval 2, time interval 3, time interval 4, and so on. Following that, the loss function for the NNet survival can be defined as this. So for the individual who failed at interval J, its loss function can be defined as the 1 minus HI, which is until J minus 1 time point, which is the survival rate up until interval J, and also times the probability of failure at interval J. So for individuals who are censored at the second half of interval J minus I, or the first half of the interval J, the loss can be calculated as this. So so we would calculate the survival 1 minus HI, which is the survival at interval I. We will multiply them all together until interval J minus I. So we tuned our various parameters of the deep learning survival models on the validation data set, and we predict the 10-year risk of ASCVD. We use the time times time cross validation to evaluate, train and evaluate the internal validation performance, and we also evaluate the model in an external validation data set. For the evaluation matrix, we use the Herald index, and we also plot the calibration plot to see its calibration performance. So for the results, these are the different race and sex groups, and the different columns are different models. So the first one is the polar cohort equation. these are the 3 different neural network survival models we tried. And in addition to that, we also tried to pull Cox-PH to including all the significant two-way interactions of our predictors. So so as you can see for the white male cohort, in the MESA external validation data set, poor cohort equation has the best C index, even though the performance are all really close between the poor cohort equation and the Cox-PH two-way interaction model. As for the 10 times 10 cross-validation result, we can see that the Cox-PH two-way interaction actually has the best performance. In the white female cohort, we can see for the 10 times 10 cross-validation, our poor cohort equation and the Cox-PH two-way interaction model has a very similar performance and has the highest index. For the external validation in the MESA dataset, the deep surf model has the best performance, but it's also just very close compared to the poor cohort equation and Cox-PH two-way interaction model. In the black male cohort, poor cohort equation has the best performance in the MESA external validation dataset. And for the black female cohort, the NNS survival has the best C index in the external validation dataset. So we for the calibration plot, this is the calibration plot for 10 cross 10 cross validation. So in the y-axis, we have an observed event rate, and in the x-axis, we have the predicted event rate. So we the way we plot this is we first would group the patient, group the sorted predictive event rate into the observed event rate into 10 groups. So we call this decile. So for each decile, we would plot the mean survival, the mean observed event rate against the mean predicted event rate in each group. So as we can see here, the predicted all these models has very similar. They are all aligned in this identity line here, so they all have pretty good performance, pretty good calibration in the white male cohort, except for an NNS survival model. It deviated a little bit towards the end. And for the white female cohort, all the models also very similar performance, except for a deep serve model and the brown dots here, the Cox-PH two-way interaction model. They underestimated the observed event rate a little bit. And for the black male cohort, so we can see here the poor cohort equation has the best calibration among all these models. And for black female cohort, the green dots here, the deep serve model has the best calibration. So for the external validation results in the MESA cohort, we can see here most of the models in all the cohort kind of overestimated the event rate, and in the so in the black female cohort, we can see, except for deep serve, which underestimated the survival rate, all the other models kind of overestimated the survival rate. And the performance of these different models kind dependent on different cohorts. So there's no one model performance better than the other model in all 4 cohorts. So for our final result, the main result from our studies is we find that 4 neural network survival models have comparable performance in the ASCVD prediction, and they didn't have a significant better performance in terms of index or calibration compared to the poor cohort equation model, but sometimes they do outperform the poor cohort equation model. So depending on the data set. And one limitation of this study is the MESA population here. We used for external validation, they received intensive screening for subclinical CVD, which would influence the health behaviors and prevent intervention, including use of effective drug therapies. So in other words, this population are being treated well for hypertension and for like subclinical cardiovascular disease. So it's expected that the population has a lower risk for cardiovascular event compared to the training data set we have. That's why we kind of observe all our models kind of over predicted the event rate. So for our study, we only used 7 predictors, which is the same for comparison purpose to the poor cohort equation, which may explain why the deep learning models didn't outperform so much, because all the predictors, they are already very carefully selected by the clinicians. So the deep learning model could may not be able to extract additional information. So for future studies, we expect to use a higher dimension of the clinical variables to see, or by including nodes, clinical nodes and images, and that might have in the deep learning survival model have better performance compared to the traditional Cox regression model. And that's, yeah, that concludes the study. Thank you.
Thank you, Yu. So we're now back on time. So any questions for Yu? Any question for the previous speaker? Yifan? Any question for Yifan? Dr. Peng?
So I have a question for Yifan. From your presentation, you compared the deep learning with this Cox model, right? But very interestingly, the deep learning alone doesn't perform better than the Cox model. I remember you have a table comparing different scores with deep learning and the Cox, they're almost the same, but the multi-modal is better. So in that slide, I feel like for deep learning, you already extracted so many additional variables, right? So many different features. Typically, we will have this overfitting problem when you have those many variables. But how come it doesn't improve at all compared to traditional Cox model? So how do you control the overfitting in your research?
I think it's a very good question, and agree with you that with the high dimensional. Well, first, compared to the Cox model, deep learning based model use high dimensional features, and it's quite easy to overfit, as you said. And to control the overfitting, we usually split the data sets into the training and test parts, and we train the model using the training sets and evaluate on the test sets. And then we do the five-fold cross-validation to make sure that the model is generalizable on this data set. And additionally, we usually have external validation sets to test the generalizability of the model. But in this study, we haven't got the external data set, and that's definitely the limitation of the current studies.
So your deep learning result is based on k-fold cross-validation?
Yes.
Okay.
Yes.
Thank you.
And it's right that when the features are low dimensional, there's no difference between the Cox model and deep learning model. Only when we add additional features from other modalities, we see that the deep learning based model can handle high dimension features better than Cox model. That's...
Okay. Thank you.
Any other question from the audience? If not, we are going to move on to our last speaker. So our last speaker is Professor Zhiyang Zhou, who is an assistant professor at the University of Manitoba, Department of Statistics. So Dr. Zhou observed his PhD in statistics from Simon Fraser University. I also graduated from that university. And he did a postdoc at the Northwestern University prior to joining University of Manitoba. So Dr. Zhou has quite a broad research interests, including deep neural network, deep learning, which I think he will be talking about. He has also research interests in functional data analysis, longitudinal data analysis, survival analysis, and the design of experiments. So Zhiyang, please go ahead.
Yeah, thank you, Professor Zhao. I hope my voice is loud and clear.
Yeah, fine.
Good.
Yeah, very good.
Thank you, Professor Zhao, for the kind and detailed introduction and the kind invitation. I feel honored to be invited to give a talk over here. Especially, I guess I never give a academic presentation in front of over 100 people, so I feel a little bit nervous. Thank you so much. Okay, so morning everyone. I can still say morning, but somehow, yeah. What I'm going to present is a deep neural network with a smooth monotonic output layer for the dynamic risk prediction. It's a collaborative work. So credits go to my co-authors from Northwestern University, Washington University in St. Louis, Cornell University, and University of California, Santa Barbara. This work was triggered by the data collected through a specific project named the Lifetime Risk Pooling Project. Such a project gathered 20 community-based observational studies and involves 200K subjects. Also, it records detailed cardiovascular disease events, and more importantly, it has over 25 years of follow-up of repeatedly measured time-varying CVD risk factors like blood pressure and cholesterol levels. So our ultimate target is to make some individual level risk prediction for CVD events. Now, let's see some formal setting. First, the time hereafter refers to the follow-up time. That is the elapsed time from the initial visit. And at first, we have to specify one scalar, T max. That's the maximum time point we are interested in. And there are capital N subjects for each subject. We've got the censoring indicator as well as the event or censoring time to the TI, whichever is smaller. Let's see the figure at the right panel. For each subject, you can see one blue dot and several red dots. The blue dots represent the initial visit, while the red ones are corresponding to subsequent visits. And the solid black box means that that subject suffered from the events on specific events, while the hollow ones and the white box means that one is censored eventually. At each visit, now y sub I of t is a vector, a very important vector. It includes all the historical information of subject I. I will cover its members one by one. First, it comes to the z sub I. Z sub I is of length dz. It contains only time invariant measurements. Well, x sub ij is of length dx. It contains time varying measurements at time tau sub ij. And the tau sub ij is the visit time for the j's visit time for the I subject. The third one is the delta tau ij. Sorry, I guess I saw a pen. Give me 1 second. The third item is the delta is the tau I sub j. That's the gap between two adjacent visit times. And the last one is Mij. It is prepared for coding missing links. The length of it is equal to the length of zi and x sub ij. So once we encounter some missing links in zi or xij, we just have to change the Mij, the corresponding Mij entry into 1, otherwise 0. So I almost forgot this j sub I of t. That's the number of visits before time t. Okay, so later speaking, y sub I of t contains all the historical information of sub I without abandoning anything. Now, our target formally becomes to figure out this, to recover this S function, capital F function. It is called survival function. That is, we know subject I lives or survives at least at time t. And we know all information up to time t of this guy. Then we would like to predict its survival probability up to time s, which is larger than time t. So this target falls into the territory of dynamic risk prediction, which makes prediction utilizing the entire history instead of values at some specific time point. There are three main streams for us to implement dynamic risk prediction. The first one is landmarking. It is less complicated in implementation and interpretation, but unfortunately, it is not fully dynamic because the predictions issued via landmarking can merely exist as specific landmark time points. The second one is joint modeling. It is more pervading. And it also allows prediction to be issued at any time points of interest. But unfortunately, it has to retain some parametric assumptions, like the mixture effect assumptions or the Cox assumption, kind of like that. So these assumptions sometimes may be taken as unrealistic. And the biggest challenge for it is the computational. It's existing in a computational aspect, because typically when we would like to optimize a complex full likelihood, we have to introduce many approximations, and eventually the resulting estimator may be somehow far from the true values. And compared with the other two, the deep learning methods may be promising, but unfortunately, most of them are not designed for repeat measurement studies, not for dynamic risk prediction. Fortunately, we've got some exceptions, includes the dynamic deep hit, DDH. So DDH actually took itself as the first deep learning method for dynamic risk prediction. As per the original paper, it is very, very competitive in terms of prediction accuracy in many numerical studies. But it also has some drawbacks. For example, the S has DDH over here means the S measure from DDH for survival function. And the S hat DDH is actually a step function. So it's bumpy. It's not smooth. And it is also restricted by such assumption that it takes value 0 at Tmax. That is, DDH assume that all the subjects had to encounter the events before Tmax. In the context of healthcare, they enforce actually all the people to get sick before Tmax, get specific sick before Tmax. Such assumption, these two restrictions somehow violate our study or people's understanding. And also it leaves us some space of improvement. Now it comes to our proposal, small, smooth monotonic output layer, small. Oh, I'm so sorry for including this math. I mean, nobody would like to see it, but you don't have to care too much about these details. From a higher level perspective, we expressed or we constructed our S measure for survival function via B splines. And that's the function we constructed from B splines. Capital O, small. It's smooth and monotonic. That's why small is named smooth monotonic output layer. After further transformation, we've got our S measure for survival function, Sn. So we just estimate the survival function directly without involving the hazard function or some other steps. And our resulting S measure for the survival function is smooth and monotonic and does not have to be 0 at Tmax. That is, we don't force all the people to get sick at Tmax. So you see, for now, the two drawbacks of DDH have been removed. That's our architecture. I mean, the architecture of our resulting networks. The first 3 modules, I mean, the three blue ones are inherited from DDH, while the yellow ones, I guess that's yellow anyway, the yellow ones belongs to us. One more word, actually, you can change the three blue module into any other neural networks. I mean, as long as you input some vector into our our yellow module, you can expect a smooth and monotonic output function. I mean, a function as the output. So our proposal is actually not only limited to dynamic risk prediction and offer more general usage, but never mind. We just over here limit our discussion to this dynamic risk prediction. Our model are changed by optimizing some very complicated loss functions, and this loss function is likelihood based. That's the main part. Wow, we also include three different penalties to enhance the prediction performance. The first one is the penalty on concordance. I'm going too deep into the details. You just have to know that we include many penalties, three different penalties, and accordingly, some tuning parameters. I guess this procedure is somehow boring, so I will skip this part. Now, definitely, we have done massive numerical studies. And over here, we just include one of them, the most important one. As I mentioned previously, this project was triggered by a data set through LRPP, a specific project. So now we're going to apply our proposed measure back to the data set from LRPP. First, we split the data by race and gender, and now we pick out the interested event as the ASCVD, some specific CVD. We include several biomarkers. Only one of them, the age, that's actually the initial age, the age at the first visit time. The initial age, we include these biomarkers, but only this age is time invariant, while all the other items are all time varying. They all vary with time. The choice of these biomarkers is according to some existing literature. Now it's a matrix to evaluate the performance of different prediction models. That's C-index, but it's a modified version of C-index to facilitate the time varying, the varying time. So no, I'm not going to too deep into the detail too, but keep that in mind. The larger, the better. I mean, when one prediction model enjoying a larger CTS over here, it is considered to be better somehow. Now you can see some results. The first one is the C-index result for the, okay, actually, to make it simpler, I only include two different prediction models because the initial target is to, because our proposal is to improve the performance of DDH and DDH, which is actually already proved to be very, very competitive. So over here, we just include two different prediction models. One is DDH, while the other one is DDH small. That is, we attach small to DDH. So you can see that in different scenarios, our DDH small enjoy a higher C-index than DDH. And you can see the same story and more scenarios. Oh, sorry, there's a missing underline over here. I'm so sorry about that. Actually, our proposal is potentially to be further extended to competing risks. That is, we can handle mutually exclusive events. And as I mentioned previously, our small is potentially to be attached to other neural networks, not just DDH. And it is also very interesting to figure out if we can figure out the theoretical bounds of prediction error of our prediction model. Okay, some references, and thank you all for kind attention. That's all for today.
Great, thank you, Taehyun. Any questions from the audience?
This is Leo again. So
yeah,
I have a question about your result. It seems like from several tables, your method, the concordance index is just like 0.01 or 0.2.1 better than the DDH. So is there any like acceptance criteria, whether this difference is meaningful, or in your research, as long as it's better, you consider it's better?
Thank you, Professor. It's a very, very good question. I have to say that not in all cases, our advantage is significant. Actually, but before going into a detailed answers, I have to. Okay, let's make it simple. Let's see. You can find that when T and S are relatively large, the advantage of our proposal may be larger, right? You can expect a 2% or 3% kind of advantage from our proposal, but and the T over here is actually the length of historical information based on which you made a prediction, and S is how far into the future you would like to make a prediction. So if you have very limited history of observations and would like to do a prediction for the near future, then you can see few advantage from our insignificant advantage from our proposal. Wow. If it is not the case, if you would like to make a long-term prediction with relatively long history of observations, it seems that our proposal may be more preferred.
Thank you.
Thank you, Professor.
So we have several questions from the chat. Paramita, do you want to ask yourself, or you want me to read the question here? Paramita? So, okay, maybe I'll just read the question. Okay, so the question is that there is a lot of focus on C-index discrimination and not on calibration. But calibration is critical in predicting absolute risk. Do you have any results comparing calibration of your models?
Yes, thank you, Professor. Actually, we have got some results in terms of calibration. And even in terms of calibration, I have to say our proposal is also somehow better. I mean, this phenomenon also exists. I mean, our proposal, the significance of our proposal is more significant. The advantage of our proposal is more significant when you would like to do some long-term prediction with long-term observations. But the advantage may disappear if you just want to do some short-term prediction with a short history of observation. I'm sorry I didn't include this result over here because this result, these slides was made earlier.
Great.
Thank you, Ian. And I also have the same question for the other two speakers.
Mm-hmm. Sorry, Professor, you mean the question about distribution of the C index?
No, about calibration as a measure of comparison between the models. I have the question for both Ifan and you.
Okay, thank you. Yeah, I hope my answer is somehow satisfying.
Yeah, thank you so much. Yeah, I understand because of time. Thank you.
Yeah, this is a very good question, and unfortunately, we don't compare the calibration in our models, and in the future, we definitely need to do that. But we are also limited to the data we have sometimes, so it's not an easy job to collect data from different resources.
And would you like to respond to that question as well?
I think we include the calibration plot in the performance evaluation, and then the result are kind of dependent on different data set, and they are all the different models has sometimes it's better, sometimes yeah.
Thank you.
Thank you.
And I see another question from Jun Zhao asking, Jun Zhao, do you want to ask yourself?
This one is actually a very simple question. So what's the distribution of the C index?
Well, thank you, Professor. Do you mean the distribution of the index for our prediction model from these repeats? I mean, kind of like these. Okay.
Yes. What do you use actually? Yeah.
Yes. Okay. So for example over here, 73.2. That's the mean of the mean C index after some replication, while 3.3 is the standard error. Oh, the unit for them are both percentage.
Okay. Yeah, this index is from 0 to 100, right?
Yeah.
Okay. Thank you. Yeah.
Thank you, Professor.
Well, I think the C index ranges from 50 to 100, not 0 to 100, right?
Oh yes, agree.
If the model is reasonably good, yeah, 50 is a cutoff for like you flip a coin, you got 50%. Yeah. Any other questions from the audience?
So I have a question for Ifan again. For the imaging processing, you mentioned you use the CNN. Did you try like any imaging processing before you do that? And what kind of CNN do you use to extract those features?
For the CNN model, we use the ResNet, which is one of the state-of-the-art model CNN for image classification. And we also fine-tune the model using the CheckS3. We use the standard data augmentation like cropping, rotation, horizontal flipping, those pre-processing on the images before feed it into the CNN.
Got it. Thank you.
Any other question? If not, let's thank the speakers and thank everyone for coming to this session. We will give you 5 minutes back for the lunch.
Yeah, thank you everyone. Great session. So the lunch break will be until 1 o'clock. So hopefully to see everyone back at 1. Thank you very much.
So Xiaohong, so we have 25 minutes, right? Or 25 minutes.
Yeah, I guess for you, if you want, you can take the whole 30 minutes.
Oh, okay. Yeah, I think probably I will control it about 25 minutes finish.
Yeah, you can decide. Sounds good.
Yeah.
So 25 minutes presentation and 5 minutes for questions, answers.
Okay.
Guys, go ahead and start if you want.
Okay.
Okay.
Okay, so maybe let me introduce Dr. Yin Yuan first. Dr. Yin Yuan is a Bedian Ash Murray Distinguished Professor and Deputy Chair in the Department of Biostatistics at University of Texas MD Anderson Cancer Center. Dr. Yuan has published over 100 statistical methodology papers on innovative Bayesian adaptive designs, including early phase trials, seamless trials, biomarker-guided trials, and basket and platform trials. The designs and software developed by Dr. Yuan's lab, which is www.trialdesign.org, has been widely used in medical research institutes and pharmaceutical companies. And Dr. Yuan has attained the prestigious honor of being elected as the ASA Fellow in 2007. Sorry, could you please mute yourself?
No, I muted them. If that happens, I'll mute them.
Okay, thank you. And Dr. Yuan wrote a book, Bayesian Design for Phase 1 to 2 Clinical Trials with Dr. Peter Saul and Dr. Wang, published by Chapman and Hall in the CRC Biostatistics series. With that, I'll pass it to Dr. Yuan.
Yeah, so thanks Xiaohong for the nice introduction. And very happy to have this chance to share some of my experience on dose optimization. I think recently there's quite a bit interest, especially from industry side, on this topic. And I think this can be illustrated by this example, sotorasib. And in May this year, FDA approved it. And I think many of us know it. And it's the first KRAS target drug. And this is a remarkable achievement because KRAS had been seen as undruggable for decades and without any success. So it's really a huge success. But this drug development has been hampered by I think I will say lack of robust dose exploration. And the FDA required Amgen to compare their approved 960 milligram with 240 milligram dose, and as one of their post-marketing requirement. And also FDA require Amgen submit a final study report by February 2023. And indicate that this study will inform possible future labeling updates. So this, I think this it's very interesting request, and it can be viewed a signal paradigm shifting with newer scrutiny and more emphasis on optimized dose for targeted drug from the FDA. And this September, FDA also released this new guidance, benefit risk assessment for new drug and biological products. And I think this is also maybe an independent event, right? But I think they are somehow related, at least scientifically. The reason in the sorafenib case, FDA require Amgen to start at the lower dose. I think scientifically it's from the benefit risk perspective. And the assumption is we can reduce that dose without really reduce the efficacy. And it immediately links to the risk benefit assessment. So I think there is overall, there is a landscape change and more emphasis on optimizing dose and optimize the risk benefit for treating patients, especially for targeted drug. And this is a slide shows the traditional paradigm. More is better. And typically what we do is we conduct a very small dose escalation trial based on the DOT, and typically it's 10 to 30 patients. And then once we finish the phase 1, probably we will do a very small cohort expansion. Sometimes we do, sometimes we don't. And then we select the MTD, and sometimes we call it a recommended phase 2 dose, because by that moment, most time we will select MTD, but certainly, in practice, we will also look, PK, PD, and the totality of data to determine that recommended phase 2 dose. But in many cases, it's the MTD, and then we move to phase 2 and 3 for further development. And this paradigm is based on build upon the development of a cytotoxic drug. And the underlying assumption is, more drug means higher efficacy. So that's why we want to, reach the highest dose a patient can tolerate, which is MTD. And if that dose is confirmed to be safe in cohort expansion, typically very small cohort expansion, 10 to 20 patients. And then we're gonna define that as recommended phase 2 dose and move forward to phase 2 and phase 3. And however, this paradigm actually, I think this is not new, right? It has been, for many years, people discuss this paradigm. It's not really suitable for novel targeted drug with different mechanism of action. And for in many cases, we know when we increase dose beyond a certain level may not enhance anti-tumor activity. So in the next slides, I will show some example. We can see clearly it happened in many targeted agents. And also another common issue in area of targeted therapy and immunotherapy is dose-limiting toxicity effect may not be even observed at clinically active doses. And also another cases is some severe toxicity may occur only after multiple cycles of experimental treatment. Okay, so this slide shows what's the difference between the MTD and optimal biological dose. And so consider, so this is the new drug. We have 5 dose level, D1 to D5. And this first row shows the true toxicity rate for each dose. And the second row shows the efficacy. And when we take the traditional paradigm, we may say, okay, this dose, maybe it's our MTD. And if we confirm it's safe enough, most likely we will move this one into phase 2 and phase 3. So that's MTD. But if you really look at the also efficacy at the same time, you will notice after dose level 2, the efficacy almost plateaued. But the toxicity actually is dose level 2, it's much lower than dose level 3. So clinically and also obviously from patient perspective, this is really a better dose. So you have almost the same efficacy, but it's safer. And this also highlighted, some real drug example whose dose and schedule were modified for safety and or tolerability after approval. And this is, I think, it just appeared in the New England Journal of Medicine. I think the author is from FDA. So you can see in many cases, like for example, for Aducanumab, and the approved drug is initially approved is 750 milligram, and later on it's modified to 450. And you can see it's not just a minor adjustment; it's a pretty big dose reduction. And again, the reason to do this is to reduce the toxicity effect. And similar, another example is niraparib. And for this drug, the approved dose originally is 300 milligram. Later on, it's reduced to 200 milligram. And there are other examples for different type of drug. And I think the whole point to show here is, when we take the traditional paradigm, and yeah, we do show, this high dose is effective. But the question is, quite often, we may have a dose which is much lower and also safer, but yield very similar efficacy. Okay, so this leads to the problem, okay, how we optimize dose, right? Since we cannot take traditional paradigm, find the MTD in phase 1, we based only on toxicity, find the MTD and directly move to phase 2 and what we can do. And I think this is also quite a frequently asked question, I think among industry, since, I think FDA, we do see FDA's push towards that direction in terms of do dose optimization, do more robust study on dose exploration rather than rush through the phase 1, find MTD and move forward. And I think a very straightforward approach is in the phase 2, rather than we study one dose, we can study multiple dose, right? So I think that's actually its approach already has been taken by some, by some study. And so what we can do is we do a phase 1. This is the toxicity-based phase 1 dose escalation. And then at the end of the phase 1, rather than pick the MTD, we pick multiple dose. Probably, for example, we pick the MTD and one dose lower, and we move to the phase 2. And at the phase 2, we randomize patient into those 2 doses. And at the end of phase 2, we pick the one dose that optimize risk benefit. And then we move that dose into a phase 3 trial. So this is a very sensible approach. And actually, you see, the paradigm the conventional paradigm we just introduced, actually it's very unique to oncology. And to move multiple dose into the phase 2, it's norm in non-oncology field. I think as many of us that's why we have a dose ranging study. But in oncology, we rarely have it. And I think this is kind of we can think, and it's kind of intermediate step between traditional MTD-based paradigm and dose ranging study. But again, because typically for the cancer trial, typically I think we have very tight timeline, and also sometimes it's difficult to enroll patients. So that's why we cannot really do full scale dose ranging study a lot of time. We just pick a couple of doses and move to phase 2. Okay, so this approach, the challenge is it's difficult to determine how many and which dose should move forward to phase 2 due to limited sample size. So in standard approach, we do a dose for phase 1 dose escalation study based on very small number of patients. And for example, if we use 3+3 design, at each dose, the maximum number of patients you treat will be 6. Many times it's 3 patients. So in here, I showed how much we really can learn from 6 patients. So for example, when you have 1 DOT out of 6 patients, when you look at the raw number of the toxicity rate, it's pretty low, right? 16.7%. But actually, if you look at the confidence interval, it can from 3% to 56%. So for the efficacy, similar rationale, right? We will have a huge uncertainty on the true response rate or toxicity rate on that dose. So in other words, when we finish the phase 1, we pick multiple doses based on the data, 3-6 patient data. So that selection is highly variable. There is a huge uncertainty. And if we choose a dose set incorrectly, for example, we miss the OBD, optimum biological dose. And then when we phase 2, it's very hard to make adjustment because we only randomize in those fixed number of doses. So that's the challenge of this approach. So naturally, a better approach is we make it adaptive, right? We let the data tell us, okay, do we want to fix that dose set? How many doses we should study? And this leads to the so-called phase 1-2 paradigm. And in this paradigm, you can see we don't really separate phase 1 and phase 2 completely. So it's a merged one phase. And during these phases, we continuously learning and optimize based on the toxicity efficacy. So in other words, there is not a point we say, okay, those 2 doses, we're gonna move to phase 2 randomize. Through the whole process, we continuously learning those doses and optimize it. And at the end of this phase 2 and 3, we will identify our optimal dose and move to phase 3. And of course, when we do this, and it's also a phase 1-2, in general, we will expect a larger sample size because it's combined the traditional phase 1 and the phase 2 together. But I think conceptually and intuitively, you will feel this is a more flexible and efficient way to optimize those. And the phase 1-2 paradigm design and the paradigm actually is not new. Okay, it has been almost existed 20 years. And one of the landmark publication on this design is a proposed FTOX design by the Shaw and the Cook. And so in that design, they join the model toxicity efficacy and have a quantity to measure risk-benefit trade-off, and then they make a decision based on that trade-off to treat this patient adaptively, eventually identify that optimum biological dose. And then later on, there is more and more a lot of design development to address different cases and also to make it more efficient. So for example, there is a non-parametric phase 1-2 design. There is a design specifically used some feature of immunotherapy and a target agent to provide a more efficient and a better dose optimization. And recent years, from last 4 or 5 years, there's more and more interest on model assist design. The reason is model-based design. It's pretty nice framework, but very complicated, difficult to understand and implement in practice. So next, I'm gonna give you a very simple example to show, give you a rough idea how it looks like, right? So in here, it's not our focus to describe model-based design, but I'm trying to give you some impression how it looks like. And so for example, this is a very typical way we model toxicity and efficacy in phase 1-2. And you need a toxicity model, you need an efficacy model. And each of them, in this case, you can do logistic regression, right? And the D is the dose, alpha, beta, it's the regression coefficient. And for the efficacy, because it's not always monotone, may plateau at a higher dose. And then you put, quadratic term, and you have a more complicated model. And because those 2 events are typically correlated and measured from the same subject, you have to do the joint modeling. And overall, you can see, without go to detail, you will see it's quite a sophisticated model, right? So this is to make it very difficult to communicate with non-statistician. And also, when you really implement this design, so you involve quite a bit, statistical modeling and estimation. And on the other hand, a more subtle issue is because the model is complicated. And in the early phase 1, phase 2, even you have like 50, 60 subject, it's still a very small sample size. And it's a sequential process. At the beginning, you treat the 3 patients, 6 patients, and 9 patients. So how this model maybe, it's very hard to, to do model diagnosis. So that means there is a big chance this model is misspecified. So when the model misspecified, and the robustness can be an issue. And so actually we do successfully implement some model-based phase 1-2 design to optimize those. So this is a recent paper I wrote with my colleague Peter, and on basically how can we use this one of the design called the late onset epitox design. So this design to really conduct the trial to optimize those. And I think we in this paper we summarized, what we learned. So if you go through it, really do this trial, you will notice how demanding it is when you do such a model-based design. And I think not every institution, company has such infrastructure or expertise to do it. So I think if you are interested in model-based design, this book I wrote with Peter, including a lot of mainly focus on those model-based design, consider toxicity and efficacy to optimize those. So in this talk, I'm more interested in the more advocate it's a model assist design. And the reason I kind of more interested in and advocate such design is because all the hard work has been done in the back end. So like you go to a good restaurant, right? So chef has been working really hard in the kitchen and the cook meal. And I think for customer, for user, you don't really worry about it. So what you need to do is just sit down enjoying the food, right? So I think that's the best based the basic rationale and the motivation of model assisted design. So before I introduce a couple model assisted design, I first introduce a couple of key concepts so you can better understand those design in such a short period of time. And the first concept is called admissible dose. So what's admissible dose? It's defined as a set of dose that satisfy the pre-specified lowest requirement on safety and efficacy. So in here, we use typically we can use a two Bayesian rule to define it. So the first line shows the safety requirement. It's very easy to understand, right? So based on the observed data, if the toxicity of a dose greater than a certain upper limit, if this posterior probability, it's less than certain cutoff. So that means this overly toxic probability is not too high. And then I think this dose is safe enough. And then the second admissible rule is about efficacy along the same line. So based on the data, if the efficacy greater than a certain lower limit, if this probability is greater than a certain cutoff, so that means data shows this dose has some sufficient efficacy, and then we think it's reasonable and it's admissible. Okay, so basically through those rules, we can define a set of dose that is reasonable, safe, and then reasonable, efficacious. Why we need this admissible rule? So if we go back to our original idea of this phase 1-2 is, you can think at each moment, what we really care is admissible dose set, right? So we want this dose reasonable, safe, reasonable, efficacious. We continuously update this admissible set, and then eventually we identify the optimal biological dose. So that's why this concept is key for the whole, framework. And I think, from other perspective, we can regard this admissible dose as a range of accepted dose between the minimum effective dose and the MTD, right? So this toxicity defines the MTD, the efficacy defines the minimum effective dose. Basically, we have a dose range within that two boundary. And then the second
yes.
one.
So can you call me later?
Yeah, I'm sorry. So if you have question, please mute yourself. The second key concept is risk-benefit trade-off. And because right now we consider risk and the benefit, we must somehow quantify them. So in here, I use a very simple example to illustrate. So the response. So remember here, response does not have to be tumor response, right? ORR. It can be any response measured by biological activity of the treatment. It can be the PD measurement, or it can be tumor response, okay? And then we have toxicity, yes or no. And then for each patient, there is only 4 possible outcomes, right? So defined by this 2x2 table. And in this case, the worst case certainly is no response, you have toxicity, that's 0 point. And the best response is the patient has a response, but no toxicity, that's 100. Okay, so once you fix those scale, and then you can give a score for the other 2 possible outcomes. And then let's say we listed this from clinician, and then a desirability of a dose can be easily defined. Because at each dose, there is a possibility how a patient located in each cell, right? And that's denoted pi 1, pi 2, pi 3, pi 4. And you just take weighted average, and then you will get a desirability of that dose. And through this device, and then we can measure how desirable a dose it is, right? Because remember, right now, we are in a 2-dimensional space because we consider both, right? It's not based only on toxicity. That's traditional paradigm we find MTD. Okay, so why here we, there is different ways to measure this cutoff. Why the utility? So I think here I list a couple reasons. First, it's highly scalable. Scalable in the sense, we don't have to restrict ourselves to binary outcome. If we have trinary, it's okay. You can imagine what you have is that table becomes a little bigger, right? So you just need to fill a little bit more score on those tables. And the second is it's highly flexible. For example, it includes the following toxicity efficacy probability trade-off as a special case. So basically what you need to do is just set those 2 cells, the sum equals 100. So basically you will get a very intuitive trade-off. So that is probability efficacy minus the probability toxicity with a certain weight. So this is the penalty. So when you increase toxicity, you receive penalty. I think everyone can understand this. This has been used in some design. And also, it's flexible enough. You set in those U1, U2, those score, and we can generate all kinds of different cases if we want. For example, if our goal is to find a dose that is safe, yields the highest efficacy, we can do that. And if we want the highest dose with equal efficacy, so basically we're trying to find the plateau point, you still can specify utility corresponding to that. Okay, so next I'm gonna give you 2 specific ways design. Use those 2 concepts and to do the dose optimization. The first one is called a utility-based Bowen design. And so this is a very simple design. There is a 2-stage. Okay, it's a 2-stage design. The first stage, we treat a certain number of patients based on toxicity. So this is the more traditional dose escalation, de-escalation. And the difference is, and then move to the second stage, what we're going to do is we, based on the observed toxicity efficacy data, we identify the admissible set. Okay, so this is our initial admissible set. And then we randomize patient into the patient on this admissible set. And the key here is we continuously update this. So this is the different from the the simple approach I talked about. Oh, we find do the phase 1, and then we pick 2 doses and into a phase 2 do randomization. In here, we don't fix that 2 doses because it's a huge uncertainty. We may make a mistake. So what we do is we take adaptive approach. We continuously update based on the data and continuously optimize this admissible set, eventually identify the optimal dose. And I think stage 1, I think many of you, heard about Bowen. It's pretty simple, straightforward thing, right? So this for stage 1. What you do is you compute the DLT rate, compare with the 2 cutoff, make a decision, escalation, de-escalation. So for example, here shows different target, what's the escalation, de-escalation boundary. So if you do a 30%, and this is the escalation boundary. It's toxicity less than 0.236, you're going to escalate. Greater than 3.58, you're going to de-escalate. Otherwise, you're going to stay at the current dose. So that finishes the stage 1. And you can see stage 1 very simple, right? You don't have to worry about a complicated model, how to fit, how to do Bayesian posterior calculation. You just compare those 2 cutoffs, make a decision. So how about the second stage? How can I continuously, update our admissible set? So that's still simple. Why? Because we already cooked the meal for you. So there is a table we can pre-tabulate it. Okay, given all kinds of possible outcomes, you can check out what's the utility for that dose. So for example, you have one. So here I shows the 6 patient treated dose. Okay. So for example, you treat 6 patients, there is a 0 toxicity, less than 1 efficacy. Right now the utility is 0. That means this dose is not admissible. Why it's not admissible? It's safe, right? But efficacy is really low. That's why it's not admissible. Score is 0. And similarly, for example, we have a dose, also treat 6 patients. There is no matter how many efficacy you have, if you have very high toxicity, greater than 2 toxicity, it's too toxic. It's not admissible. Remember, when we define admissible, we base the 2 conditions, safe and efficacy. And for other cases, for example, you have 0 toxicity, 1 efficacy, the utility is 50, and then you have 2 efficacy, that's 50.9. So you just can easily look up those utility. And then you can refine at each moment when you update, look at your interim data, you look up the table, you know which dose is admissible. Or I should randomize patient in those 2 doses. And you continuously update it. And at the end of the trial, you based on the data, you look up the utility. Oh, this dose level, those 2 is the optimal dose. Then you are done. Okay, the other approach is a single stage design. Okay, for that 2-stage design, U-Boeing, the advantage is it's allow us obtain more data regarding the toxicity profile, because you move all the way up to the MTD to identify the range, and then you optimize those. The other approach is I don't really trying to push those as high as possible. And this approach is more efficient if our goal is to identify the OBD. But the first approach is more efficient if we also want to collect more data in terms of dose toxicity profile. And in some cases, FDA do require that. They want you have more data on whole dose response curve. So those 2 designs have different utility. And in this case, I think due to the time limitation, I won't go to the detail. But again, as I promised, it's a model assist design. What you really need to do eventually is just look up this decision tree. You don't have to do model feeding, those things. You just compute the DLT rate, make a decision which branch you're going to do. And again, so when you determine the dose, utility, desirability, you still have a table to look up. So this makes your life much easier. You don't really have worry about fitting the model, all kinds of difficult things. Okay, so the key question is once you have this, how you get those tables so you can do the trial. And I think you go to the trialdesign.org. I think many of you probably know about it. So this is the website Dr. Jackie and I set up to help us, to help people to do a novel design. So if you go to the Boeing suite, so there's a decision tree which you're going to use. And here you see the U-Boeing and the type Boeing 1-2 located in here. So I think if you do there, this looks like. And I think, thank you. I think probably, yeah, we won't have time for question, but I think probably we can leave it at the end, right?
Maybe we can take one question from the audience. Okay, actually, Dr. Yuan, I have one question for you. So I noticed in your previous slide, when you calculate the utility score, if the number of toxicity, for example, is 0 and the number of efficacy is 4, comparing with number of toxicity is 1 and efficacy is 5, you actually have a higher utility score in the later case. So are you giving a better, like a higher weight to the more efficacious and a little higher toxicity dose in your computational method?
Yes, thank you. Yeah, thank you. That's a good question. I think it quite depends on how we set up this utility, especially those 2 cases, right? So you can see this case is how much you are willing to tolerate toxicity for exchange response. So in this case, we're more favorable. We favor a response over toxicity. So you can see it's have higher score than the case. You don't have toxicity, you don't have efficacy. So, but I think based on the trial objective, if you are more focused on safety, you can put this higher, this lower. So you can achieve that goal. I think the utility, the value will switch in terms of order.
Okay, so do you allow the utility score to keep increasing in this kind of pattern, or you set up a limit until the toxicity reaches some value and you don't let it increase anymore. For example, like if the toxicity score is 3 and the other one is 7, and maybe it can go to 4, toxicity 4, efficacy is 8. I don't think you need to let it increase, let the utility score increase, like, you know, indefinitely, right?
Yeah, you're right. So that's controlled by this admissible rule.
I see.
Yeah, so this is just the measure the, you know, outcome, nominal outcome we may have. For each patient, you only have four pulse outcome. So it have nothing to do with the probability, how toxic a dose it is. It's just for one patient, which how much I receive in terms of desirability for each possible outcome. So I think that's a nice thing. You know, I like utility because it's much easier to understand rather than probability. When you talk probability with clinician, say, oh, how much probability you need to trade off for another probability. It's very difficult to understand. But for this, in this case, each patient only have 4 possible outcomes. And I think it's much easier to communicate with this clinician. But yeah, you hit the key point. So we can through those 2 cutoffs to control the safety and the efficacy boundary.
Thank you.
Yeah, thank you.
There were a couple of questions that got entered into the chat. So if you can maybe have a look there, and later on when you have some time, you can
Yes,
answer.
we will leave the questions at the end of the session. So now let's go to the, we have the second presenter, Jingjing Ye. Jingjing is a senior director, currently leads data science and operational excellence with global statistics and data science in Beijing. She has over 15 years of experience in pharmaceutical industry and the US FDA, with focus in cancer drug discovery and development. Sorry. Okay, her statistical and regulatory experience expands full spectrum on patients' treatment journey from diagnosis, treatment to living with the condition. She received her PhD degree in statistics from University of California, Davis. Jingjing, I'm passing this to you.
Thank you, Xiaohong. Can you hear me and see my slides?
Yes, I can see your slide and I can hear well.
Okay, thank you, everybody. Good afternoon. And I want to thank the organizer for inviting me to give a presentation on this relatively new concept within the master protocol. And I'm sure everybody knows that master protocol is a really hot topic right now. And this is still a highly evolving research area in the clinical trial space. It's only one type of the innovative trial designs and has been used in the oncology area for a while. And it has increasing its popularity in the other therapeutic areas. You probably know that for the past year, during the pandemic, there were 16 platform trials were designed in the space. So it's a highly active area. But I think, you know, in this presentation, I just want to give you some of the research that we've been doing in terms of like a simultaneously false regulatory error. That's in the space where there's special case when there's master protocol with the shared control. And first, I want to thank our collaborators in this research, Nicole, Cindy, and William, and for working with me on this. And here's the brief outline of that, the presentations. So I want to go into first about the traditional drug development paradigm. As you can see from the left-hand side of the slides. So during the traditional drug paradigm, each treatment is studied independently against its own control. And then, you know, for three different treatments, and they are required basically 3 different studies, 2-arm studies to conduct it. And some of those actually, in fact, can be using the same control. So what's been seen as the master protocol is that, especially in the platform trial, it is utilization of a shared control, which, as you can see here, is actually the same 3 treatment can be combined into one master protocol and against the same control. And in the left-hand side, each of those will be randomized into the 2-arm independent trials versus the right-hand side, where the randomization will be among those 3 treatment groups versus a shared control. And there certainly efficiency can be gained in this space using shared control. First of all, patients can be has more opportunity to be randomized into the investigational drug than the control arm. And then there's certain efficiency can be gained, especially in the operational space, because you're gonna have a one shared platform in terms of like the database, the IRC, IRB, all those safety monitoring, things like that. So there certainly is efficiency gain in terms of that. So like I mentioned, we focus on our research on the platform trial, and that's one type of the master protocols. And this can also be called the perpetual umbrella, where those are actually is using in the one disease space, and treatment can go in and out of the platform at different times. And the advantage, like I just mentioned, there's a shared infrastructure, shared operation to potentially increase efficiency. And the research has been shown that it can reduce the sample size. And there's also less patient exposed to the suboptimal treatment. And it is more ethical to conduct. So before we get into the simultaneous error, I really want us to talk about what traditionally been talked about in the clinical trial space, which is the family-wise Taehyun error, FWER. As everybody here is very familiar with this space, it is the probability of at least one treatment is positive, assuming none is active. And as you can see from the bottom of the plot, so we actually have we plotted the number of experimental arms on the x-axis and the family-wise Taehyun error rate, FWER, on the y-axis. So as everybody here is well aware, as the number of comparisons increases, your family-wise Taehyun error increases as well. And that is for the independent two-arm studies, the greenish line here. And then we plotted the case when we calculated the eco-randomization. This means that all those experimental arm has an one-to-one eco-randomization to the shared control, and the eco number of is randomized to those investigational arm at the same time to those within the platform. So if there's a 3 investigational arm versus the shared control, so 1/4 of this 25% chance patient are randomized to the shared control, and 25% chance to be randomized to those 3 different investigational arms. So then as we use that shared control, when we actually plot this, the family-wise Taehyun error is actually smaller compared to that of in the independent trial. This is due to the positive correlation of test statistics when the shared control arm is in use. So the previous slides, you have seen that that's the Taehyun error, right? And so similarly, if we also interested in learning about the family-wise type 2 error, which is the probability of at least one treatment is negative when all are active. And we plotted again at the bottom of the slides, showing that the number of arms in the platform on the x-axis and the family-wise type 2 error on the y-axis. Sorry, it seems like some of those slides have some time control. Okay, so then if we focus on the independent 2-arm study cases, so that is the blue curve on top of the other 2 curves. So that is also going to increase as the number of comparisons were conducted. And then the other 2 curves, the right one is showing the eco-randomization. I mean, the eco-randomization is the green curve. So the treatment arms and the control have the same number of patients randomized. And the right curve is half of the shared control arm has a double number of patients than the treatment arm. So there's 2-to-1 randomization ratio is used in the shared in the master protocol with the shared control. So as you can see, the family-wise type 2 error is also smaller when shared control is used due to the positive correlation. But when there's more patients randomized to the shared control, it's approaching to the independent 2-arm studies. It's just because the positive correlation is reduced. So it's approaching that. So based on these two slides, two results, basically it looks like it's a good thing, right? Because the family-wise error, both in terms of the type 1 and type 2, are going to be smaller in that platform trial setting with the shared control than the independent 2-arm studies. So should we just go ahead and use the platform trial with shared control? Just hold that thought, because we want to introduce in this new, relatively new error that is going to be present when we have this platform with shared control, and that is called the simultaneously false regulatory error. So like I just mentioned, due to the shared control, the test statistics now are positively correlated. At the bottom of the plot, you can show that if we have through 4 experimental treatment with 1 shared control, and if the shared control is the truth, showing here is actually is the same efficacy as the experimental treatment. In other words, it basically is none of the treatment arm is in fact active. It is the same as control arm. But for some reason, the control arm, when we actually running the trial, it is random low. In this case, when 4 of those treatment arms are compared to this random or low control arm, there's going to be more than 1 chance those can be falsely rejected. So they're going to be false positives. And there can be more than 1 false positive when we have the platform trial with the shared control. So that's when the false positive simultaneously word comes into play. So that means we have more than 1 chance of false positive when the platform trial is used and the control arm happens to be random low. At another side, there also can be simultaneously false negative error. And that can occur if you switch these two, right? So then in fact, the control arm, the truth is very low. So all of those experimental treatment will be effective. However, for some reason, the control arm is in fact random high. So when you actually compare all those experimental treatment versus those random high control arm, you are unable to reject your hypothesis. You're gonna have false negative. And this chance can be happen more than 1 treatment arms. So therefore, you can have more than 1 false negative errors, and hence simultaneously false negative error. As you can think, if this platform trial is in support of registration, and different treatment arms are used into the same platform, and this thing can happen, the control arm random high or random low can happen, then either cases, the simultaneously false positive or simultaneously false negative can happen. And both of those can be considered as the simultaneously false regulatory error, because the regulatory agency is going to rely on this to make false decisions. So can we quantify that? So we want to talk about this multiple false positive error first.
Washington.
I think, can you please mute your phone? Thank you. So we will define this multiple false positive error as the probability of making multiple, at least 2, and that is where the simultaneous come into play, and at least 2 false positive decisions when none of the arms is active. So when the null is true. So this plot basically showing the multiple false positive errors when using shared control compared to the independent two-arm studies. So now the blue curve here is at the bottom of the plot. So the two independent two-arm studies are smaller than when this is the master protocol when the shared control is used. So then this two curve difference is showing the green is the equal randomization to the experimental arm, each experimental arm to the shared control. And then this red curve again is the 2-to-1 randomization, that there's more patients randomized to the shared control. So it's going to approach the independent two-arm studies when more patients are randomized to the shared control. So now you see the error reversed, right? From the previous family-wise type 1 error and family-wise type 2 error. Now there's the multiple error will increase when the shared control is in use. And that is the multiple false positive error is going to be increasing than the two-arm studies. And same thing, if we define the multiple false negative error, and that is the probability of making at least 2 false negative decisions when all of those treatments are active. And in this case, the blue curve is the independent two-arm studies, and the green curve is still the equal randomization, and the red curve is 2-to-1 randomization, more patients randomized to the shared control. It actually looks quite similar. And the line actually crosses about when there's 6 independent, 6 investigational arms are used in the same platform. And then as same as in previously slides, when more patients are randomized to the shared control, is going to approach the independent case. But this case, as you remember, this is only done when all of the arms are either active or inactive. But in reality, it's going to be a mixed bag. Some are maybe active, some arms are not. So therefore, what we propose in our research is utilizing a false discovery rate, because that's going to come very relevant in this case, while we're going to have simultaneously error. So as you know, Soric, then later Story proposed an alternative error rate, which is the false discovery rate framework. And this is basically can be shown as a 2x2 table here. So the M here will be the M number of investigational arms in the platform, and that's going to be testing against to the shared control. So you're going to have total M hypotheses. And then among those, M0 that is null hypothesis, basically M0 is not active, and M minus M0 is active, truly active. It's going to be mixed bag. And then the FDR is defined as expected proportion of those false positive among all those treatments declared effective. That is basically is those test positive, but is in fact the null is true. So that is the ratio where is this V divided by the R. And that is when the hypothesis, R number of hypotheses was rejected, but in fact, those are wrongly rejected. So the V divided by R is the false positive among those declared effective. That's the hypothesis was rejected. So similarly, the expected false negative is among those that was not rejected, but in fact, those actually the null hypothesis should be, the alternative should be true. So then the T divided by M minus R. So that is the proportion of false negatives. So then where the simultaneous come into play, right? So this is really where we have this V and T is larger than 1. So that's when we have more than 1 wrongly rejected hypothesis. So that's where we call this S FDR. So the FDR is the false discovery rate, and S stands for the simultaneously. So then we call this SFDR and SFNR. That is really where those can be more than one wrongly rejected hypothesis or wrongly not rejected hypothesis. Okay, so now we really want to quantify that. So we're gonna derive and throw out some of those formulas here when we're trying to derive this. Although it looks complicated, but the idea is in fact really simple. So this is derivation when we have a very simple case, when the M0 is known, when the number of inactive arms are known. And then we start with this and derive what is the simultaneously false discovery rate, SFDR is. So this is really can be written as M0 summing all the possibility to the total. Well, we already know M0. So then we really is summing up the where those wrongly rejected V is, and among all those M0, given that alpha and beta are the typically defined type 1 and type 2 error. And then, this is for the independent two-arm trials when we haven't considered a positive correlation. I will consider that later. So it is really to be summarizing all possibility together and using the typical alpha and betas. And those are just the formulas there. The idea is fairly simple. But in reality, the M0 is unknown. So it's going to be a mixed scenarios. So what we can do is really to summing all those M from 2 all the way to the total number of arms in the studies. So this is where this additional summation comes in. And in particular, those M0 will follow the binomial distribution with the probability P here. It denotes that probability that the arm is active. So then the 1 minus P will be the inactive probability. So the M0, basically the expected value will be M times 1 minus P, which is the expected number of inactive arms within the whole platform. And we just basically calculated based on that. And the same idea applies to the SF, the simultaneously false negative errors. We're just basically summing all possible ways that this can happen, more than one. So the interpretation is really for SFNR and SFDR is that the probability among all those M investigational arms that there can be at least 2 null hypotheses that are wrongly rejected. That is a SFDR, and the SFNR is the probability among M investigation arm that there can be at least 2 alternative hypotheses that are wrongly not rejected. So we will show this into a numerical setting so that you can have an idea where those numbers are. So those plots basically showing the when there's 4 investigational arm, 6, 10, and 15 are using the same platform. And then the black curve is the simultaneous false discovery rate, the false positive errors, simultaneous false positive errors, and the red curve is the false negative errors. And then the x-axis here is the probability of active arms. So the more probability of this is the probability of active arms basically means if this is 60%, the arm is active among those 4 investigational arms, 4 times 0.6 is roughly 2 to 3 arms that is in fact is truly active. And then so same, we just plotted how those simultaneous error rates are. And as you can see here, when P is in fact 0, and that means all arms will be inactive. And when P is equals to 1, that means all arms will be active. So all those arms should be truly active. But then, as you can see, as the arm increases, this false negative error increases. So then maybe a good illustration can be saying, okay, so when there's, say, 80% chance that the arm is actually active, and when there's only 4 arms in the platform, and that's equivalent to be 3 of those arms in fact truly active, and then the chance of that you are unable to identify at least 2 out of those 3 arms truly active is in fact 10% when there's only 4 arms in the platform. But when there's 15 arms in the platform, and this same probability increases to be 40%. That means, a 15-arm trial, 80% arms that are active, that equivalent to be 12 of those arms are truly active arms. But you have 40% chance that you're unable to detect at least 2 out of those 12 arms that is active in the platform. So it is not something you're just gonna to ignore, and especially you want to get the drug approved, right? But at the same time, for the simultaneously false negative error, it actually stays relatively pretty low, and almost everything was small, consistently smaller than the typical 0.025 level. So even though it's increased from the independent studies, it's still relatively low. So what we presented is with the independent case, now really considered as the correlation, and this is really simple, just to integrate the correlation into the distribution. So instead of those binomial distributions, we're just gonna have a covariance matrices, and those covariance matrix is the positive correlations, and those are directly, it can be written as the number of patients in the treatment arms over the number of treatment arms plus the number of shared control arms. So when these equal randomization, this correlation will be 0.5. So then this is going to be how you're going to calculate the simultaneously false positive errors and false negative errors, and given this correlations. And here's the results showing that. So then the left-hand side plot showing the simultaneously false discovery rate, and the right-hand side showing the simultaneously false negative false non-discovery rate. And then the black curve is the independent studies, and then the 3 color lines are showing the randomization ratio as 1:1, 1:2, or 1:3. The 1:3 is showing there's 3 times more patients are randomized to the shared control than the treatment arms. So those curves are going to be approaching the independent studies, and in both cases. And then as we heard earlier, the simultaneously false discovery rate is going to increase from the independent study, even though numerically all of those are still relatively small, even at the time where there's 15 investigational arms is in the platform. And then the simultaneously false discovery rate is similar to the independent cases in this case. And then one other good property is, like I mentioned earlier, when we introduced the simultaneously false discovery rate, we defined the multiple false positive error rate and multiple false negative errors. Those are when every arms in the platform is non-active, or all of the arms is active. So as you can actually see here, there's actually 2 special cases as what we define for the simultaneously false discovery rate. So if the multiple false positive error will be equivalent to when the p is 0 in our definitions for the simultaneously false discovery rate, and then this will be when p equals to 1 when there's and all of those arms are active, and those can be you can see that's exactly the same when we actually show the plot. So these are just special cases of our definition for the simultaneous error. And I think I'm running short on time, but I mean, you can ask me, okay, so what do I have to know about this in the real world, right? So then, because many times when we're using the platform trials, those are actually in fact running by different companies. And if they're actually, submitting their trial results to the regulatory agencies, and they are, they're not going to have control over other companies' data, or be able to influence too much on how to control the simultaneous error. And that is a fair, comments on that. So then we did running some of the additional analysis just to show at the design stage, those can still be considered, depends on what you really want to achieve into the master protocol. So those can be designed as, design changes, whether you want to have a more randomization into your shared control arm, which is in the 1, 2, 3 randomization. It's going to approaching really the independent 2-arm trials, or you can increase or decrease your type 1 error, rate to really make it approaching to the independent 2-arm trials. So those are basically how the simultaneous error can be compared to the independent 2-arm trial when you actually made the design changes at the planning stage. And these are numbers assuming only 30% of those arms are active. And you can change that to any other percentage. As we already shown the formula, you can just plug in and calculate that. But what I'm going to say is, no matter what, the simultaneous false discovery rate and simultaneous false negative rate are still relatively small in terms of numerical values. So what we're really trying to show in our research is that when the shared control is used in the platform, there's in fact a different kind of error than you typically is for everybody here to be aware. And first of all, as you heard, the family-wise error rate is actually lower, both for the type 1 and type 2, but then you're gonna have those simultaneous false positive and false negative errors can occur. Even though the false positive errors is larger, false negative error seems to be, is actually similar to the independent 2 cases. But I mean, as we're showing here, the strict type 1 error control is really unnecessary compared to different regimen to the shared control, because numerically, it is really fairly small. So I think that's all for the references. Thank you.
Thank you, Jingjing. Actually, I have one question here from Bidan. How is this multiple arms in platform trial different from or compared to the multiple doses in the phase 2b dose ranging trial?
Right. Yeah. So then actually, it is actually, you can think, generally it is quite similar. It is, right? So I mean, but what I'm going to say, there's the statistically you can think is similar, because if you want to only choose and move 1 dose forward, and that is going to be that is going to be different from where you want to see if individual drugs is in fact is effective compared to the shared control. Because all those different doses are for the basic same compound. You just either move some doses or all of, you are unlikely to be able to move all of doses forward. But then for individual drugs, you do want to make individual decisions on each of those drugs. So I think that is one distinction here in terms of like the different doses.
Thank you. Any other questions? Okay, thank you, Jingjing. If there are more questions, we will leave it to the end. Let's go to our third speaker. Our third speaker is Dr. Taehyun. He is a senior manager in dermatology of immunology statistics at AbbVie. Taehyun was a key contributor to the successful submission and global approval of Skyrizi auto-injector and new formulation. He's currently a lead project statistician for multiple development programs. In the past 4 years, Taehyun has published multiple methodological manuscripts, including 6 as a first author in adaptive clinical trials, Bayesian statistics, machine learning, and multiplicity control. He is also an active referee of 7 statistical journals. Taehyun received his PhD degree in biostatistics from University of Michigan, Ann Arbor. Okay, Taehyun, I'll pass it to you.
Yeah, thank you, Xiaofeng. Can you see my share screen and hear me?
Yes, I can see your screen and I can hear you clearly.
Yeah, thank you for the introduction, and also thanks Xiaofeng and Ziqian for organizing this session and inviting me to present our work. So for my talk, I will talk about practical response adaptive randomization design. And we also will talk about two associated efficient inference procedures. So here are some disclosure statement from AbbVie. And also this is a joint work with Lu from UCB, Ziqian Yihua from AbbVie, Lan Ju from Vertex, and also Evan from BMS. So for my presentation, I will first talk about the introduction and then introduce or propose the RAR design. Next, I will talk about those two types of efficient inference procedures. After that, I will show some simulation results, and then there are some discussion at the end. So for this type of response, adaptive randomization design, so the intuitively it's relatively straightforward. So essentially, we want to assign more subject to the better treatment group based on or accumulating outcome data. So it's like one type of adaptive design. And also based on the relative finalized FDA guidance on this adaptive design. So statistical, ethical, and pragmatic rationale support this type of design. So for statistical, usually it means like we may see a higher power. And for ethical, it's essentially we are randomizing patients to the better performing arm. And pragmatic means that maybe patients are more likely to stay in this trial because they are receiving a better treatment. And we all know that there are many types of this type of RAR design. And for this DBCD, this is a doubly adaptive biased coin design. Is one of the most popular design. And essentially at the study design stage, people will specify a desired allocation function, for example, to maximize the power in a two group design or try to minimize the failure rate, something like that. So essentially they turn to this as an optimization problem. And based on their type of design, their cumulative final sample proportion will lean towards to this desired allocation function. And also recently, there are many this type of design to adjust covariates. So for the motivation of our work is more on the seamless phase 2-3 design. So this is kind of similar to our first speaker in presentation. So he focused on this seamless phase 1-2. But for our scope, it's more like we want to integrate the phase 2 dose ranging study and the phase 3 confirmatory study. And to meet the regulatory their requirement, usually we need the final sample size for our placebo group and this selected treatment group to reach a pre-specified target value. So for example, we need to fully evaluate its safety profile. And also, for example, we need to obtain sufficient exposure data and calculate, for example, the compliance of our study drug administration. So that's kind of a potential issue of the previous DBCD design as we discussed before, and we have some simulation results later on to further demonstrate this kind of question. And to address this problem, so we propose this called response adaptive block randomization design. So motivated by our previous work. So essentially we try to adaptively randomize subjects to different treatment groups based on their order of a standardized effect size. But as we see later on, for this certain measure, you can substitute with other measure. You can also adjust for their covariates or use different measures. And also we want to have this pre-specified block randomization factor. So essentially we want to facilitate its application in practice. So we may pre-specify those schedule in IRT system. So now let's look at a simple setup for demonstration. So we suppose we have a continuous outcome here, and we use small I to denote the subject and the G to denote the group. So we use G equals to 0 to denote the placebo, and G from 1 to M to denote M active treatment groups, and we assume a normal response. And in that case, at the study design stage, we can define this so-called block randomization vector R. So you have R0 as the constant. I mean, the block in a certain block, you have R0 number of subjects to placebo. So you can treat it as a fixed. So you always, for example, have 1/3 of subject randomized to placebo, and then you have R1 to RM in a decreasing order. So you may imagine that we will try to assign more subjects to R1, and the least favorable treatment arm will be to RM. And for demonstration, we consider this two-stage study design. So we just have one interim adaptation on randomization. But later on in simulation, essentially we can sequentially adaptively randomize each subject enrolled. And in the first period, usually we have this so-called burn-in period. So we equally randomize all the subjects to M+1 group. So we have M active treatment arms and also +1 the placebo group. So it's we try to get an initial knowledge of the performance of different groups. And then in the second stage, we can adaptively randomize those subjects because we got the data from the burn-in period. So we try to calculate their standardized effect size, and we can assign those R1 to RM based on those decreasing order of this effect size. So the best treatment group, they are assigning to R1. So it is the largest block size. And for R0, that was always for placebo. So we try to fix the placebo because agency, when we design this confirmatory clinical trial, agency usually favor that we fix the placebo rate. And we use this superscript 1 to denote the first stage, but for the second stage, that's for 2. And as we mentioned before, this type of design, we try to facilitate its implementation in IRT systems. So let's see how it goes. So suppose we have M equals to 2 active treatment groups. And for the block size, suppose we have block size of 6, and we have, for example, R0 equals to 2. So 1/3 subject always to placebo, and 3, so about 50% to the better treatment arm, and 1/6 to the worst performing group. So at the design stage, so essentially we can pre-specify those schedules. So for example, the first schedule, this is equal randomization schedule. So we have P denotes placebo, L for low dose, H for high dose. So that's equal randomization. And for the second dose, you can see that the low dose gets 3. So it means that low dose perform better in this schedule. And then for the third schedule, maybe high dose gets 3. And during the burn-in period, so remember we got some initial data. So in the burn-in period, we always active the Q1, the schedule 1. So we got the equal randomization. And after this burn-in period, so for example, if we want to sequentially adaptively randomize each subject enrolled, we can calculate this measure, this R measure. So that will be the standardized effect size, as we mentioned in the previous slide. And if the low dose performing better, we active Q2. So low dose is performing at 50% probability of randomization. And if high dose is performing better, we active Q3. So imagine if you have more active doses, eventually you can pre-specify more schedules in advance. And as our second speaker, Jing mentioned before, so this family-wide error rate control is very important especially in confirmatory clinical trial. And for this type of design, because we have multiple doses and multiple comparisons, so essentially we have 2 dimensions. So within the comparison between each treatment group versus placebo, how to control this type of error rate, because we have the adaptation at interim to modify or randomization probability. And the second dimension on the top of that, we also have this multiple comparison. So how to properly control its family-wide error rate, especially in the strong sense. And we introduced two kind of inference procedures. So the first one is like we use the unweighted statistics to address the first question, and then use some existing multiplicity adjustment to address the second one. And the second procedure is like we use the CHW statistic or the inverse weighting combination function plus the newly proposed multiplicity adjustment. So now let's first look at this first inference procedure. So in general, if we have adaptive design, for example, the sample size re-estimation design, we will may observe an inflation of type 1 error rate. But for this type of design, actually we can prove that the traditional unweighted statistics will have controlled type 1 error rate at the nominal level. So the justification or the intuition is that we assign more subject to the better performing group. So that's kind of similar to the promising zone approach. That's kind of different from sample size re-estimation, because in that kind of design, we assign more subject usually to the worst performing group. So in this case, we will may observe a deflation of type 1 error rate. So actually, in this type of design, you can still use your traditional unweighted statistic, and you can still get an accurate type 1 error rate at nominal level within your comparison. And for multiple comparison, so let's write it straightforward, because for each group, you get a control type 1 error rate, and then you can apply some existing multiple comparison approach, for example, Bonbroni or even Downnet, and to control your family-wide error rate. So for the second inference procedure, so for within comparison, so that's also some existing work, because when you have some adaptation in your trial modification, so usually we can think of CHW statistic or inverse normal combination test approach. So essentially by combining those p-values from different stages using pre-specified weights, we can still get accurate nominal level error rate control within this comparison. And for between comparison, so that's kind of our motivation of our work. So we try to aggregate all the existing treatment groups to new treatment group, and we try to utilize this closure test principle to conduct hypothesis testing, because eventually this response, adaptive randomization, we assign more subject to a better performing group. So intuitively, more subject, usually a larger sample size corresponds to a higher power. So in that case, when we aggregate existing treatment group to new group, so when we look at this closure testing principle, we got more subject in our active treatment arm, especially in those better performing groups. So we may observe a better power. So that's a high level intuition of our proposed multiplicity adjustment. So now let's look at some simulation studies. So for the first one is about this inference procedure one. So remember we have those unweighted statistics for a pairwise comparison. And we consider this clinical trial with 3 active treatment groups, so D1, D2, D3, and placebo. And then we remember previously we want to have this toy example with a 2-stage design. So in the burn-in period, we will have 60 or 20 as the burn-in period. And for the total sample size, that would be 2 times this burn-in sample size. And then for those subsequent second stage subject, they are adaptively and sequentially randomized to this trial. And then we use this S1 to denote the selected treatment group. So because we only have one and only one at the final stage, so for our analysis. So at that stage, we conduct our analysis and try to select the best treatment group based on their effect size or even the smallest p-value. And we use S2, S3 for the second best and the worst group. So for our study objective, so we want to identify and confirm the efficacy of this best performing group. For example, we want to control this family-wide error rate at one-sided 2.5%. And also we want to achieve a desired final sample size for or selected treatment group and placebo. So that it would be to meet our regulatory requirements. So for this one, essentially we are looking for the type 1 error rate control. So for the box I labeled here, that would be for a benchmark from non-adaptive design. So essentially without multiplicity adjustment for each pairwise comparison, we got approximately 2.5%. And then for this proposed unweighted statistic under or RABR design, so we can see that the type 1 error rate has a deflation. So that will be still controlled. So you can still use your unweighted statistic in your inference. And then for the next slide, we are looking for their potential power. So we use the downnet to correct this multiplicity issue. And then we have several comparators. For example, this fixed means or traditional eco-randomization design, and also DBCD with certain different hyperparameters. So we can see that generally our method has a higher overall power. So that would be confirm and identify the best treatment arms. And also even or D3, so that's the best performing group. So that would be the probability of selecting this arm and confirm the efficacy of this specific best arm. So generally, our proposed method has the highest power across all the scenarios we evaluated. And for this part, it's about the question of reaching the desired final sample size. So essentially, for example, we want to have placebo reaching 35% and the selected treatment arm reach 35%. So we can see that under those scenarios, so for our method, generally, this was well achieved. But for the third scenario, that was the hardest one, because if you see their mean response, so the best treatment group has 1.2 and the second best has 1.15. So that would be kind of the hardest scenario because those two arms have very similar performance. But when you see on the right-hand side for the DBCD, actually its final size will be very different because its allocation function depends on this underlying response rate. So the conclusion of this slide is that our method can more robustly reach this desired final sample size in order to meet regulatory requirements. And now let's quickly see some simulation results for this inference procedure too. So for this type of design, so that's the first row, and we want to compare with the traditional seamless phase 2-3 design. So they potentially drop certain arms at interim and then use those inverse normal combination testing approach. So for the conclusion here, in the first block, you will see our method also have an accurate type 1 error control. That's expected because with the CHW statistic and also the closure testing principle. And also for the selected row and the overall row, when we look at the power, so generally our proposed design also has the best power performance as compared with this seamless phase 2-3 design and the traditional Bonferroni or traditional downnet test with fixed randomization. So in that case, we have 3 active treatment arms. So you may imagine what happens when we have 10 active treatment arms. So generally, the power gain will be even higher. So for example, in this case, we have over 7% as compared with this seamless phase 2-3 and much higher than the Bonferroni on the downnet test. And also this is for the global power and also for the selected power, that would be even higher, 75% versus 60 or 40 or 50. So I think due to the time constraints, I will not go into the detail of our case study, but eventually the take-home message is that in some study, we don't have a previous dose finding study. And in that case, usually the eco-randomization is the best one we can go with only a phase 2 and then followed by phase 3. But if we want to redesign it later, given that they have very similar safety profile maybe we can develop this RABR with a better performance. So again, the message that we got higher power as compared with this fixed randomization and DBCD. And also it gets a more accurate average sample size at final, because we want to reach placebo at 72, S1, the selected arm at 72. So in general, it has a more accurate average sample size at the end. So for some discussion, so for this type of design, we want to try to address the different questions from the DBCD. So the comparison essentially, we want to focus that we want to robustly and accurately reach the target allocation of placebo and this selected treatment group. So that will kind of have a different objective from the DBCD. So especially this objective is more applicable to our pharmaceutical industry, because we need to meet the regulatory requirements of the exposure response analysis, or also need to build a sufficient large safety profile. And for the unweighted statistics in inference procedure 1, so it's relatively easier to compute than these weighted statistics. And also it allows you to sequentially randomize each subject, because imagine after each subject you adaptive randomize. In that case, you cannot calculate the weighted statistics, because with only one single subject, the statistics may not exist, or it just give you a missing value. And of course, you can also do some free combination. For example, you can do this unweighted statistic plus or newly proposed the multiplicity adjustment. And also the inference procedure 4, that would be the most straightforward one. So you directly use the CHW statistic and some existing multiplicity adjustment. And for our work, I think this first one discussed about the inference procedure 1. So we have this RABR, and also we use unweighted statistic in the pairwise comparison and the existing multiplicity adjustment for multiple comparison. And for this work, we also have our package developed. So if you are interested in the performance, you can evaluate some scenario versus some other typical seamless phase 2-3 design. And finally, our inference procedure 2 was summarized in this manuscript. So essentially, we try to aggregate different treatment groups and utilize the closure testing principle to control the family-wise error rate. And here are some additional reference I mentioned in this presentation. Yes, that's the end of my presentation, and thanks for your attention.
Thank you, Taehyun. Now we can take questions from the audience. So Taehyun, maybe I just ask one very dumb question. So actually, I remember you mentioned it's it's difficult to handle the questions without the design you proposed, like in the large phase 3 studies. So originally, why were you thinking about this? What's the real example motivated you?
Thanks, Xiaopeng, for the question. I think originally we have a phase 2 study, and we want to utilize it as a confirmatory trial. So that's the motivation of our group. We try to propose this design. But after several rounds of negotiation, I think currently we kind of abandon this type of design and stay with the burn-in period, the equal randomization, because we still want to use this as one of the confirmatory studies. So for some departments, they are relatively conservative on this adaptive design, but the motivation is based on one of our phase 2 study at AbbVie.
I see. So what you're saying is after you propose the design, it was actually not used in the end.
Yeah. So as we mentioned before, let me go back to this slide. So essentially, the good part of this RAR is that at the burn-in period, you still have the equal randomization. So you can still specify all your activities, and you can approach regulatory agency to see if they can buy or innovative design. So at the end, the team decides the benefit risk is not that much, and then we just use the Q1 all the way to the end. So our trial is just the equal randomization design. and we just abandon, for example, Q2, Q3. But if regulatory agency accepted, we can still initiate this kind of program. So during the burning period, give us some room for this kind of negotiation and also save some operational time.
Thank you, Taehyun, for the introduction on the history. Actually, I think time is up for our session, but I think there's one question. Jingjing, you already answered the question from Jun Zhao, and also I think there's one more question from Ahmad, and he asked a question. I think this is for Dr. Yuan. In most cases, safety or toxicity is observed earlier than efficacy and could be months apart. How can you incorporate the combined information without time delays? The reason why I saw the answer from Dr. Yuan, but since this is one of the important research topic for you in the recent years, I think it's worthwhile for you to answer it directly.
Yeah, so yeah, thank you for the question. So I think there's a couple ways we can address it. The first approach is we can try to identify if there are any biomarker, surrogate biomarker can be used. For example, some PD measure. And remember here, so as long as this biomarker reasonable enough, it should be fine. So because what we do is during the process, we use that biomarker to update the desirability of each dose. Potentially, I think we can use adaptive randomization to assign the patient, like our last speaker. So can assign patient to the better treatment. And you can imagine, even you have some deviation, it's okay, because after all, it's randomized, right? Now, at the end of phase 1/2, you can wait a little bit. When you get everyone scored in your clinical efficacy endpoint, and then you pick the winner. So that means you can use a relative rough surrogate biomarker to guide the patient assignment and adaptation, but it won't affect at the end. You pick the so-called optimal dose based on your final endpoint of interest. So that's the first suggestion. Second one, you can use more simple design. Actually, I used a couple of times in the trial, depends on the setting. The other one is, for example, I cannot find that surrogate biomarker. What can I do? A simple way is, for example, when you use U-Boy, at the second stage, you don't really update your admissible set that frequently. You can update, for example, after 10 patients, and based on your observed data. And in that case, because you have already finished stage 1, stage 2, you already treat a couple, 10 or 20 patients. So some patients' efficacy already get scored. And in this case, you have some data to guide patient allocation and adaptation. And again, when you reach the end, you still can use, after follow-up, you can use the ultimate endpoint to pick the OBD. That's the second approach. Last approach is, okay, you still want to do more adaptation as you want. And I think this methodology can handle, for example, titer point 1-2. Titer means time to event. So in that case, the basic idea is use observed data to predict what's going to happen for those patients whose response has not been scored yet, and use that to guide patient allocation. But again, the patient allocation is just to optimize patient benefit and also kind of concentrate the patient around the OBD. So at the end, when we do phase 3, we have more data to make a better decision. But it's not totally critical, because at the end of phase 1/2, you still have a chance to look at totality of the data, make the final decision. And that's also one concern people always have when they use utility or trade-off. Oh, I cannot use that as single metric to pick the optimal dose. So that's true, because in practice, it's always complicated. And so what we typically do is in the protocol, we will say, okay, we're going to use that as to guide our dose assignment and allocation. At the end of the trial, that's a recommendation. We're going to use a reference, but we will use the totality of data eventually to pick the OBD. So I think that gives you some statistical principle and the rigorous at the same time give you some flexibility to accommodate what really happen in practice. So I think that's some approach we have been adopted. So hopefully,
thank you. Thank you very much, Yang, and thank you for the whole session and all the speakers. We are already way over time, so I just want to say thank you all for your presentation, and we will skip this break. How about that? You know, we can start the next session right now. So, session chair Saurabh, are you here?
Yeah.
Hi. Yeah.
Hi, Lee.
Hi, Xiaotian. Yeah, you are the chair. Sorry.
Yeah.
Please
thank
go
you.
ahead.
Yeah. Thank you, Lee and Saurabh. Yeah, it's my great pleasure to chair this session. So everybody, welcome to this session. So today we have very good presentations, and yeah. So yeah, for the audience, for your questions, please leave your questions to the end of the session. Okay, so let's get started. So first speaker is Qing Xia. So Qing is a PhD candidate at the Department of Biostatistics and Data Science at the University of Kansas Medical Center. And her research lies in batch effect correction of longitudinal genomic data using empirical bias and simulation-based sample size calculation. And she was a summer intern in Astellas Pharma and working on time-to-event dose-finding designs to accelerate oncology phase 1 trial. She developed the R-Shiny application to compare operating characteristics of existing design. So take it away, Qing.
Thank you. Thank you for the introduction. Yeah, I'm gonna share my screen. Okay, so hello everyone. My name is Qing, and thank you for being here. So I was summer intern at Astellas, and during the summer, I was doing this. I was being involved in this joint research work with Kentaro, Shufan, and Islands. So today I will talk about the time to event Bayesian optimal interval design to accelerate the dose finding, accounting for the toxicity grade. So we call it the tight J-Bo-in design for short. So in today's presentation, I will briefly introduce the background of the study. And then I will talk about the methodology of our proposed design, tight J-Bo-in. And in order to evaluate the performance of our methods, we compare with other designs. We conducted the simulation study and obtained the operating characteristics. So I will illustrate the simulation result. And then I will end up my presentation by a brief discussions. Okay, so the main goal of the phase 1 clinical trials is to evaluate the safety of the new drug or therapy, and also to identify the maximum tolerated dose. We call it MTD for short. That is defined as the highest dose level with an acceptable level of toxicity. And here the toxicity is usually a binary outcomes, which is called the dose limiting toxicity DLT. So each patient either has DLT or not is the Bernoulli trial with a probability is the DLT rate. As you can see in these figures, the DLT rate is actually an increasing function as the dose level increase. And after the phase 1 clinical trials, we're gonna recognize MTD levels, dose level, and this dose level will be passed to the phase 2 to evaluate the efficacy. So here is a simple example about the dose finding studies, the complete data design with the binary toxicity. So we start as the lowest dose level, and based on each cohort complete data, then we make the decision for next cohort. So here, however, for the molecular target agent or immuno-oncology therapies, which often presents the different toxicity grade instead of the binary toxicity. So if we pick the cohort number 5 outcomes as examples, if we ignore the toxicity grade and only use the binary outcomes to indicate the outcome of the cohort, so we only got one patient out of 3 patients observed the DLT. So we will make the decision as the stay for the cohort number 6. But if we take the toxicity grade into the considerations, we observe the 1 patient experienced the DLT, and with 2 patients have observed the low or moderate toxicity grade, such as the grade 2 toxicity, which is less than the 1 DLT. So then we will have less than greater than 1 DLT out of 3 patients. So it's not safe. We still make the decision for the stay. Another thing is the study design proposed for the binary toxicity is not applicable for the study design with the toxicity grade. So that is the to solve the problems. Many approaches consider the toxicity grade into the dose finding clinical trials by assigning the severity weights to each toxicity grade, and then transfer to a composite score. So here I list 3 studies which are most relevant to our studies. The first study is Yuan et al. In 2007, they proposed a MATTERS, which is equivalent toxicity score, ETS approach. This score actually converts the toxicity grade to the numeric score, which is the unit of the DLT. So if we normalize the ETS, this normalized ETS is between the 0 and 1. So they also show that this normalized ETS is the quasi-Bernoulli outcomes, which has the similar property of a binary endpoint, which means we can use the Bernoulli likelihood to deal with it. And Dr. Mu proposed the JBOE MATTERS, which consider considering different toxicity grade scoring system, not only the equivalent toxicity score, they have other numeric scoring system. So, but they, however, for the novel anti-cancer agents, such as the immuno-oncology therapies, who usually have the late onset toxicity, which means they need longer assessment windows. So that slows down the trials. And also when the patient alcohol rate is very fast, then when the patient entered the study, but the study are still suspended for the DLT evaluation for the pending patients. So we need to turn away this patient. That is the waste of the patient resources. So to solve the issue, the time to event tight designs are proposed to allow the sequential enrollment for new patients, even though the previous patients enrolled in the study are not finished the DLT evaluations. So I will use a simple example to show you the difference between the complete data design, such as the JBOE MATTERS, or the time to event design, such as the tight JBOE MATTER we proposed. So in the complete data designs, this study first enrolled 3 patients, and the number 4 patient can only be started after the previous 3 patients get finished for the DLT evaluations. And before this time point, we have 3 patients enter the studies, but because the study is suspended, so we need to turn away these 3 patients. But the situation is a little bit different when we use the time to event designs. So we also enroll the first 3 patients, and the number 4 patient actually can get started, even though the patient 2 and the patient 3 are still pending for the DLT evaluation at this time point. And by using the time to event design, we successfully enroll these 3 10-weight patients into the studies. So the last slide for the introduction is the structure of the full study design we included in our simulation studies. The buy-in design is the model, is the foundation of our matters, and the buy-in matter is the model existed study design. So it's kind of combined the good features of the simplicity in practice and also have a good performance. And based on the buy-in matters, Yuan et al. In 2018, they extend the Bowen method to tight Bowen methods by allowing the sequential enrollment of the patients. And another direction here, the Bowen method is also extended to the J-Bowen methods by Dr. Mu and the teams using the different scoring systems to convert the toxicity grade. They can consider the toxicity grade into the dose finding studies. And our method here is an extension from the J-Bowen methods. We're gonna use the weight functions of the pending patient to construct the weighted likelihood. And by doing that, we can allow the sequential enrollment in our studies. Okay, so next slide, I will introduce you the method part of our methods. Before that, I think it's very helpful if I can give you the notation, some introduction of the notation I'm gonna use in these presentations. For the complete data design, such as Bowen method or J-Bowen methods, we kind of use the binomial data. NG is how many patients being enrolled, and XG is how many patients have experienced DLT, which is how many succeed. And they have the probability PG here is the probability of the DLT or the probability of quasi-Bernoulli probability that is monotonically increased with the dose level increase. For the time to event study design, such as the tight Bowen design or tight J-Bowen designs, at the certain trial time tau here, we observed the tilde XJ, which is how many patients we observed the DLT, or the normalized total DLT unit in the tight J-Bowen method. And here the NG is how many patients have been treated. RJ is how many pending patients. MJ is how many completed patients. The last thing is the UI, which is the follow-up time for the pending patient I. This variable is very important because we're going to use these variables to construct a weight functions and allow us to include the pending patients into our studies. Okay, so this one is the equivalent toxicity score. We use the ETS score because that is the measurements combining the different toxicity grade into the unit of the DLT. So to calculate a target ETS, we use a predefined toxicity grade profile, which is provided here. This is proposed by Dr. Yuan in 2007. And in these papers, in this profile, they kind of give a link between the toxicity grade and the DLT. For example, for the grade 0 and 1, that is equal to the DLT equal to 0. And for the grade 2, that is equal to 0.5 DLT. For the grade 3, that is equal to 1 DLT. Grade 4 is a little bit higher, that is 1.5 DLT. So for each categories levels, we have a toxicity profile, which is a probability, and all these four values end up to 1. So you can see based on this toxicity profile, we are also able to calculate the target DLT rate in the binary outcomes, which is we sum up the 0.23 to with 0.1, which is equal to 0.33. That is the target DLT rate. But if we use the ETS scoring system, that means we need to calculate the ETS. So here we use the DLT and the toxicity profile using this formula to calculate the target ETS is 0.47. This value need to be normalized by the 1.5 because the maximum of the DLT here is 1.5. Then the normalized target ETS is the quasi-Bernoulli endpoint because it is between 0 to 1 and it has the similar property as the Bernoulli outcomes. Okay, so before we talk about the tight J-Bowen method, I think it's very helpful if we give a little bit review about the J-Bowen methods. For the J-Bowen methods, it's designed to deal with the Bernoulli trial, which is a binary endpoint, and the quasi-Bernoulli and the continuous toxicity endpoint. Here, we only focus on the quasi-Bernoulli toxicity endpoint. So we're gonna use this formula to calculate the quasi-Bernoulli toxicity probability. So you can think this probability is similar as the probability of the Bernoulli trial. And then this one can be calculated by the summation of yi divided by how many patients enter the studies. But I need to specify here, yi is not the observed outcomes. Yi is a normalized observed outcomes. For example, we observe the patient I have the outcomes is 0.05. We need to divide it by the maximum of the DLT, which is 1.5. So the normalized values is the quasi-Bernoulli toxicity outcomes. And after we calculate the estimate of the probability, then we are ready to compare this value with two predetermined boundary, lambda E and lambda D. I will talk about this later in detail about how to calculate the lambda E and lambda D. Okay, so in order to extend the JBOE method to the tight JBOE methods, we constructed the weight functions, which is between 0 to 1. And we also assume the UI, which is a follow-up time for the pending patient I, is follow the uniform distributions. And the weight functions for the uniform is just the CDF of the uniform. So here is examples. For the patient 1 to patient 4, all these 4 patients are complete cases. So the weight function, the weight for these 4 patients are all equal to 1. But for the patient 5 and the patient 6, they are still pending. And we need to calculate the weight functions using the follow-up time divided by the assessment windows, which is 28 days in these examples. So these two weights are going to be used to construct our likelihood functions, which is showing here. So the step 1 for the tight JBOE methods is the construct the weighted likelihood functions. We inspired by the Dr. Li and Dr. Yuan in 2019. And in this paper, they extended the completed design to the tight design using the likelihood functions targeted to the binary outcomes. So we kind of extend this one to the quasi-Bernoulli outcomes. And then the likelihood is constructed as following here. The first 2 terms are very similar as the Bernoulli likelihood. So here is the probability of the success and the numbers of success, and the probability of failure and the numbers of failures. Another thing here is this part, the third term here is a place we put the pending patients. So from this step to this step, we use the Taylor extensions. We use the first moment of the Taylor extension to get the approximations. So finally, we got the likelihood functions is as follows. In this formula, the XJ, as I mentioned, it is the total observed quasi-Bernoulli toxicity outcomes. And the tilde MG here is how many patients observed without DLT, and plus the total weight of all pending patients. So finally, we constructed the effective toxicity data. You think this one as the binomial data. Here is the effective total size, total sample size, and this one is effective success. So we can use this effective binomial data to calculate the estimate for the quasi-Bernoulli outcome probability μg hat here. After we calculate this one, we are ready to compare this estimate with lambda E and lambda D to make the decision for next cohort of patients. So here, we calculate the lambda E and the lambda D using this formula. Within these 2 formulas, we only have phi1, phi0, and phi2. These 3 values are unknown, but the phi0, which is the target ETS score, target ETS, and the lambda and the phi1 could be set at a default, which is 60% of the probability, and the highest probability phi2 can be default set as the 1.4 times the target probability phi0. So after we have this value, we are ready to calculate lambda E and lambda D. Okay, so in order to evaluate our methods, before I talk about the simulation, I want to make a small conclusion about the method part, because the tight J-Boolean design is developed based on the J-Boolean, the Boolean design. So that belongs to the model assisted methods. So it can combine the simplicity in the practice and also the good MTD estimation accuracy. So in order to validate the tight J-Boolean design, we constructed the simulation study. So we include 4 different study design, as I mentioned before: the Boolean design, tight Boolean design, J-Boolean design, and tight J-Boolean designs. And we consider 10 different scenarios, which is first published in Dr. Mu's papers, which consider which we have the MTD defined as the dose level who has the ETS closest to the target ETS. I will talk this later. So the maximum numbers of the patient is 13, and the assessment window is 28 days. The time to toxicity follows the variable distribution, and we have this. We utilize the same safety rules, and we use the 50% pending patients result in the trial suspensions. Okay, so here is the 10 scenarios. So for each grade, we have DLT outcomes showing here. And we have the scenario number 3 for 3, 6, 7, 8, 9, 10. We have the MTD defined by ETS and the MTD defined by DLT rate are not matching. So we keep the MTD defined by ETS to help us determine which one is the MTD. Okay, so to calculate the target ETS for the each scenario, to calculate the DLT rate for each dose levels, we sum the probability for the grade 3 plus the probability for the grade 4 to calculate for the DLT rate shown here. Okay, so we also consider 2 actual settings. One is faster actual settings. Each patient entered the study after 7 days. And the default setting is each patient entered the study after 14 days. So that means within 1 assessment windows, we got 2 patients enrolled. We considered 6 different performance metrics. The first one is the percentage of correct selection and the percentage of correct allocation. So these 2 values are performance metrics to evaluate the MTD estimation accuracy. So the higher, the better. To evaluate the overdosing problem controlling, so we use the percentage of overdosing selection, percentage of overdosing allocation, and the percentage of early terminations. So these 3 values, the lower the better. The last thing is we also want to evaluate the speed of the trial using the average trial durations. So next, let's talk about the simulation results. So since we have the 2 different actual settings, but they have the similar patterns, so I only focus on the faster actual settings in today's presentations. The first take-home message for us is when we compare the PCS, PCA, which are the performance metrics to evaluate the MTD estimate accuracy, the higher the better. So we got the tight J-Boolean method has the highest PCS and have relative high PCA compared to the J-Boolean method. It's very similar, a little bit lower. So we conclude that the tight J-Boolean methods has high accuracy in the MTD estimations. And the second take-home message, when we look at these 3 columns, which are performance metrics to evaluate the overdosing problem. So here, the lower the better. So we see the tight J-Boolean methods have the lowest value for these 2 columns and the relatively low values for the percentage of early terminations. So we conclude that the tight J-Boolean methods can control the overdosing problem. The last take-home message is the most important because that evaluates the trial durations. So after we compare the tight J-Boolean method with the J-Boolean methods, we conclude that the tight GBO emitter can shorten the trial duration by around 26%, which equal to 105 days in the faster ACRO setting, and 11%, which is 62 days in the default settings. So here is all, I will show you the scenario specific plot for the faster ACRO settings in order to give you straightforward feelings of the performance for 4 different emitters. So the green line, the purple line are GBO emitters and the tight GBO emitter respectively. So we can see for the first 2 plot, the green line, the purple line are quite high than other 2 designs. And in the middle 2 plot, we can see the GBO emitter and the tight GBO emitter are quite low when we compare to other design, which is good. And for the last plot showing here, we can see the tight BOIN and the tight GBO design have very short trial durations, but combined with other two performance, we're gonna conclude that the tight GBO design combine all these matters, all this good performance in other performance metrics.
Sorry
so
to interrupt, Qing. Just a gentle reminder that we have about 2 minutes left. If you can quickly wrap up. Thank you.
Sure, thank you. So last, I will give you a brief discussion. To summary, based on these 10 scenarios, we think the GBO emitter and the tight GBO emitter are extremely helpful for the study with the toxicity grade. But because they have relative high MTD estimation accuracy, and they can control the overdosing risk. And when we combine the trial durations, we can see the tight GBO design are the only one design we can combine all these good features. But before we use these methods, we're gonna need the clinical people to carefully consider the quasi-Bernoulli endpoint and also the toxicity profile. And we need the biostats team to evaluate using the simulations. And the tight GBO emitter in today's presentation only used for the quasi-Bernoulli outcomes, but it can be extended to the continuous endpoint after we change the likelihood functions. So this manuscript is already submitted to the journal. And thank you, Kentaro, Chufang, and Alan for this work. And yes, so that is my presentation. Thank you.
Yeah, thank you, Qing, for the very nice presentation. Yeah, so okay, so let's move on to our second speaker. I'm happy to introduce our second speaker, who is also my colleague in AbbVie, Hong Li. So Hong is a clinical pipeline statistician, and she had more than 10 years of clinical trial experience in pharmaceutical company and clinical institutes. And she actively advocates the creative innovation solution to improve the trial efficiency and effectiveness. And she promoted and integrated the adaptive Bayesian study design in AbbVie clinical studies. She's also interested in the statistical methodology development and its practical application in drug development. And she had published more than 30 scientific topics in the stats and the clinical journal and conference. And she will, today she'll present her recent research on handling death as an intercurrent event in time to recovery analysis in COVID-19 treatment clinical trials. The research aims to raise awareness of the importance of appropriate strategies for constructing a robust time to event analysis and to shed light on the planning of current or future COVID-19 treatment trials. Okay, so Hong, floor is yours. Could you unmute yourself? Hong?
Hi, Sheldon, can you hear me well?
Yeah, I can hear you now.
Oh, I would like to make sure you can see my screen well. It's the full screen of my slides, or?
Yeah, I can see your first slide.
Oh, first slides, not the full screen or presentation?
I think it's a full screen mode.
Okay, great. Yeah, thank you so much for attending our session of innovative design and analysis in time to event trial. I would like to also appreciate the conference committee offering the great opportunity for us to share our research work on COVID-19 study. I'm Hong Li, a clinical statistician from AbbVie. I will present a topic of handling competing death in time to event analysis in COVID-19 studies. This research was actually inspired by AbbVie COVID trials in the consideration to rigorously evaluate COVID therapy and appropriately interpret the findings and the results. It will be highly relevant and beneficial to current and future COVID study design and analysis planning. This is the disclosure slide. In the next 25 minutes, I will mainly walk you through the background introduction, methodology, simulation study, and conclusions. In the COVID clinical trial evaluating treatment effects on time to recovery, an appropriate strategy to handle intercurrent events of death becomes particularly critical. The intercurrent death can be also regarded as a competing risk to prevent observing the outcome event of interest. For example, in our COVID study, the death will hinder the observation of recovery events or hospital discharge, which is usually our primary efficacy endpoint or key secondary endpoint in COVID study. FDA and EMA provided guidance to recommend a composite strategy for death handling, with the death taking a sufficiently unfavorable value or undesirable value, and not simply considering death as a form of missing data. The competing death has a potential impact on time to recovery or hospital discharge estimation. Given one illustrative example here, assume the treatment has no benefit to improve the recovery rate or hospital discharge rate compared to the control group. That is, the hazard ratio of hospital discharge or hazard ratio of recovery is equal to 1. But when the hazard ratio of death is greater than 1, for example, 4 in the scenario 1, that implies more death induced or observed by treatment. Then we will end up with observing less subjects to be recovered or discharged from the hospital in the treatment group due to more diseased and died subjects who can never be recovered in the treatment arm. In the contradictory case scenario 2 here, if the hazard ratio of death is 1/4, less than 1, less subjects died in treatment group. Intuitively, we are able to observe more recovery subjects in the treatment arm. Therefore, the instance of recovery is actually coupled with the competing death. Up to date, the most popular alternative approach to analyze competing risk data employs the cumulative incidence function, abbreviated as the CAF, as the statistic. The CAF, cumulative incidence function, is defined as the marginal probability of outcome event of interest, regardless of the occurrence of competing risk. The incidence function is a product of outcome event hazard, time. Sorry, any question? Actually, the instance function consists of the product of outcome event hazard times the overall survival. The overall survival in the paradigm of CIF, in cumulative incidence function, actually incorporates the hazard of outcome event of interest and the hazard of competing risk. The cause-specific hazard of outcome event and the competing risk are assumed to be independent, but it doesn't mean the incidence function of event and competing risk have to be independent with each other, although they are mutually exclusive, and the incidence function from all possible events should be summed up to 100%. It's worth to note that the overall survival takes into consideration that a subject must survive from all competing risk events in order to experience the event of outcome interest. The cumulative incidence function, CF, is simply the integration of incidence function from time 0 to time t. There are a couple of well-established CF-based competing risk analysis methods developed in the past decades. The first one is non-parametric CF approaches developed by Croy. Basically, Croy simply defined the non-parametric fashion of hazard of outcome event and the non-parametric Kaplan-Meier overall survival. The between group comparison was implemented by a modified log-rank chi-square test. Another method is called the subdistribution hazard model, developed by Wang and Croy. It's actually another semi-parametric proportional hazard model. In terms of the subdistribution hazard, I would like to discuss more regarding its definition and how it's related to cumulative incidence function, CIF. The regular hazard ratio is defined as the instantaneous rate of outcome event within event-free subjects as the risk subject population. The subdistribution hazard evaluates the same instantaneous rate of outcome event within the event-free subjects, but also including the subjects who experience competing risk. In our case, the diseased subjects who died during the study will be included in the risk set as well. And you can intuitively perceive the risk set as the denominator to evaluate the subdistribution hazard. It implies that the diseased subject died subjects during the study will have no chance to experience recovery or hospital discharge. And their time to recovery or time to hospital discharge will be set as immortal infinite time, which is aligned and complies with the FDA EMA guidance to treat the death as the sufficient undesirable or unfavorable value. One of the nice features of subdistribution hazard is that the subdistribution hazard ratio corresponds to the ratio of CIF in a direct relation. In the presence of competing risk data, the regular survival analysis, such as Kaplan-Meier, Cox proportion, restricted mean survival, may yield potential bias. As we discussed before, the overall survival in the paradigm, in the context of CF, cumulative incidence function, incorporate both outcome hazard and the competing risk hazard, as shown here. However, the overall survival computed in the regular survival analysis, such as Kaplan-Meier, did not include the effect of competing risk hazard, which leads to always upward bias. The regular survival analysis actually assumed the subject who experienced the competing risk would have the same distribution of the outcome interest as the subject who didn't experience the competing risk. In our case or COVID study, the regular survival analysis will assume the died subjects follow the same distribution of the recovery or hospital discharge as the alive subjects in the study. Kaplan-Meier approach always produce overly estimated cumulative incidence function, as we discussed before, and thus the CF's cumulative incidence function from all possible events may add up to more than 100% given the Kaplan-Meier algorithm. The overall survival from the Kaplan-Meier will be the same as the CF overall survival, only in the case there is no appearance of competing risk data. And thus, we conducted the simulation study to evaluate different survival analysis along with the different censoring strategy in the context of competing death. We investigated regular survival analysis methods, including Cox proportional hazard model, log rank, restricted mean survival with the two different censoring strategies. The first one, to censor the death at the death time by the non-informed censoring rationale. The second censoring strategy is to censor the death at the maximum follow-up time period, 28 days during the study. In the competing risk analysis, CF-based approaches, including the non-parametric CF test and the subdistribution model, were compared to the regular survival analysis approaches. It's worth to note that the competing risk CF-based approaches and the 28 days imputed censoring strategy are in compliance with the FDA recommended composite variable strategy by taking the death with the sufficient unfavorable values. The simulation setting were based on most commonly seen COVID-19 trial attritions. The time to event data was generated by both the cause hazard driven model and the latent failure time model. More technical details can be are provided in the reference paper in this slide. The median discharge day in control group are considered as 8, 12, and 16 days. And hazard ratio of discharge changing from 1, 1.3, and 1.45 to demo the treatment effects on recovery from no benefit to more compelling benefits. Death hazard ratio is ranged from 1 to 1/4 to indicate the treatment on the death effect. In each simulated trial, 180 subjects in each arm and 10,000 iterations for each analysis method except the CF non-parametric one. And the one-sided alpha 0.025 was adopted to claim the superiority efficacy claim. For the sake of additional clarification, the cause-specific hazard ratio of discharge or recovery was used to generate the simulation data. But in the presence of competing death, the hazard ratio of recovery or hospital discharge equal to 1 does not necessarily imply no treatment effects on recovery or hospital discharge, since the incidence rate of recovery is coupled and impacted by the competing death as well. Due to the time constraint, I will mainly focus on a couple of representative simulation scenarios. In the case of hazard ratio of recovery is 1, it implies treatment has no benefit to recovery. However, the death hazard can be changed from 1 to 1/4. In the panel plots, the x-axis indicates the hazard ratio of death, and the y-axis denotes the probability to declare a significant treatment effects from all different methods and the censoring strategy. From left to right in the x-axis, the death risk is reduced in the treatment group. We evaluate the proportion to claim the significant efficacy, which is indicated by y-axis. In a low death rate of control group, all analysis methods exhibit the same results with the low chance to claim significant inference. However, when the death rate in control group increases up to 15 or 25%, if the treatment has less risk to reduce the death or less death subjects observed in the treatment group, For instance, the death hazard ratio is 1/4, less than 1. The competing risk analysis methods, including the subdistribution model, non-parametric CF methods, along with the regular survival analysis, to impute the death as the 28 days censoring, demonstrate a higher probability to claim the significant treatment effects. The regular survival analysis, including Cox proportion hazard, log rank, restrict mean survival, to censor death at death time with a non-informative censoring rationale, will retain the low chance to claim the treatment effects regardless the varying death hazard ratio, as shown here. In a brief summary, when a death rate is low, all methods yields very similar results. In the case of moderate or large death rate, competing risk and 28 days imputed censoring strategy based on the regular survival analysis have a greater chance to declare the significant efficacy. When the discharge hazard ratio increases up to 1.3, we are still able to see the separation of the competing risk methods and the 28 days imputed censoring strategy differentiated with the regular survival analysis with the non-informative censoring strategy. In other words, when the treatment has the positive effect to improve the recovery rate or hospital discharge rate, and also help reduce the death rate, the competing risk approach, including the CF non-parametric test, subdistribution model, and the all other regular survival analysis, adopting the 28 days imputed censoring strategy will help enhance the success rate to claim the treatment significant efficacy. The results of recovery or hospital discharge hazard ratio equal to 1.45 are similar as the finding we observed before. The chance to claim significant treatment effects reaches to the plateau greater than 90% due to the hazard ratio of discharge is as high as 1.45. And we are supposed to achieve the plateau probability of significant results out of the data. But we can still see the separation of the computer risk analysis and the 28 days imputed censoring strategy versus the non-informative censoring strategy. In conclusion, we aim to raise the awareness of appropriate handling in current competing death in the drug assessment of time to recovery in COVID-19 study, especially when the death rate is moderate and high. In general, the non-informative censoring strategy is not recommended. Computer risk analysis and the censoring death at maximum follow-up time can be considered as one as the primary, another as the sensitivity analysis in compliance and align with FDA and the EMA guidance for the competing death handling. Our research work strive to shed light on the planning and the designing stage for the current and the future COVID-19 treatment trials and geared towards the robust treatment evaluation and assessment. For the implementation convenience, we provide the SAS procedure and the R package for all above mentioned methods. I would like to greatly appreciate our team members and the AbbVie Study Innovation Group support and their valuable inputs for this research work. Thank you.
Okay, thank you, Hong, for the very interesting and informative presentation. Let's move on to our third speaker today. It's also my colleague at AbbVie, David. So David has 30+ years of experience working in the biotechnology and the pharmaceutical industries providing study design, data management, statistical analysis, product development plans, and the regulatory support, including submissions, defense, and approval. And he started his career at Syntax Research, which later became Roche, where he spent the majority of his career. And in this capacity, he worked on several therapeutic areas, such as pain disease, ulcer disease, pulmonary disease, organ transplantation, virology, osteoporosis, and solid tumor. And after a long career with Roche Genentech, he joined Pharmacometrics AbbVie, where he has been working in hematological malignancies. So in this capacity, he has collaboratively worked on very interesting problems like this one he's presenting today. Okay, so David, you may start.
Can you see me?
Yeah, I can see you from camera, and also I can see the
slides. Okay, thank you. So the title of my talk is Bayesian Interim Decision Strategy in Phase 3 Time to Event Study. And the goal of this presentation is to demonstrate that the use of Bayesian interim decision strategy for stopping a time to event trial for futility. Surab and I jointly worked on this problem, but I want to acknowledge his major contribution in arriving at this proposed solution. The next slide is the disclosure slide, and I'll go to motivation and challenges. Accumulating data in a long-term trial trials can be very informative in the context when the study population under consideration is naive non-Hodgkin lymphoma patients, and when such oncology trials include short-term surrogate. In such a case, there are some specific challenges. One is the patient population is heterogeneous due to one subgroup of patients with shorter time to event versus the remaining group of patients with prolonged time to event. The second one is such heterogeneity is not linked to reliable prognostic baseline covariates, which can be used in either randomization or at the analysis stage. Finally, due to this mixture of time to event interval, the overall treatment effect can vary over time so that the interim estimate of the effect cannot reliably predict the final outcome. Therefore, such interim analysis require rigorous planning on when and how such interim assessment needs to be done. Study design and setting. In our case study, we assume a study with a sample size of 840 non-Hodgkin lymphoma patients to be enrolled over a period of 36 months with a 1:1 allocation to treatment and control. After 6 cycles, approximately 6 months, each enrolled subject will be evaluated for complete response, and the subgroup of subjects who achieve CR are likely to experience a prolonged time to event interval compared to the non-CR subjects. The trial is designed to stop when we have a total of 336 events. For regulatory approval, the study will need to show that the new treatment benefit significantly in the overall population, irrespective of how many achieved CR status in which arm, and the comparison between the arm will need to use the log-rank test in the overall population. Model parameters for the overall distribution. So PG is the proportion of subjects with the CR, where G takes 1 if it is treatment or 0 if it is control. And lambda G superscript C is the constant hazard rate for CR subjects, where the median time to event will be given by log 2 lambda G superscript C. And lambda G without the superscript is the constant hazard rate for the non-CR subjects, where the median time is given by the same formula. Using the above notation and assumption, time to event for a subject in each arm will follow a mixture of 2 exponential distribution with the resulting density. And the density is given as the proportion of non-CR subjects multiplied by the exponential density for the non-CR subjects, plus the proportion of CR subjects multiplied by the exponential density for the CR subjects. Model assumptions for the parameters. From the historical data, we assume that about 50% of the subjects are expected to achieve CR in the control group. 2, the median time to event in the non-CR subjects is 24 months, but in the CR subjects, it's about 180 months, which is much longer than the non-CR subjects. And it is also assumed that the probability of survival in the CR subjects between the treatment groups are similar. Overall hazard rate and hazard ratio. So the hazard function expression is based on the true assumed underlying proportion of the two subpopulations and underlying true hazard rates of subjects within those subpopulations. The hazard function expression depicts the instantaneous hazard rate over time for a subject who is randomly selected from the mixture population. Once we have the hazard function expression for both the control and the experimental, then we can derive the hazard ratio for the experimental versus control arm, and we see it to be non-monotonic and exceeds 1. From the figure of the hazard curve that displays on your right, for a given hazard ratio of 0.7, the non-monotonicity of the curve and the time at which it crosses 1 are functions of the proportion of CR subjects in each arm, and also a function of the CR rate differences between the arms. And this is also reflected by the differences in power for these various combinations. Key parameters of interest. Although there are many parameters that are considered, there are two aspects of effectiveness for the treatment effect. One is the improvement of survival time in the non-CR subjects that will be measured by hazard ratio λ1 over λ0. And 2 is the improvement of proportion of subjects achieving CR that will be measured by delta P, which is equals to P1 minus P0. The heat map was obtained by using simulation studies according to the study design, the two key parameters of interest, and the expected study execution parameters, which are planned number of enrolled subjects, randomization ratio, dropout rates, and enrollment rates. The heat map show how the power of reaching statistical significance using log rank test increases as both parameters, hazard ratio and delta P, or either of them demonstrate large benefit in relation to experimental arm. So a good decision rule or strategy should consider both estimates jointly along with the uncertainties at any interim assessment. Bayesian decision framework. So accumulating data in a long-term clinical trial can be very informative to make important futility decisions regarding the trial. Bayesian predictive probability of success is a very useful decision matrix since it considers estimates of the key parameters of interest along with their uncertainty. In the Bayesian setting, probability of success can be defined as follows, where psi of dA theta is the conditional power of the study given the data dA has already been observed, and pi theta given dA is the posterior distribution of the key parameter of interest theta. But there are many challenges to compute the probability of success in a complex study such as this. One challenge is to obtain the probability of success given the interim data. And in this setting, there is no formula available to compute the conditional power and the computationally it will be very expensive to simulate it because we will have to consider every possible intermediate data dA. So to define the following are our decision probability success hat of dA, and our decision metric is given has in this formula, where psi theta is the power of the study given theta instead of the conditional power, which has already been computed for various combination of hazard ratio and delta. Illustrating using simulated accumulating data at 24 months. So suppose that after 24 months from FEI, available data will be analyzed to make a go, no-go decision. We generated a simulated trial data according to the study design. The generated set is given on the right side using a hazard ratio of 0.7 and delta P equals to 0.1, for which we have already computed simulation, but we'll pretend that the simulation information is cut when the simulated value reaches the time interval of 24 months from first patient randomized. Even though for this example, we use specific values of the parameter, we'll pretend that the values of these parameters are unknown. In this case, we observe 67% or 540 subjects out of the 840 were enrolled by the time of the first randomized subject reached 24 months, of which 420 subjects would have had their CR status known, while the remaining 120 subjects would not have had their 6 months radiological assessment, and therefore their CR status will be unknown. In addition, a subset of subjects would have had their time to event occurred, while the remaining would have been censored at this time. The table on the right displays this information. Assigning prior to the posterior probability of the key parameters. So the conjugate prior for the parameter proportion of CR is taken to be the beta distribution, and the conjugate prior for the hazard rate lambdas are taken to be the gamma distribution as given below. We choose hyperparameters such that all priors are weakly informative and thus cover a wide range of likely values of the parameter in both arms. So the proportion of CR follows a beta 2-2 distribution, and the hazard rate for the non-CR follows a gamma 10,340, while the hazard rate for the CR subjects follow a gamma of 2,350. This implies that the median time to event can vary widely, and the 95% confidence interval in this case will be 51 months to 678 months. Thus, all priors are weakly informative. Computing Bayes posterior distribution at interim. Yeah, so any Bayes inference involves computing posterior distribution that is defined using the Bayes rule as given in this formula, where the L of theta D is the data likelihood, and where pi theta is the joint prior for the parameters, that is delta P and hazard ratio, and PD is the normalizing constant. From the above expression, we estimated the posterior distribution for joint parameters delta P hazard ratio given the observed data D24. Simulated from MCMC draws using STAN. The figure on the right side shows the density for the parameter delta P and hazard ratio when delta P is equals to 0.1 and 0.7. Now we look at the distribution of probability of success. Using the posterior distribution for the interim mock data at 24 months, we estimated the probability of success hat is 0.73. However, since probability of success hat is a conditional probability conditioned on a randomly observed data, hence it is a random variable, and we can generate hundreds of virtual trials. And for each simulated trial, we can generate a probability of success hat at 24 months, which is the interim decision-making assessment. Those estimated values can be used to estimate the distribution of POS hat. The heat map was obtained using a pair of values of delta P and hazard ratio. In this case, delta P equals to 0.1 and hazard ratio is 0.7. And the probability of success density for 24-month interim data is given on the right. The question we are trying to answer is what should be the threshold for probability of success hat for a go decision versus a no-go decision. So distribution of POS at 24 months on the delta P hazard ratio grid. So our goal here is to determine what threshold value should we use for the POS to make a go decision and for a no-go decision. Here we have POS distribution for 9 pairs, delta P hazard ratio values. By studying these grids of densities, we can have a general understanding how informative a probability of success hat value will be for different underlying set of parameter values. For example, if the POS is 0.7 or 0.8, then it's most likely it's coming favorable to the hazard ratio less than 0.7 and delta P greater than 0.5. So we should be supporting a go decision. But if it is less than 0.2, then it may be a signal to stop. However, if it is in between, then we may have to do a further assessment to assess the benefit. Now let's go to two-stage design. So we can formalize this concept into two-stage design, decision strategy. This two-stage decision strategy will be as follows. If POS at 24 months is greater than go-1 threshold, then the study can continue. Else, if POS is less than no-go-1 threshold, the study stops. But if it is in between, then you look at the POS at 36 months, and if it is greater than go-2, then continue the study, else stop for futility. Performance of this two-stage decision strategy. So now that we have formalized our two-stage decision strategy, we need to assess the performance of this two-stage decision strategy using virtual trials. In any quantitative-based decision strategy, we should evaluate its performance before implementing it. This can be done through simulation of virtual trials, where within each virtual trial, the decision metrics are obtained and the final outcome of the trial is noted. For our two-stage decision strategy, we can carry out such simulation to record the estimated POS hat at 24 months and 36 months, respectively, and record the final outcome of the virtual trial based on the log rank p-value. Once these values are computed for several virtual trials with various set of parameter values, we can use the database to evaluate the performance of the strategy easily. For example, let us consider the following two-stage decision strategy for a fixed set of threshold at the interim decision assessment of 24 months. So if POS is high, say greater than 60%, then we will continue the study. Or if POS is very low, less than 20%, then we stop the trial. But if POS is between the low and high, then we do an interim decision assessment 2 at 36 months. And if POS at 36 months is greater than 62, then we continue the study, else we stop for futility. The table below shows the performance of the two-stage decision strategy for the above fixed set of thresholds. And the table shows that the two-stage strategy given above is yielding an accuracy of 90% when the alternative hypothesis is true, and 98% when the null hypothesis is true. So empirical selection of thresholds. Sorry. Okay, performance. Wait, let me get my thing here. Sorry for the disturbance. Okay, empirical selection of thresholds using accuracy plots. Table discussed in the last slide summarizes the performance of the decision rule for a chosen set of thresholds. The performance may be good enough to implement those thresholds, but we may still want to see if we can improve the performance of this decision rule. Since we have pre-computed POS values at 24 months and 36 months, and the final outcomes of each of the virtual trials, we can easily produce similar table for another set of threshold values by simply counting the frequencies and the proportions of how often we are making the correct go or no-go decision. To illustrate this, we consider 4 sets of thresholds where we vary go 1 taking the values 0.6 and 0.65, and go 2 taking the values 0.62 and 0.65. That is, we have 4 combinations for go 1 and go 2, where we have 0.6, 0.65, that is the bottom left plots, 0.6, 0.62 is the top left plots, 0.65, 0.62 is the top right plots, and 0.65, 0.65 is the bottom right plot. And we are holding the no-go one fixed at 0.2. Then we compute the accuracy of the decision rules and plot for the various combination of hazard ratio, where the hazard ratio can go from 0.5, 0.7, and 1.0, while the delta P can go from 0.1, 0.05, and 0, as shown in the figure. Since our goal is to achieve high accuracy on the target and the null profiles, then the go 1, go 2 is equals to 0.6, 0.62, which is the top left plot, would still be the best choice among the 4 threshold choices that we investigated. ROC curve to evaluate the decision strategy. Consider a single stage strategy at 24 months to make the final go, no-go decision based on a POS hat threshold. And this can give us an ROC curve by varying the POS threshold go 1 and plotting the correct go rate or the true positive rate, TPR, which is shown on the vertical axis, against the incorrect go rate or the false positive rate, FPR, which is shown on the horizontal axis. For example, we see that with go 1 equals to 0.6 gives a 75% true positive rate, that is the correct go rate, with a 20% false positive rate, that is the incorrect go rate. So the ROC curves are popular. It provides a decision-independent structure to understand the discriminating power of the decision matrix. Both ROC curves being so further away from the random prediction, we get the assurance that the POS hat has a high discriminating power to predict the outcome and can be used as an effective metric in decision making. These 2 ROC curves together can also be utilized to come up with a good set of thresholds for the 2-stage decision strategy. Expected utility of rational decision making. So ROC curve do not commit any set of utilities, that is penalties or rewards associated with the correct go or incorrect go, or correct no-go or incorrect no-go decision, and therefore are not ideal to calibrate the thresholds. Expected utility theory deals with how people should make rational decisions under uncertainty, which is given by this formula, where delta represents the go or no-go decision based on a set of assumed POS hat thresholds at 24 and 36 months. And O represents the final outcome. And if it is positive, O is equals to 1. If the final log rank p-value is significant, and if it's negative, then O is equals to 0 if the final log rank p-value is not significant at the end. We assign utility for various consequences and estimate the probability of each consequence from the virtual trial and and assumed thresholds. We define the rational decision rule delta star that maximizes the expected utility defined above by this formula. And researchers have applied such expected utility framework to evaluate and calibrate predictive models. Each action in the given table can lead to an outcome. And if we call this action outcome combination a consequence, in our example, there are 4 into 2, 8 possible consequences. Please note, when we decide to stop the study, which is delta 3 or delta 4, delta equals to 3 or delta equals to 4, we do not observe the final outcome in the real trial. But when we simulate a virtual trial, we still could check the final outcome in those situations, and this will help us to evaluate the good decision rule. Calibration of threshold using expected utility. So a standard decision analysis framework assigns one of the best possible consequence, a utility of 1, and least possible consequence, a utility of 0. So the go action at IDA 1 having a positive study, we assigned 1, which is U11 equals to 1. And the incorrect go at IDA 2 is the worst, and that is U20 equals to 0. We also think that the correct go action will have higher utility than the stop action, and the table below gives this thing. Surface plot evaluates the expected utility for the go 1 and go 2 threshold, and the expected utility is maximized at go 1 equals to 0.62 and go 2 equals to 0.624, and with a maximum utility value of 0.68. Summary. So a quantitative rigorous two-stage interim decision-making framework is proposed that implements a very complex pattern of survival data, formally utilizes the short-term surrogates such as CR status that may be available in such trials from decision-making. In this presentation, proposed decision matrix POS to compute the predictive signal of study success, discussed how to evaluate the performance such a decision making using virtual trial, and using ROC framework showed POS has a very high predictive power, using expected utility framework to discuss how to tailor the strategy by properly calibrating the threshold for specific study setting, and how to determine a good time to start such treatment. Ongoing investigation is to further and leverage historical data, use adaptive sampling, and also to incorporate beneficial treatment effect in time to event for the CR patient groups. Thank you very much.
Okay, thank you, David, for the nice presentation. I see now we are open to Q&A, and I think we have about 10 minutes for the questions, if our audience have any questions. I think we don't have any so far from the chat. So maybe I have a question for Hong. So for the second presentation, so I think so this is using the competing risk framework. It's not very common in the traditional setting, and so regarding the regulatory perspective, I believe you have some communication with the the regulatory. If so, can you tell us a little bit more about your experience? How they received this proposal and analysis. Some quantities like CIF, rather than the traditional quantities. So, yeah.
Yeah, hi, Shatin. That's a great question. I think it's always critical to have the close alignment and collaboration interaction with the FDA and the regulatory agency to ensure our analysis robustly evaluate the treatment effects. Actually, per the current literature, due to the COVID-19 pandemic urgency, a lot of trials were designed in rushed manner. The regular survival analysis that adopted the like two censoring strategy we mentioned in the simulation study have been widely applied to the current COVID trial. But there are a lot of literature already raised the awareness we should be more cautious to consider the competing death in the overall survival analysis. That's why we in our research work, this work also develop a manuscript development. We provide the reference for the to compare the regular survival analysis methods versus the competing risk approach, including the CIF-based methods, has been actually adopted for multiple COVID-19 trials, including clinical trials and the studies. I think it's under the way to get to the FDA or regulatory agency fully aware of that the overall comprehensive evaluation for the treatment effects on the recovery, incorporating the death effect. That's why in our final recommendation, we would suggest to consider either impute the 28 days censoring strategy as the primary efficacy endpoint, or consider the competing risk CIF method as the sensitivity analysis to provide the totality of evidence for the treatment effects evaluation in the COVID-19 trials.
Okay,
I'm not sure if it's provide some clarification for your concern and question.
Yeah, thank you, Hong, for sharing the experience. That's very interesting to know. And so we know a COVID-19 situation is, everyone is in such a rush to, getting things done, and that's a very valuable experience. Thank you for sharing.
Yeah, by the way, Taehyun, I noticed the subject stream model has been implemented in a lot of current COVID studies and published in the high profile journal.
Okay, good to know. Good to know.
Thank you.
Okay, let's see. I think we have a question here from the chat. So from Ahmed, so this is also for Hong. Can you differentiate between deaths related to toxicity versus deaths due to COVID-19, lack of efficacy?
Yeah, I think that's a great question. Actually, in our current research work, we didn't differentiate the death reason or underlying mechanism for death. The primary focus for this research work is to incorporate the competing death when we evaluate the positive event, like recovery or hospital discharge endpoint. But that's an excellent question. I think in the future research work, we will definitely further investigate the underlying death reason and the potential impact for the overall comprehensive evaluation for the treatment therapy effects on the positive events, such as a hospital discharge and the time to recovery.
Okay, thank you. Yeah, so and the audience, feel free to unmute yourself if you have questions. So don't forget to unmute yourself.
Yeah, I guess I have one question. This is Shura for Xing. I think you have mentioned that tight G-Boyne shortens the trial duration and what is your experience based on your simulation work, how that compares to the other competing methods that you have studied, especially the other one being that the tight Boyne that you recommended also together with tight G-buoyant?
Yes. So I think the answer to this question related to what is the studies, basically in my summer internship, I compare the tight design with the complete data design, and the main part for my summer intern project is we developed a Shiny R application to compare all these designs among the tight design and also the complete data design, and we run the simulation under the same specification for different design, and then we compare the operating characteristic. And the topic I present today is a little bit extensions, which we consider the toxicity grade. So I think which method we're gonna use is really depends on what the study design, such as in today's presentation, when we need to consider the toxicity grade, that means we cannot use the tight buoy-in design. We only can use the tight J-buoy-in designs, which will give us the better performance. So I'm not sure if I answer your question.
I think in a way, I think so. Generally, you answered, but I hope you are not driving at this time. So I just want to make sure that.
No,
okay.
I just on the way to pick up my kid. So,
okay.
So sorry about that.
No problem. No problem. Okay, thank you. Thank you very much.
Yeah, you're welcome.
And let's see if any other people have any questions. I think we have 1 or 2 minutes left.
All right, so I had a quick question for Hong. I may have missed this in the presentation, but when you use the Cox model and you censor for death, did that ever perform poorly in any situations, or is that, because I know that's a really simple approach, did that always perform well?
Yeah, I think that's an excellent question. Based on our results, actually, it does not imply the Cox proportion or regular survival analysis always yield the unpleasant results or biased results. But when the death rate goes up to moderate or high level, we will be more cautious for the censoring strategy and how to incorporate the incurrent death effect in the final estimation. But as you noticed that in the left-hand side panel, when the death rate is pretty low, like 5%, all analysis methods yields the very similar results. In that case, I would assume actually cost proportion model perform pretty well if the death rate is pretty high, pretty low. Or if we assume extreme scenario, the death rate is 0, then actually the Cox proportion and the Kaplan-Meier approach will produce the same overall survival as the competing risk CF-based method. So I think that will depends on the study setting parameters and the highly impacted by the death rate and the differentiate death profile in 2 treatment groups.
Thanks.
Yeah, thank you so much.
Yeah, so I think we are about right on time. So I will say thanks again to all the speakers and all the audience who stay until the last minute of the last session today, and give it back to Lee for closing remarks.
Yeah, thank you, Xiaotian. Thank you for all the speakers, and thanks a lot everyone for staying until the end of this Friday afternoon. It's a great meeting. Thanks a lot for everyone to dialing in, to participate, to ask questions. Very nice discussions for some of the presentations. And thanks again for our organizing committees to make this happen this year. I guess, just have a good weekend, everyone, and hopefully to see you all next year.